So hey students, this is Basavaraj Sir, your biology master teacher. Welcome to Vedantu Neet English students. I hope everyone is safe and sound and doing extremely well in life. So before we start even a single concept for today, quickly tell me in the chat if my voice is audible to you and the presentation behind me is visible to each and every one of you. So good afternoon students. I know this is the section which you have been waiting for. That is the entire one shot of your plant physiology. Now students, I'll be teaching you all the way from the basics to the advanced level so that you can solve every single question out there belonging to plant physiology. So all I want from your side in today's video is gonna be watch the video till the very end, because this is a very crucial moment in your preparation, because you cannot keep any backlogs or any type of negative things left. So this is the day when you say final goodbye, done and dusted to the plant physiology, the final revision for plant physiology. And if you're watching for the first time, this is going to be the video where you need to start. So my name is Basavaraj Sir. I'm your biology master teacher. Let's get started. Students, I want every one of you to like the video first. All of you mark your attendance. All of you quickly mark your attendance by liking the video, and as soon as possible share the video right now to each and every single one of your friends, because I know there are many students out there who think plant physiology is a major backlog. So can we start? Are you ready? Yes Laharika, yes Akalsha, yes Preeti, yes Ravindrani. Can we start, Chinnu? Can we start now? A quick green hearts in the chat and fire in the chat if you want to complete plant physiology today. Yes. Now students, again I'm reminding you to subscribe to the channel, because this is a channel where we are doing everything from scratch and we are making sure the entire preparation is continued, no gaps whatsoever. Now starting with a basic definition of your photosynthesis. Students, we'll be doing the entire 3 chapters of your plant physiology, starting with the photosynthesis in higher plants. Ready? Yes. Now tell me in the chat, what is the meaning of photo? Photo means light. All of you know photo means light, and synthesis means to produce. Yes. So we have a special type of organisms on earth called as plants, which are technically said to be autotrophs in nature. Yes, they are autotrophs in nature. Now what is the meaning of autotrophs, students? What is the meaning of autotrophs? Tell me in the chat. Autotrophs are the organisms which are able to produce their own food, are called as your autotrophs. So plants are technically your autotrophs or organisms. Yes. Now students, tell me in the chat if this is going to be a plant cell. This is going to be a plant cell. Now tell me in the chat, what does a plant cell require to do photosynthesis? Somewhere in the memory you will have, no, your class 6 teacher, class 7 teacher will have told you in the memory that for a plant to do photosynthesis, it requires CO2. Yes, all of you know this. A plant will require CO2. But what is the use of this CO2? This CO2 gets converted to glucose. Yes, this CO2 gets converted to glucose. That is the basic definition which is happening. But what if I tell you this CO2 is actually getting reduced to glucose? Now what is the meaning of reduction? Now tell me in the chat. Reduction means gaining of electrons or gain of protons. Yes, gain of electrons or gain of protons. With me? Now students, can you tell me, here we do not have any protons, here we have protons. So can I tell photosynthesis is a reduction process? Photosynthesis is a reduction process. Yes, all of you know that. Now tell me in the chat, do we water our plants? If you have a plant in your house, you would water it. Now what is the requirement of water? The answer is very simple, students. I told you here the CO2 is getting reduced to glucose. Now there is extra hydrogen here. Now where does extra hydrogen come from? The extra hydrogen is from the water. Water is your hydrogen or proton donor in case of your plants. In the case of your plants. Understanding? understanding. That's how you should learn plant physiology. With the basics, we'll start. Now, tell me in the chat, this is a plant cell. Here we have the sun. Now, sun is providing something called as sunlight, also called as your photons. So the plants are absorbing the light. Yes, plants are absorbing the light. Can I call it a endothermic reaction? Yes. Can I call it a endothermic reaction? It is a endothermic reaction. Yes. Now, in the case of plants, do plants convert inorganic substances which is present in the soil and make it organic? Yes. So plants convert what? Plants convert inorganic substances into organic substances. From inorganic to organic substance, the conversion is happening. So can I call photosynthesis a anabolic process? Can I call it a anabolic process? Yes, I can. Yes. Students, so what is photosynthesis? Photosynthesis is a process in which a particular plant is producing food. That is, it is undergoing reduction. It is an endothermic reaction, and also from inorganic substance, organic substance is being produced. Hence, it is also called as a anabolic process. With me, all of you? Yes, with me. Now, students, I already mentioned H2O is the hydrogen donor here. H2O is the hydrogen donor. Now, students, this is what we know. This is what we know. But where did we learn it from? Is there any experimental proof that this is what happens inside a plant cell? The answer is yes. That is, if you want to understand photosynthesis, you need to look at a few different experiments. Before we go to the experiments, let me tell you the final definition of your photosynthesis. That is, photosynthesis is a physico-chemical process. Photosynthesis is a physico-chemical process in which there's a physical reaction is happening. That is, exchange of gases. In the same, it is a chemical reaction also. That is, it is an enzyme mediated chemical reaction. Now, what are these enzymes? We will be discussing in your dark reaction. Okay. Now, students, plants are capable of synthesizing organic compounds by utilizing the light energy. That's what is the meaning of photosynthesis. Plants convert light energy into chemical energy by the process of photosynthesis. What is the importance of photosynthesis? Primary source of food on earth. That is, primary producers. In every single food chain, we find plants as your primary producers. Responsible for the release of oxygen. That is, the byproduct of your entire photosynthesis is going to be releasing of oxygen. Byproduct is going to be release of oxygen. Done? Yes, students. Now, students, let's understand some early experiments. What were the different scientists who worked on your plants, different type of plants? Starting off with the first experiment, that is going to be your variegated leaf experiment. Now, what is this variegated leaf experiment? The meaning of variegated means different types of leaf. That is, we have a leaf which has much more dark green in color nature, and we also have the light green region, dark green region, light green region. Now, what is the meaning of this? Meaning of this is, in the darker regions, there is presence of more chlorophyll. Yes. That is, when you do an iodine test, when you take iodine and you add it on a variegated leaf, wherever we have the darker regions, it's completely change color to blue-black. The darker regions change color to blue-black. Now, what is the conclusion from this experiment? The main conclusion which you derive from this experiment is that in this darker regions, there is production of starch, and also chlorophyll is important for photosynthesis. That is, presence of starch, that is violet black region, indicates photosynthesis has happened. Chlorophyll is necessary for photosynthesis. First experiment done. You should know the conclusion. In your examination, they will ask you directly the conclusion. They will not ask you anything else. They will only and only ask you conclusion. Done. The second experiment. The second experiment is called as your Moll's half-leaf experiment. Now, what is happening in this experiment? Listen to me very carefully, all of you. In this experiment, we take a leaf We take this leaf and half of this leaf is added in KOH. Yes, in KOH. When this half leaf is present in KOH, this particular KOH will absorb every single last CO2 molecule. Completely, it will absorb all the CO2. But in this region we have a lot of CO2. CO2 is present here. Here CO2 is completely absent. CO2 is present. If there is CO2 is present, will it result in the formation of starch? Yes. So when you do the same iodine test, we can see that in the exposed region, there is starch formation, and in the KOH region where there was no CO2, there is no starch formation. What does it tell you? It tells you that CO2 is essential for photosynthesis. That's all. Done. Second experiment, done and dusted. Every single one of you like the video right now, and every single time I finish one experiment, I want to see green arrows in the chat. We quickly are summarizing the entire experiments now. Okay. The next one is called as your bell jar experiment. The bell jar experiment was given by a famous scientist who discovered oxygen, whose name is Joseph Priestley. It was given by Priestley's experiment. Now, what does it tell you? It is a basic, simple experiment. Do you see a bell jar here? Yes, we see a bell jar. So your uncle Priestley told us that, in this, when the candle is burning, or when the mouse is present inside, the burning of candle and the mouse, they are actually damaging the air. What are they doing? They are damaging the air. So something was present in the air before. Now it is completely absent. It is not present anymore. And as soon as whatever was present in your air, once it is consumed completely, the candle will extinguish and the mouse will also die. But later he made a change in his experimental setup. Now what was this change? The change was that he added a mint plant inside. So whatever this burning of candle and this cute rat here, whatever they are damaging or whatever they're consuming in the air, that was replaced by the mint flower, the mint plant. Back then they did not know that there's something which is missing, which is being absorbed, which is being removed from the air. That was nothing but oxygen, right? So your burning of candle, that is combustion, breathing by the respiration, breathing by the rat, that is respiration. Both respiration and burning combustion are consuming oxygen, and the oxygen is replaced by the mint plant. Done. That is, hypothesis says that plant restore to the air whatever breathing animals and burning candle removed. That is, burning of candle and the respiration by the plant, by the rat, were consuming the oxygen. The oxygen is replaced by the plant. That's all. Done. Bell jar experiment done. Then we have your engine house experiment. Then we have the engine house experiment. Now what is this experiment, students? He did a similar setup. Basically he copied Joseph Priestley. Same setup. Whatever setup was used by Joseph Priestley, he used the same setup. But the main difference is that he did the experiment in sunlight, the same experiment in your shade. First time experiment in sunlight, same experiment in shade. He noticed that during the daytime there was release of gas bubbles, and during the night time there were no bubbles. What does it tell you? It told us that sunlight is important for photosynthesis. That is the conclusion. Then what did he do? See here. Conclusion: sunlight is essential. Bubbles formed around the green parts only, because he knew that in the green parts of the plant, that is the location where the photosynthesis is happening. Something is happening. Bubbles means oxygen. That's all. Done. Next experiment also done. Then we have your von Soch experiment. Now what did he tell us? He basically told us the location of your chlorophyll. He also told us that where is food stored as. That is, provided evidence for production of glucose by the plants. His study showed that the chlorophyll is located inside the chloroplast. So where do you find chlorophyll, students? Chlorophyll is located inside the chloroplast. And who told us that? It was von Soch. It was von Soch experiment. Done. Leaves synthesize glucose, which is later stored as starch. Students, you don't need to read the entire paragraph. Learn these 3 points and you're set. You're set. You do not have to even think ahead. Okay? So, von Soch experiment, done. The next one is your T.W. Engelmann experiment. The next one is your T.W. Engelmann experiment. Very easy again, students. Very, very easy. Listen to me very carefully. That is, we have a prism here. We have a prism. Now, on this prism, we are adding white light. White light. Now, this white light is being scattered. Yes, the white light is being scattered around. In the region, the PAR region is called as photosynthetically active radiation. Now, he noticed that he took a green color algae. The green color algae is called as your cladophora. So he took an algae here. It is not given here. Can you see? This is your algae. This is your algae. Now he noticed that this algae is able to do photosynthesis better in 2 particular wavelengths. That is at 400 nanometer as well as 700 nanometer. In these 2 regions, he noticed that plants were able to do better photosynthesis. But how did he come to that conclusion? He came to a conclusion with the help of bacteria such as your aerobic bacteria. That is aerobic bacteria. He noticed that if a plant is, you tell me in the chat, if the plant is doing photosynthesis, will it release oxygen? Yes, it will release oxygen. If plant is releasing oxygen, can aerobic bacteria utilize that oxygen? Yes. So technically, if plant is doing more photosynthesis here, it means more oxygen here. More oxygen here. If there is more oxygen here, it means more aerobic bacteria here. More aerobic bacteria here. Similarly, if the plant is able to do more photosynthesis in this region, more photosynthesis means more oxygen, it means more aerobic bacteria here. That's all. More aerobic bacteria. The confirmation was done with the help of the aerobic bacteria that 400 and 700 nanometer plants are able to do better photosynthesis. Can you see here? See, aerobic bacteria detects the site of your evolution of your detected as evolution of O2. Bacteria accumulates in the regions of red and blue light regions. Blue and red light wavelength optimum for your photosynthesis. That is one of the conclusions which we derive from your T.W. Engelmann experiment. And this was the one of those experiments which gave us your action spectrum for the very first time. Action spectrum for the very first time. You can read this, you will get it in the notes, don't worry. See, the conclusion is what? Aerobic bacteria detects the presence of O2. Formation of O2 highest in the red and blue light. That's why I told you. Blue and red light wavelength optimum for the photosynthesis. Done. One more experiment. Done. Next experiment is this is what we learned till now. That is, we need carbon dioxide photosynthesis. We learned that KOH experiment. We also know that water is the hydrogen donor. We know that, but experiment is still coming. We know that plants require sunlight, plants require chlorophyll, plants are able to produce glucose, and plants are releasing oxygen as the byproduct. One by one, we are actually proving your entire equation. We are proving the entire equation with the help of experiments, different type of experiments. Done. Now students, the next one is your Cornelis van Niel experiment. The next one is your Cornelis van Niel experiment. Now he was much more of a microbiologist. He was a major proton donor in your photosynthesis is water. See here, in the photosynthesis, he observed that the green and purple sulfur bacteria did not release oxygen during the photosynthesis. Now, what does this line tell you? In case of your green and purple sulfur bacteria, H2S is the hydrogen donor in the case of bacteria. But in the case of plants, H2O is the hydrogen donor. Now you tell me, in the plants, the byproduct is going to be oxygen. In the case of this green and purple sulfur bacteria, the byproduct is going to be sulfur. With the help of this method, he told us that the hydrogen donor in the case of your photosynthesis is water. Clear? Conclusion is, oxidation of water results in the liberation of O2. Yes, water is undergoing oxidation. Now, what is oxidation? Oxidation means loss of electrons or loss of protons. Yes, loss of electrons or loss of protons. So, in the case of your water, it is losing the H2 and it is oxygen is related as byproduct. Done. Water provided hydrogen in order to reduce the CO2 to carbohydrates. All of us know, I told you in the starting itself, CO2 is being reduced to carbohydrates, and this reduction process, the reducing agent, the hydrogen is coming from the water. Oxygen evolved from the green plants, come from the water and not from the CO2. Clear? Because the CO2 is getting converted to glucose. CO2 is getting converted to glucose. But water is providing what? Water is providing the electrons or hydrogens. Done. Very important line, students. NCERT line. I want everyone to mark this line and understand this line. And students, if you have any doubts regarding this today's topic, let me know in the comment section and I will clear it for you personally. Okay. Now he gave an equation, H2A plus CO2 will provide your H2A, H2O and H2O. Yeah, here A is going to be your sulfur or oxygen which is released. In the case of plants, oxygen is released. In the case of your purple sulfur bacteria, sulfur is released. Done. Experiments done and dusted. Now, let's understand the site of photosynthesis now. Can we start? Site of photosynthesis. Now tell me in the chat, where does the photosynthesis happen? If I ask you a question, if I ask any student, all of you will basically tell me photosynthesis happens in your green plants. Happens in the green plants. Now if I ask you specifically, students, if I ask you specifically inside green plants, where does it happen? Inside green plants, it happens in your leaves. You'll tell me, sir, all the photosynthesis happen in the leaves because it is green color. Now if I ask you specifically inside leaf where it happens, you will tell me mesophyll cells. You will tell me mesophyll cells. Yes, mesophyll cells. Now, but if I ask you inside mesophyll cell where it happens, you will tell me a site called as chloroplast. A site called as chloroplast. So technically, all the photosynthesis which is happening in the plants is happening inside the chloroplast. Now, students, open your book. Open your book. I know some of you are sitting without book now. Open your book right now and draw this diagram which will help you in your examination. Okay, draw with me, all of you. Now let's draw a structure of your chloroplast. Ultra structure of your chloroplast. Wow. If you look at the structure of your chloroplast, it is the double membrane structure. If you look at the structure of your chloroplast, it is double membrane. This is the outer membrane, and this is the inner membrane, outer membrane as well as the inner membrane. Now inside this chloroplast there are coin-like structure. There are coin-like structure called as thylakoids. Called as what? Thylakoids. Called as thylakoids. Now when we have a group of these coins, when we have many coins arranged like this, the entire stack of coins is called as grana or granum. Grana or granum. So imagine we have multiple stacks like this. We have multiple stacks inside the chloroplast. Multiple stacks. Many stacks are there. Now students, these stacks are interconnected. Yes, these stacks are interconnected. Now, what is this interconnection called as? When these many stacks are interconnected, this is called as your stroma lamellae. Stroma lamellae. Clear? Now, students, that is your stroma lamellae. Stromal lamellae or stroma lamellae? Stromal lamellae. Okay. Now, there is a matrix here. There is a matrix here. Now, what is this matrix called as? Inside the chloroplast, there is a matrix called as your stroma. Matrix called as stroma. Matrix called as stroma. Now, students, I want all of you to focus 100% now. 100% focus. I'll tell you very important lines which can help you in long run. Okay. That is, inside this thylakoid, students, on this thylakoid, there are something called as photosystems. What do we have? We have two photosystems here. We have PS2 and we have PS1. PS2 and PS1. Similarly, on the stroma lamellae, we also have for your photosystems. We have PS1. Now, that is very important to remember that PS2 is mainly present on your grana, and PS2 and PS1 are present on your grana, and PS1 is only present on your stroma lamellae. That is very important point. Please remember that. Now, students, next thing you need to know here is on the thylakoid, all the light reaction takes place. On thylakoid, all the light reaction happens, and on the stroma, inside the stroma, on the stroma, all the dark reaction happens here. All the dark reaction happens in the stroma. Now, what is the light reaction? What is the dark reaction? Do not worry. I will tell you in the next few minutes. Okay, done. This is the site of your photosynthesis. This is where the photosynthesis is happening inside the chloroplast. Hopefully, there should be no doubt here. Very easy. Are you understanding, students? If you're understanding, just say yes sir in the chat and I will look at the chat and I'll be happy. Okay. Now, see here, I told you already. This is your chloroplast, outer membrane, inner membrane. We have stroma, which is a matrix here. And we also have the coins. Each coin is called as thylakoid. The entire stack is called as grana. And the connection between the two thylakoids or two grana is called as stroma lamellae. Stroma lamellae. Together they are called as membrane system. And on this thylakoid, light reaction is happening. And in the stroma region, dark reaction is happening. Okay. Now, students, let's understand pigments. What are pigments and what type of pigments are involved in your photosynthesis? Now, all of you want to be scientists sometime? Yes. Let's be a little bit of scientists and let's see what is happening in the pigments. Now, if you take a leaf, you need to take a leaf. Take a leaf and completely crush it. Now, eat chutney, you make one. Chutney. Make a chutney like this and you pass it through a filter paper. Strain the extract. And once you get the extract here, the pure one here, take a drop of it and add it on this particular filter paper. Add it on this paper. Now, once you add on this paper, take this paper and dip it in a solvent. Now, once you add the paper into the solvent, what will happen? The solvent which is here, the solvent will move up the water, move up the paper. You notice, right? Take a piece of paper and add it water. What will happen? The paper will get wet, right? Basically, the paper here is getting wet with the help of solvent. But what is the difference here? The difference here is, as the solvent is moving up, it is taking along with it the different pigments. And finally, after some time, we get a complete set of pigments which is present in your leaf. That is gonna be your chlorophyll A, chlorophyll B, xanthophyll, as well as your carotenoids. These are different pigments which are responsible for your photosynthesis. Clear? This experiment is given in your NCERT. It is given, and this experiment is called as your paper chromatography. What is it called as? Paper chromatography. It is a method in which we can separate out pigments. A chromatographic separation of your leaf pigments shows that the green color of leaf is not due to the single pigment. The green color is not due to single pigment. It is many different pigments, that is, but due to the 4 pigments, that is chlorophyll A, chlorophyll B, xanthophyll and carotenoids. But tell me students, what is a pigment now? If you are my student, can anyone tell me extra information, little bit extra information for the concept to be clarified? Okay, one concept to be clarified. What is actually a pigment then? What is a pigment? Is it a hormone? Is it a chemical substance? Is it a a type of anything else? What is a pigment? If I ask you what is a pigment, what will come to your mind technically? When I say pigment, the only thing which is coming in your mind right now is going to be color, right? All of you might be thinking pigment means color, right? All of you might be thinking pigment means color, right? But sir, what exactly is a pigment? Can you tell us that? Yes, I can. Students, remember, what exactly is a pigment, students? Pigment is a chemical compound. Pigment is a chemical compound, that is which converts light energy into chemical energy. Pardon my cursive handwriting, that is my cursive handwriting, a little bit I write very fast, I write that cursive handwriting comes. Okay, so what is a pigment, students? Pigment is a type of chemical compound which converts your light energy into your chemical energy. But if I ask you, how does it work? How does the pigment actually work, students? I don't have a line, no? It's fine, right here only. If this is your pigment here, students, if this is your pigment right here, this is your pigment. Now on this pigment, a photon is landing on this pigment. Imagine a photon is landing on this pigment. What happens, students? What happens if the photon lands on this pigment? This is at fixed wavelength. At a fixed wavelength, a particular photon I land on the pigment. Whatever electrons which are present here, whatever electrons which are present inside the pigment, can the electrons get excited? Yes, they'll get excited. Now students, tell me in the chat, simple chemistry. When the electron is excited, does it come back to the ground state? Sometime it comes back, right? So when the electron is coming back, are there any different phenomena which is happening? If you're a chemistry student, you will tell me, sir, when an electron is coming back to the ground state, will it emit light? It will emit light. Will it emit heat? Yes, it will emit light as well as it emit heat. Or this particular energy which is there, can the energy be transferred to another pigment? Transfer the energy to another pigment. Yes, we can transfer the energy to another pigment, or we can use this energy to do some reaction, Yes, all these 4 possibilities can happen. All these 4 different possibilities are able to happen. But in this chapter, we are not focused in this, because this you will be learning in your chemistry. I am not going to teach that. Disha will teach you that. But here, these 2 reasons, that is transfer of energy to another pigment and use of energy to do some reaction, these 2 points which we will be discussing in today's class. Okay, that is the basic which you need to remember, because we will be using these 2 principles to explain reaction center, to explain you photochemical reactions and everything later on. Okay. Now students, let's understand one more concept called as your absorption spectrum. What is this absorption spectrum? Okay, clear? What is this absorption spectrum? Can anyone tell me in the chat if you remember what is absorption spectrum? I'll tell you one basic definition. After that, you will never forget what is absorption spectrum. Okay, start. Ready? Students, remember, absorption spectrum is a spectrum which tells you that at which wavelength a particular pigment is being absorbed. I'll repeat again. I can write it, but I have written material. I'll show you. Absorption spectrum is a spectrum which is showing you that which pigment is getting absorbed at which wavelength of light. That is your absorption spectrum. Example is going to be your chlorophyll A is getting absorbed at here and this region. Chlorophyll B is getting strongly absorbed at 450 nanometer. So each pigment is getting absorbed at different wavelength. That is your absorption spectrum. Clear, all of you? Clear? That is, pigments have ability to absorb light at specific wavelength. The absorption spectrum of leaf pigment indicates that wavelength of light absorbed by each pigment. So what is it telling you? The absorption spectrum is telling you, the leaf pigment indicates the wavelength of light absorbed by each pigment. Maximum absorption of your chlorophyll A is happening at your blue as well as red region. Regions. Clear, all of you? Is this concept clear to all of you? What is your absorption spectrum? Students, if you are not understanding, I have told it in a much more easier manner so everyone can understand. So keep rewind once again and listen to my words or write down my words and understand. Because that's much more easier than that, no one can tell you. That's such the basic definition of your absorption spectrum. But what is this action spectrum? What is the meaning of action? Action means work. Now, what is the work we are doing here? Not we the plants. The work we are talking here is photosynthesis. Yes, it is photosynthesis. That is the work here. Now, students, action spectrum is a spectrum which is showing you at which wavelength photosynthesis is happening. That is, at which wavelength photosynthesis is happening. This is your action spectrum. Done. Oops. Check. Sound check. Are you able to hear me? I think I pulled the cable too hard. Oops. Clear? Is this line clear to all of you? So you can see here, photosynthesis is higher in this region. Photosynthesis is also higher in this region. This is for your chlorophyll A. Okay, shows the effectiveness of different wavelength of light for a particular reaction. Clear? Now let's superimpose your action spectrum with your absorption spectrum. Can we? Yes. Now students, can you see here? Where is it? When we superimpose your absorption spectrum as well as your action spectrum, we get a graph like this. But if you notice here, between your 500 to 600 nanometer, can you see absorption is not happening. I should have used the NCERT line, but it's not. NCERT diagram is not there. But listen to me very carefully. NCERT diagram should have been there. It's not there. But open your NCERT right now. I think I missed out NCERT diagram here. If you open your NCERT diagram right now, you can notice between your 500 to 600 nanometer, there is no absorption. There is no absorption of your chlorophyll A. No absorption of your chlorophyll A. But is there photosynthesis happening? The answer is yes. Even though there is no absorption of your chlorophyll A, photosynthesis is still happening. What is the meaning of that? The meaning of that is there are other pigments. There are other pigments which are absorbing light and which are doing photosynthesis same time. Okay, they are not doing photosynthesis, they are actually providing the energy to chlorophyll A. So there are other pigments called as your accessory pigments. These pigments are absorbing the wavelength of light during 500 600, but are they doing the function? No, they are absorbing the light and they are giving the energy to your chlorophyll A to do the photosynthesis. That is the meaning of that. Okay, that is the meaning of that. Now students, let's understand photosynthetic pigments classification. Photosynthesis classification. Students, if you are able to follow till now and the last 45 minutes, you have understood so many concepts. Do you realize that? In the last 45 minutes, in similar fashion, in the same rate, we'll be finishing the entire plant physiology till now today. So I will request with each and every student to watch the video till the end if you want to completely finish your syllabus today. Okay, sit with me. Now students, photosynthetic pigment. We have two types of pigments. One is called as your chief pigment. The other one is called as your accessory pigments. Now what is this chief pigment and what is this accessory pigments? Think of chief pigment as your principal, as your school principal, and your accessory pigments as your school teachers, right? So in your class there would be some backbenchers who would make a lot of noise. What will your teacher say? Teacher will be like, I will give you TC, or go to principal room, right? They will tell, go to the principal's room. Why? Because students, remember, teachers will teach, but do they have the power to remove you from the school? No. That is, the power to remove you from the school is with the principal. Yes, with the principal. That is your chief pigment. Similarly, how your principal has the higher power, here chief pigment, which is your chlorophyll A, has the higher power. That is, only and only chlorophyll A, remember, only and only chlorophyll A is able to do photosynthesis. When I say photosynthesis, I mean enzymatic action. It is able to do enzymatic action. Why? Because your accessory pigments are like teachers. They will take all the energy and they will give it to the chlorophyll A. That's why they are called as accessory pigments. They absorb light and transfer it to the chlorophyll A. Those are your accessory pigments. Example is going to be your chlorophyll B, carotenoids and xanthophyll. Three of them are your accessory pigments. Are they able to do photosynthesis? No. But are they able to absorb the light? Yes. They are absorbing the light and they are giving to the chlorophyll A. Okay, chlorophyll A. The other function of your accessory pigments is going to be your protect chlorophyll A from photooxidation. Imagine if all the students from all throughout the school, if they directly go to the principal's room, will the principal be able to handle everyone? No. To make sure principal is able to handle all the students in the school, do we have separate class teachers? Yes. That is, class teachers are protecting the principal from all the backbenchers. Okay. That is, protect the chlorophyll A from the photooxidation, to make sure the chlorophyll A does not get exposed to a lot of sunlight, lot of wavelength of light at once. That's all. Now students, you need to understand what is a photosystem now. What is the photosystem? Ready? Listen to me very carefully. The pigments are organized. We just learned the pigments, right? All the pigments, chlorophyll A, chlorophyll B, carotenoids, xanthophyll, all the pigments are organized into two discrete photochemical light harvesting complex. So all the pigments are accumulated in a structure called as your light harvesting complex. I think I missed one point here. Students, this is also very important. This is also very important. There's a trick to remember this. If I remember this, Vibgyor, yes. Vibgyor. Vibgyor. That is, chlorophyll A is going to be from, this is a neat PYQ, I forgot almost. Students, chlorophyll A light is blue-green to blue-green in pure state. Blue-green is going to be your chlorophyll A. Blue-green. Then we have chlorophyll B, that is yellow-green. Can you see? Yellow-green is going to be your chlorophyll B. Chlorophyll B is going to be your yellow-green. Then we have carotenoids, that is your yellow. Then we have yellow to yellow-orange. Yellow to yellow-orange carotenoids. Then we have xanthophyll, which is yellow color. Xanthophyll, which is yellow color. That trick you can utilize. And you can understand that which pigment is present in which color. Now, why is this important? Because this is a neat PYQ, students. This right here is a neat PYQ. So remember that. Okay. Now, where were we? We were learning about photosystem now, right? Now, within the photosystem. So, students, what is photosystem made up of? If they ask you a question, what is photosystem made up of? Photosystem is made up of two different units. Photosystem is made up of two different units. One is called as your light harvesting unit, and we also have something called as reaction center. We have something called as antenna or light harvesting system. The other one is called as your reaction center. This light harvesting or system or antenna system is basically your accessory pigments. Can you see? Includes pigments that help to make the photosynthesis more efficient by absorbing different wavelength of light. That is your accessory pigments. Accessory pigments. What about your reaction center? Reaction center is like your main boss or principal. Chlorophyll A, the chief pigment is present. So all of you know what is your photosystem now. All of you know what is a photosystem. Clear? Students, remember this light harvesting systems as well as your reaction center, they together make up what? They together make up your LHC. Remember that. Light harvesting complex. Together they are called as light harvesting complex, reaction center as well as your antenna. They are called as light harvesting complexes. And both are present in your photosystem. And if you look at your NCRT and in your syllabus, or in the nature, we have two types of photosystem. One is called as your photosystem 2. The other one is called as your photosystem 1. Photosystem 2 is located. Starting I told you, remember? Starting I told you. I told you in the starting, this is your grana. This is also your grana. Between the two connections called as stroma lamellae. Stroma lamellae. This is your grana. This is your thylakoid. In the starting, did I tell you in the starting? Photosystem 2 is located in the stacked regions or oppressed regions. Oppressed means flat. Oppressed means flat. Oppressed regions of your thylakoid membrane. Students, we have photosystem 2 here and we also have PS1 here. What is a photosystem? Photosystem is a structural unit which has your light harvesting complexes such as accessory, the chief pigment. One is your reaction center, the other one is your antenna. Please remember this. What is photosystem? Photosystem is made up of antenna as well as reaction center. And here we have stroma lamellae. On the stroma lamellae we have your PS1. We have the PS1. Clear? We have PS1 on your stroma lamellae and PS2 and PS1 present on your thylakoid. Now with that, can we do a quick comparison between your photosystem 1 and photosystem 2? Can we start? Yes. Photosystem 1, it can absorb wavelength of 700 nanometer. And why we call it photosystem 1 because it was first discovered. First discovered. Based on the discovery, we call it photosystem 1 and photosystem 2. It can absorb wavelength of light of 700 nanometer. This can absorb the wavelength of 680 nanometer. So remember, photosystem 1 works on your 700 nanometer and photosystem 2 works on your 680 nanometer. 680 nanometer. Now, PS1 lies on the outer surface of thylakoid, while your PS2 occurs on the inner surface of your thylakoid. It is found in your grana and stroma lamellae. PS1, right? PS1 is found in both in your grana and stroma lamellae. Confusion. One second, students. One second, friends. One second. PS1 is found in your stroma lamellae only. Stroma lamellae only. What about your PS2? PS1 is found both, right? PS1 is found in both. Yes, PS1 is found in both. Correct only I wrote, no? Yeah, correct only. I got confused. I wrote PS2 is written here. Yes, PS1 is found in both grana as well as your stroma lamellae. We just learned, right? PS1 is present here also. PS1 is present here also. Done. What about your PS2? PS2 is only present on your grana lamellae. Okay? On your grana lamellae. This is your grana lamellae. This is your grana lamellae. Done. Now, what is the phase of photosynthesis? What is the phases of photosynthesis? The entire photosystem can be divided into two parts. That is light dependent reaction. It is called as light dependent reaction. And one other one is called as your light independent reaction, also called as your dark reaction. So the entire photosynthesis is divided into two parts. One is your light dependent, also called as your light reaction. Other one is your light independent reaction, also called as your dark reaction. Now, does this mean dark reaction happens in your dark and the light reaction happens in the light? No. This is merely light dependent because it requires light. But the meaning of dark reaction is that it is light independent. It does not require light. It needs product of a light reaction. The dark reaction requires product of a light reaction. It doesn't require a light, that's all. Clear? It occurs within the membrane of your chloroplast. Trapping of light energy by the pigment, synthesis of your ATP and NADH is used. So, remember, light reaction is happening on your thylakoid, while your dark reaction is happening in the stroma. Dark reaction is happening in the stroma. Clear? Now, what is dark reaction? It is utilizing of sugar by using ATP and NADH. Synthesis of sugar by using the ATP and NADH. Let me briefly tell you, in light reaction, the products are formed using the light energy. Light energy is being trapped and ATP and NADH is produced in your light reaction. In your dark reaction, there is formation of sugar. In your dark reaction, there is formation of sugar as well as your using of ATP and NADPH. Using ATP and NADPH. And if you talk about light reaction pathway, the entire light reaction pathway can be divided into two parts. The one is called as your non-cyclic photophosphorylation. Non-cyclic photophosphorylation. The other one is called as cyclic photophosphorylation. What is the difference? What is the difference between two? The main difference between the two is non-cyclic is not continuous. Once the electron is gone, it is lost. In the case of a cyclic photophosphorylation, the electron is in a cyclic manner. The electron is being cycled back. Electron is being cycled back in your cyclic photophosphorylation. Now, can we draw the entire thing and learn once? Because a lot of questions have come from here. So, can we draw once? Yes. Can we start with your non-cyclic photophosphorylation? Can we start, students? I want to see some energy in the chat because this is a concept which has, troubled so many students out there. Many students out there it has troubled. So when you do the diagram of your entire non-cyclic photophosphorylation, you will understand how many photosystems are present here, what is happening in a sequential manner. So I want all of you to open your book and start writing. Open your book and start writing. Okay. Now students, remember, in your non-cyclic photophosphorylation, the main photosystem is going to be both. That is, we have the let's do it here. This is your chlorophyll A, which is the reaction center plus antenna. This is the reaction center plus the antenna. Together they are called as what? Reaction center plus antenna. Together they are called as your photosystem. Now this photosystem is photosystem 2. Now tell me in the chat, photosystem 2 works at which wavelength? Photosystem 2 works at around 680 nanometer. It works at 680 nanometer. Now what is happening here? We have the sun. We have the photons are traveling here. Photons are being trapped. The photons is being trapped by your auxiliary pigments and also your chlorophyll A. Yes, sir. Yes. Now, students, all the energy will be sent to your chlorophyll A. Yes. All the energy will be sent to chlorophyll A. True. Now, all of you know chlorophyll A is a which type of pigment? It is a pigment. So can the electrons in a pigment be excited? Yes. So what happens here? The electron which is present here gets excited. That is, the electron is getting excited. Two electrons are being excited here. Now this, who is catching these electrons, students? Who is the person who is catching the electrons? The person who is catching the electrons is called as your primary electron acceptor. It's called as what? Primary electron acceptor. Now, who is this primary electron acceptor? Does he have a name or does she have a name? Yes, the name given for your primary electron acceptor is going to be your pheophytin. The primary electron acceptor is going to be pheophytin. Now, from electron from the pheophytin will be sent to PQ. We send to the PQ. What is PQ? PQ is plastoquinone. PQ is plastoquinone. Clear? Plastoquinone. Now from PQ, the electrons will be sent to you. CY. Now what is the CY? CY is your cytochrome BC1 complex. Cytochrome BC1 complex. Now from CY, the electron will be sent to PC. Electron will be sent to PC. Now what is this PC? PC Priyanka Chopra, right? No. PC is your plastocyanin. PC is your plastocyanin. So the electron is being transferred, jumping. Electrons are being jumped here. Two electrons. Done. Now all of you should notice here, PQ to CY. In this PQ to CY, 1 ADP is getting converted to ATP. So there is formation of 1 ATP here. Formation of 1 ATP between PQ and CY. Now from here, PC, the electrons are now sent to one more photosystem. Sent to one more photosystem. This is your chlorophyll A. Now this photosystem, that is reaction center plus the auxiliary pigments or the antenna, this is called as your photosystem 1. This is called as your photosystem 1. Photosystem 1. PS1. Why written as 8? 1 second, friends. This is called as your PS1. The PS1 works at around 700 nanometer. The PS1 works at around 700 nanometer. Now again, can the electrons be excited here? Yes, the electrons can get excited. Now again, the electrons here, the two electrons are being excited. Can they be taken up by your primary electron acceptor here again? The primary electron acceptor is going to be your FES. From FES, the electron sent to FD. Electron sent to your ferredoxin. Now from ferredoxin, the electrons are sent to your NADP. The NADP, when it takes the two electrons, the NADP, when it takes the two electrons plus 2H plus from the stroma region, 2H plus from the stroma region plus 2 electrons from here, the NAD plus NADP, the NADP gets converted to NADPH plus H plus H plus. That is, it is gaining electrons, it is gaining protons, it means it is getting reduced. It is getting reduced. If I ask you a question, which is the final electron acceptor in the case of your non-cyclic photophosphorylation, your answer should be NADP. Should be NADP. Clear? Now students, for this conversion to happen, there is a enzyme here, ferredoxin reductase, ferredoxin NADPH reductase. Clear? What is that? This thing. Ferredoxin NADPH reductase. I'll write it down. Clear? Ferredoxin NADPH reductase, FNR. Now I'll make you write down a few points here. Now I told you the electrons are sent here. Electrons is going here, electron is going here. The two electron is being traveling. Electron is being traveling everywhere. Now you can see one thing. If the electron is finally reaching here, electron is finally reaching there. For this cycle to continuously happen, will they require continuous source of electrons? Yes, electron needs to be coming sent here. Now, how do we get the continuous source of electrons now? What is the trick here? There's no trick here, but there is a process called as your photolysis of water. Now, what is this photolysis of water? Here, water is getting photolyzed. It is 2H plus O2, right? 2H plus 2A2 plus 2 electrons. Now, these 2 electrons are sent here. What about this 2H plus? This is sent to the lumen. This is sent to the lumen. What about this O2? The O2 is released outside the thylakoid. This is released out. It is released out. Clear? It's called as photolysis of water, also called as splitting of water is happening from in the photosystem 2. Now let's write down some few basic points which we understand from this particular non-cyclic photophosphorylation. The first thing which you need to notice here is, the first point I want all of you to understand here is O2 is evolved here. Yes, we can see there is a release of oxygen here. Then there is photolysis of water. That is, splitting of water is happening here. Yes, splitting of water is happening here. Then there is ATP formation is happening here. Yes, can you see here? ATP formation is happening here, and formation of NADP is also happening here. Formation of NADP is happening here. Now, this is non-cyclic in nature. Yes, it is non-cyclic in nature. And does this look like a Z? Does this look like a Z? This is called as your Z scheme. Called as your Z scheme. They're called as what? Z scheme. Done. That is your entire non-cyclic photophosphorylation. What happens in the first? The chlorophyll is absorbing the light, electrons being excited. From here, the electron is going to pure fighting, then PQ to CY to PC. PQ is going to be your plastoquinone. CY is going to be your cytochrome B6 or C1 complex. PC is going to be your plastocyanin. Then we have PS1, 7 nanometer. Electrons given to the FES, then FD, then to NADP. NADP is getting reduced to your NADPH plus H plus H plus. And to the continuous supply of electrons, there is photolysis of water is also happening at the same time. Photolysis of water is happening at the same time. This is your complete non-cyclic photophosphorylation, and if you get any question from here, you should be able to answer. You should be able to answer. Now let's understand cyclic. Can you understand cyclic photophosphorylation? Again, very simple. Now in cyclic photophosphorylation, we only have one photosystem. Now this is your reaction center, chlorophyll A. A reaction center will have antennas. A reaction center will have antennas. So reaction center plus the antenna, they are together, they are called as what? Together they are called as photosystem. Now here the photosystem is going to be your PS1, which works at around 700 nanometer. 700 nanometer. And where do you find PS1 here? Now this only PS1. The only time where the PS1 is present, it is found in your stroma lamellae. Stroma yes, stroma lamella. Now here electrons are being ejected out. Yes, when the photon is incident, when light is coming, the electrons are being ejected out. Similarly, now here again we have primary electron acceptor. The primary electron acceptor is going to be your FES again. From FES, electron sent to your FD. Right? Similarly, how we learned here, FES to FD. Now from FD, it doesn't go to NADP now. From FD, it doesn't go to your NADP, but it goes to your PQ, CY, PQ, CY as well as PC. Do you see this? From FD, we have the same PQ, CY and PC. The same thing is happening here. From FES, FD, PQ, CY, PC. All of you know what is PC? Plastocyanin. Now from plastocyanin, the electrons are sent back to the plastocyanin. The electrons are sent back to the photosystem. Sent back to your photosystem. But is there any amount of ATP production here? The answer is yes. Here in this step, 1 NAD is getting converted to 1 ATP. Yes. And similarly, between your PQ and CY, 1 NAD, ADP is getting converted to your ATP. ADP is getting converted to ATP. Clear? This is your entire of your cyclic photophosphorylation. Entire of your cyclic photophosphorylation. I hope there should be no doubt. I don't know why students, some of you still face difficulties, but this is the easiest concept which is out there. There should be no confusion regarding the cyclic as well as non-cyclic photophosphorylation from now onwards. Clear? All of you know now, right? Do not make the same mistake in exam, students. They can ask you questions based on this. They can also ask you in PS1 which photosystem is present here. In your PS1, which is the primary electron acceptor here. Where is ATP being produced? Totally 2 ATP. All of you know now. All of you know where ATP is being produced. So students, this is how you learn entire plant physiology without any difficulties. You can finish everything. Okay. Now let's see a quick difference. Let's see a quick difference. That is, in cyclic photophosphorylation, which we just learned, electrons move in a cyclic pattern. What about in the case of your non-cyclic, electrons in a linear pattern? That is, in non-cyclic Z scheme, the electrons are never sent back. Only photosystem 1 is involved. Both photosystem 1 and 2 is involved. Electrons are first expelled from your reaction center 1. Because we only have one reaction center, electrons are being sent there. Electrons are being sent there. Electrons are first expelled from your reaction center 2. Reaction center 2, right? Sir, write all primary electron acceptors separately. See here, I think it's given here. It's given here. See, electrons return to your photosystem 700 after passing through the electron transport system. They go through the electron transport system. They're coming back to your P700 in the case of cyclic. But in the case of non-cyclic, electrons do not return to your P680, but electrons will go where? The electrons are finally going to your NADP. NADP, right? NADP. Next, in cyclic photophosphorylation, synthesis of ATP during the light reaction of your photosynthesis by cyclic passage of electron to and from the P700. I think it's the same definition again. Occurs in isolated chloroplast and photosynthetic bacteria. Very important line. So your cyclic photophosphorylation mainly happens in your bacteria. What about your non-cyclic, which happens in most of your algae, higher plants and cyanobacteria? It occurs in your anisogenic photosynthesis. That is, no oxygen is evolved. I told you, right? In the case of cyclic condition, there is no evolution of any form of oxygen. But in the case of your non-cyclic photophosphorylation, there is release of oxygen because of splitting of water. The final electron acceptor is going to be your P700. That is, it's coming back, right? It is coming back in a cycle. 700. The final electron acceptor is going to be your NAD+. NAD+. The first electron acceptor in your cyclic is going to be your FES. FES in the case first, also called as your primary electron acceptor. The primary electron acceptor in the case of your non-cyclic is going to be your pheophytin. It's going to be your pheophytin. Done. Will you ever make any mistake in your cyclic and non-cyclic photophosphorylation? Never. Done for your life. Okay. Now, can we start with chemiosmotic hypothesis now? Can we start with chemiosmotic hypothesis? Yes, we can, students. Students, don't think of chemiosmotic hypothesis as a very big, very big term. It is the most simplest concept ever. The most simplest concept ever. Now, what is this chemiosmotic hypothesis? Let me tell you in a very simple manner. This is a hypothesis which explains the production of ATP. That's all. Which explains the production of ATP. See, here I told you, in chloroplast, ATP is being produced. I told you in the case of your non-cyclic photophosphorylation, there is production of ATP. But how? This is where chemiosmotic hypothesis comes into picture. Chemiosmotic hypothesis explains how ATP is being produced. Clear? Let's see the definition here. Synthesis of ATP is called as your phosphorylation. All of you know that. Synthesis of ATP in the presence of light during the photosynthesis is called as your photophosphorylation. ATP is synthesized during cyclic and non-cyclic photophosphorylation. Chemiosmotic hypothesis explain the mechanism of synthesis. The mechanism is explained with the help of your chemiosmotic hypothesis. Can we start? So before we start the hypothesis, I want everyone to write down the basic requirements. The basic requirements. There are 4 basic requirements for the hypothesis to work, for the chemiosmotic hypothesis. The first point here is going to be your proton gradient. The second one is going to be your intact membrane. The third one is going to be your proton pump. Proton pump. Proton. Proton pump. The fourth one is going to be your ATP synthase. That is F0, F1 complex. Done. What are the basic requirements here? The basic requirements for a particular chemiosmotic hypothesis to happen is we need a proton gradient, we need intact membrane, we need a proton pump, we also need ATP synthase. Now, let's understand what is your hypothesis. Now, if you look at the diagram, if you look at this diagram, what do you think this is? What do you think this is right here? Students, this diagram right here is actually magnification. It is actually the magnification of one thylakoid. What is this? This diagram right here is a magnification of one thylakoid here. Can you see? This is one thylakoid. This is the magnification of one thylakoid. So what are you seeing in thylakoid membrane, students? What do you notice in this thylakoid membrane? Can you see here? PS2, PS1, light is being transferred here, photolysis of water. Basically, in this first part, they are explaining you. In this first part here, or this particular part, they are explaining what? Non-cyclic photophosphorylation. This is your non-cyclic photophosphorylation, right? That is, light is coming here. Electrons are being excited. That is PQ, plastoquinone. Then it is going to cytochrome BCX complex. Then PC. From PC is going to your photosystem 1. Then it is going to FD here, to FNR, and NADP is getting converted to NADPH, right? NADP is getting converted to NADPH. This is your non-cyclic photophosphorylation right here. See here. Same thing we wrote, right? Same thing we wrote. Where is it? See here. PQ, CY, PC, chlorophyll A, FES, FD. See here. PQ, CY. This is CY, right? Cytochrome BC1 complex, B6F complex, PC, and as well as your photosystem 1, FD and FNR. So when this reaction is happening, in non-cyclic photophosphorylation is happening, there is utilization of your H+. So basically, you're trying to identify in which location proton gradient is happening. Now, what is this proton gradient? Proton gradient is basically the difference in proton. You're basically trying to establish a proton difference between your stroma as well as your thylakoid lumen. This is your thylakoid, right? Inside the thylakoid lumen. See here, inside part. This is thylakoid lumen. This is your thylakoid lumen. So you're trying to establish a proton gradient between your stroma as well as your thylakoid lumen. Now, how do we make this difference in your proton gradient? The first step is photolysis of water. Also your oxidation of water or splitting of water. In the splitting of water I mentioned earlier, oxygen is released as a byproduct. Electrons are sent to your photosystem. What about the H+? I told you the H+ is sent to where? The H+ is sent to the lumen. So can I tell H+ concentration is increasing in the lumen? Done. Now students, when we one more thing which you know here is PQ, plastoquinone. This PQ is doing what? This PQ is taking H+ from the stroma and it is sending it inside. It is sending it inside. It is getting plastoquinone, that is PQ to PQH and back to PQ. That is when the electrons are being circled here, it is taking a proton from the stroma and it is sending into the lumen. So can I tell again, H plus concentration is increasing again. We have a hydrogen donor here, hydrogen donor. Now one more thing you should understand here is, in the last step I told you NADP is getting converted to NADPH, and I told you in that reaction 1H plus is used from your stroma region. H plus is used in the stroma region to make the NADPH, for it to reduce. So can I tell the H plus concentration is reducing in your stroma? H plus concentration is reducing in the stroma? Yes. That is how we establish the proton gradient. 3 steps. Please write down 3 steps. Please remember the 3 steps. The first one is going to be your splitting of water. H plus is increasing. H plus is increasing. Second one again, H plus is increasing because of your PQ. The third one is your H plus is decreasing in the stroma because H plus is being utilized for formation of your NADPH. 3 points done. That's how we define the proton gradient here. Now once we have enough of protons, once we have enough of proton gradient here, what happens? Can you see this structure called as your ATP synthase? ATP synthase structure here, right? This ATP structure, what will happen here is all these protons will enter here. All these protons will enter here, and once protons are entering here, what happens? There's a F0, F1 ring. There is F0 and F1. This ring will rotate. This ring will rotate. When this ring is rotating, ADP is getting converted to ATP. ADP is getting converted to ATP. You see here? And H plus is coming out. That is, H plus are required. H plus are required for the 3 H plus are required for turbine movement. 1 H plus is required for carrying the phosphate from the stroma region. And finally, ATP is being produced here with the help of proton gradient. Clear? Clear? That's how you should understand the chemiosmotic hypothesis. Chemiosmotic hypothesis. Now every single point I told you is already written here. Every single point is already written here. You can go through these points and you can understand it in a much more better manner. Okay. Now let's start a dark reaction, all of you. Let's start with your dark reaction. Now what was happening till now, students? Till now, whatever products which we have, because of your thylakoid membrane, it was absorbing the light, we got ATP, we have NADPH. Now this ATP as well as your NADPH is completely utilized in your dark reaction. That is also called as your biosynthetic phase, also called as your Calvin cycle. It is also called as your Calvin cycle, also called as your C3 cycle. Also called as your C3 cycle. Here light is not required, requires the products of your light reaction, that is your NADPH as well as ATP, as well as your ATP. Okay, as well as your ATP. That is, the dark reaction is also called as your PCR cycle. Also called as what? PCR cycle, that is photosynthetic carbon reduction cycle. Carbon from the atmosphere is fixed into the sugar molecule. So whatever carbon dioxide is present in the air, the carbon dioxide is entering into the stroma, and the carbon dioxide is converted, packaged and delivered in the form of glucose. So all the carbon is becoming now what? Glucose now. In your dark reaction, the glucose molecule is further reduced to form carbohydrates. Carbohydrates. Clear? See, what do we have here? The Kelvin cycle occurs in the stroma region. We know that already. The aqueous fluid surrounding the thylakoid, that is the stroma. That CO2 enters the cell through the stomata. That is, a particular leaf stomata is there. CO2 is entering inside now. Imagine CO2 is entering inside a cell through stomata. Okay. What are raw materials required? The raw materials required in your C3 cycle are going to be ATP, NADH as well as CO2. ATP, NADPH as well as CO2. Students, your entire dark reaction is made up into three different phases. The first phase is called as carboxylation, in which CO2 is used. Second one is called as your reduction, in which ADP and ATP are used. The third one is called as your regeneration, again in which ATP is again used. So remember, three steps, all of you, repeat after me. Three steps are going to be your carboxylation, as well as your reduction, as well as your regeneration. Clear? Can we draw a cycle now? All of you? Students, quickly like the video. I want everyone to like the video because the next cycle is going to be the most important cycle, which has been asked repeatedly. Questions have come from here. That is going to be your Calvin cycle. It's going to be your Calvin cycle. So what is happening here is we have a ketone sugar called as your RUBP. We have a ketone sugar here called as RUBP. Now what is this RUBP? It's called as your ribulose 1,5 bisphosphate. 1,5 bisphosphate. Ribulose 1,5 bisphosphate. 1,5 bisphosphate. Now this is your 5 carbon, a type of your ketone sugar. Then to this RUBP, what happens is, to this RUBP, a molecule of your CO2 is entering. To this particular RUBP, CO2 is getting attached. When RUBP is 5 carbon plus 1 carbon, should we get a 6 carbon molecule? See, we have 5 carbon here plus 1 carbon. Should we get 6 carbon molecule? Answer should be yes, 6 carbon molecule. But are we getting a 6 carbon molecule? No. We are getting a 3 carbon molecule called as your phosphoglyceric acid. We are getting 2 molecules of your PGA. Which is a 3 carbon compound. The first step in which RUBP gets converted to your PGA is called as your carboxylation. It is called as what? Carboxylation. Clear all of you? The enzyme which is involved in your carboxylation is called as your Rubisco enzyme. Clear? Now students, listen to me very carefully. This PGA now gets converted to triose phosphate. When this PGA is getting converted to triophosphate, 2 molecules of ATP and 2 molecules of NADPH is used. Two ADP. Similarly, how many? 2 again. So in the second step is called as your reduction. In your reduction step, PG is getting converted to triose phosphate, and when this is happening, 2 ATP and 2 NADH are completely utilized. Clear? Now we have triose phosphate. Now we have the triose phosphate. Now this triose phosphate gets converted to your glucose, which is stored as a starch in your plants later on. Now this triose phosphate again gets converted to your RuBp. Again gets converted to RuBp. In this process, again 1 ATP is converted to 1 ATP is converted to 1 ADP. ATP is being converted to ADP, and this last step is called as what? The last step is called as your regeneration. Called as your regeneration. Done. This is your entire cycle of your C3 cycle, also called as your Calvin cycle. Also called as your Calvin cycle. Now you can ask me question, sir, why is it called as your C3 cycle? Because the first stable product here, can you see? The first stable product is a 3-carbon molecule. Yes, 3 carbon. That is why it's called as your C3 cycle, also called as your Calvin cycle. So whatever ATP and NADH which are produced in your light reaction are used here to produce the starch or glucose Clear? Now here one more thing you need to understand is how much of for one CO2 to fix how many ATP and NADH is required. To fix one CO2 we require 2 ATP here and 1 ATP here. So in total of 3 ATP is required to fix one CO2. Similarly, we also require 2 NADPH to fix one CO2. This is your one CO2. Clear? To fix one CO2 we require 3 ATP and 2 NADH. Now if I ask you a question, how many ATP are required to fix one molecule of glucose? To fix one molecule of glucose, this cycle has to run how many times? In glucose, how many carbon are there? C6H12O6. So there are 6 carbon there. Yes. So this cycle has to run 6 times to make one molecule of glucose. And in one molecule of glucose, how many ATP and NADPH required? Just multiply into 6, you will get your answer. Clear? That's going to be your 18 ATP, 18 and 12 NADPH as well as 12 NADPH. Done. See here, 6 CO2, 1 glucose, 18 ATP and 12 NADH. And the plants, students, and the plants in which the cycle occurs, example is going to be your wheat, soybean as well as your oats. Wheat, soybean, oats are the examples for your C3 plants in which the C3 cycle is running. Now students, I told you about your RuBp, right? I told you about Rubisco. Now in this Rubisco, what is the CNO? The CNO is carboxylase as well as your O is your oxygenase. Carboxylase as well as your oxygenase. That is, this particular enzyme Rubisco can actually fuse with oxygen. Now what happens if it joins with your CO2, C3 cycle will run. Now what will happen if Rubisco enzyme joins with your oxygen? Then the C3 plants will not go under C3 cycle. They will undergo something called as C2 cycle. If the Rubisco enzyme is fixing with oxygenase, if fixing with your oxygen, the particular plant will undergo C2 cycle, which is also called as your photorespiration. What is happening in photorespiration? It occurs in your C3 plants. O2 is consumed and CO2 is released in the process. Rubisco oxygenates RuBp. Unlike your C3 cycle, where Rubisco is fusing with your CO2 and PGA is formed in your dark cycle, we just learned. But in your photorespiration, Rubisco is fusing with your RuBp. And when does this happen? When is this happening? there are two main or three main reasons. That is, Rubisco is fusing with your ROBP when there is high temperature or low CO2 or very high O2 or very high O2. Clear? So oxygenase photorespiration will happen when? Photorespiration will happen when there is low CO2 to O2 ratio and very high temperatures, or when there is very high temperatures. So what is happening in your photorespiration? RUBP is fusing with oxygen to produce PGA, which is a 3 carbon. So all of you know RUBP is 5 carbon. Here is no carbon. So technically we should get a 5 carbon compound here. So if your PGA is 3 carbon, plus we get something called as PG, which is 2 carbon. So all of us know PGA is phosphoglyceric acid, 3-phosphoglyceric acid. What is this PG now? Is it the PG which you stay in? No. What is this PG? The PG here is phosphoglycolate. The PG here is going to be phosphoglycolate. Phosphoglycolate. Clear? Now what is happening in your photorespiration? In a photorespiration, this phosphoglycolate will undergo many changes. This phosphoglycolate will go under many changes. Later it will again form PGA. Again form PGA. Now here, in the case of your, when your PG is getting converted to PGA, there is no sugar formation. There is no sugar formation at all here. There is also no ATP formation. There is absolutely no ATP formation. But ATP is being consumed here. ATP is being consumed here. At the same time, students, CO2 is being evolved. Plants require CO2. Plants will require CO2, but what is happening here? CO2 is actually being released outside here. And sugar is not produced here. ATP is not getting not produced. ATP is being consumed here at the same time. ATP is not being consumed here. Now you can tell me, is this a useful process or is it a useless process? Obviously, students, if there is no sugar, there is no ATP, ATP is being consumed, oxygen is being evolved. Can plants even use this? No. It is also called as your wasteful process. It is also called as your wasteful process. Clear? So if you notice here, no carbon fixation occurs, rather there is the release of CO2 within your utilization of ATP. That is why it's called as your wasteful process. Again, when does the C2 cycle occur? Are there any specific time when the C2 cycle will occur? Yes, whenever the O2 concentration is very high, or when there is very high light conditions, this will happen. Similarly, when there is very high temperatures, and also during the low CO2 conditions. When oxygen is present high, when there is high light or high temperature or low CO2, a plant will undergo. Which type of plant? C3 plant will undergo something called as your photorespiration. Photorespiration. Clear? Now students, can a plant, be like, no, I don't want to do photorespiration. Is that an option? Tell me in the chat. Is there any option in which a plant will be like, no, I don't want to do photorespiration? Can a plant be very smart? Yes. Some plants are very smart. Those plants are called as your C4 plants. C4 plants are so special that they have separate mechanism. They have a very separate mechanism to counter the photorespiration. Now, how do they counter the photorespiration? With the help of a special structure called as your Kranz anatomy. Now, what is this Kranz anatomy which is found in your C4 plants? In your Kranz anatomy, if you notice, in the middle we have the bundle sheet cells. In the middle we have vascular bundles. The vascular bundles are completely covered by your bundle sheet cells, and bundle sheet cells are completely covered by your mesophyll cells. Now, bundle sheet cells are arranged in the form of wreath here. This is called as wreath in a much more circular manner. It's called as wreath. That is, Leaf anatomy of a C4 plant, which is Kranz anatomy. Bundle sheet cells are arranged in the form of wreath around the vascular bundles. Here, can you see? The vascular bundles are arranged in a wreath-like structure around the vascular bundles. Bundle sheet cells are characterized by having a large number of cytoplasm, thick-walled, and no intercellular spaces. Now you tell me, if the bundle sheets are so thick, and as well as your bundle sheets are so thick, can the oxygen enter inside? Tell me in the chat, can the oxygen enter inside your bundle sheet cells? No. That is what is called as Kranz anatomy. Kranz anatomy made sure there is no oxygen at all whenever Rubisco is present. If Rubisco has only CO2, it will be forced to do only C3 cycle. And they're making sure the oxygen is not coming inside. Okay, oxygen is not coming inside. Clear? Your C4 plants can tolerate high temperatures. They respond to high light intensities. They lack photorespiration. Very important line. That is, photorespiration is completely absent in the case of your C4 plants. Great productivity of biomass. Very great productivity of biomass. And this C4 pathway was also called as your Hatch and Slack pathway. Why? Because the C4 pathway was discovered by your Hatch and Slack. That is, the two Austrian scientists, Marshall D. Davidson, Hatch and Slack, they found it. They found that the tropical plants are much more efficient in your CO2 utilization. They're very smart. That is, they have two types of cells. One is your mesophyll cell, other one is your bundle sheet cells. Bundle sheet cells as well as your mesophyll cells. Clear? Now, what are the different phases here? We have initial fixation, transportation, final fixation, and regeneration. Now, this can we understand with the help of a diagram? Can we? Let's understand C4 cycle with the help of a simple diagram. Yes, students. With the help of a simple diagram. Here we have two types of cells. Here we have two types of cells. One is called as your mesophyll cell. One is called as your bundle sheath cells. One is called as your mesophyll cell, other one is called as your bundle sheath cells. Now tell me in the chat, can the oxygen enter the bundle sheath cells? Can the oxygen even enter the bundle sheath cells? The answer should be no, they cannot enter. Why? Because the bundle sheath cells are in a wreath-like structure which has no intercellular spaces. If there is no intercellular spaces, can the O2 even enter inside? No, O2 cannot enter inside. Clear? Now let's understand what exactly is happening in your C4 cycle. Okay. Now, as usual, let's take different color. As usual, CO2 is entering inside. Yes, CO2 is entering inside your mesophyll cell, entering inside the mesophyll cells. Now this CO2 here in the case attaches to a structure called as PEP. Now what is PEP? Phosphoenol pyruvate, which is a 3-carbon compound. Yes, it is a 3-carbon compound. Also it's called as your phosphoenol pyruvate. Do you remember this PEP in your, you will study this in your glycolysis. You will study this in glycolysis later on. Okay. Now this PEP, when CO2 fixes with PEP, it results in the formation of a 4-carbon. See, 3 plus 1 is 4. 4-carbon compound called as your oxaloacetic acid. OAA. Oxaloacetic acid. Now if I ask you a question, which is the first stable product in the case of your C4 cycle, the answer should be OAA. That is 4-carbon compound, which is oxaloacetic acid, which is the first stable compound. Now here the reason it is C4, that is 4-carbon, that is why it's called as C4 cycle. That's why it's called as your C4 cycle. Now this OA gets converted to malic acid. And one more thing you need to remember here is the enzyme, that is your pepcase enzyme, also called as your pep carboxylase enzyme, which helps in the conversion, Clear? Now we have malic acid. Now this malic acid is entering your bundle sheets now. Converted to malic acid. Malic acid is entering your bundle sheet now. Till here everything is clear. Now this malic acid gets converted to OAA, which is 4 carbon. Now this OA gets converted to pyruvic acid. Pyruvic acid, all of you know, it is 3 carbon. Yes, it is a 3 carbon compound. So 4 carbon to 3 carbon means 1 carbon is released outside. Now what happens to this carbon? Now this carbon will enter a separate cycle called as your C3 cycle. So inside your C4 cycle, C3 cycle is happening inside your bundle sheet. C3 cycle is happening inside the bundle sheet in your C4 cycle. Clear? Remember again, this CO2 will join with this CO2 will be again taken up by your RUBP. We can have RUBP plus your CO2, that will give your PGA. That is happening inside your cycle here. But what happens to pyruvic acid? This pyruvic acid is sent up again. And this pyruvic acid again gets converted to your PEP. Now in this conversion, 2 ATP is being consumed. 2 ATP is being consumed to 2 ADP. Clear? Converted to 2 APP. That's all is your C4 cycle, friends. That's all is your C4 cycle. You should know what is happening in your bundle sheet. You should know what is happening in your mesophyll chain. Now one more question comes in. If C3 cycle is happening here, do you find Rubisco enzyme here? Answer is yes. You find Rubisco enzyme here. Do you find Rubisco enzyme in your mesophyll cells? No. You will not find Rubisco enzyme in your mesophyll cells. Do you find your pepcase here? We find pepcase here. The question is, do you find pepcase in your C4, in your bundle sheet? The answer is no. You do not find pepcase in the bundle sheet. Clear? Let's completely summarize this once. We have your CO2 is entering here. CO2 plus PEP, that is phosphoenol pyruvate, in the presence of your pepcase. What is formation? Formation of oxaloacetic acid formation is happening. This is 1 carbon. This is your 3 carbon. 3 plus 1 is going to be your 4 carbon. Now this oxaloacetic acid is converted to malic acid. Now this malic acid is transferred to your bundle sheet cells. Now this malic acid gets converted to OA. This OA gets converted to pyruvic acid. 4 carbon, 3 carbon. Means 1 carbon is lost. Now what happens to 1 carbon? This 1 carbon will enter the C3 cycle. And all of us know what do we get in C3 cycle? With the help of C3 cycle we get PGA. This PGA will get converted to triose phosphate. We just learned. Gets converted to triose phosphate. The triose phosphate will produce your sugar, and we have formation of sugar, which is generally happening. Now again, if I ask you question, how many ATP in your C3 cycle? All of you know, we just learned the calculation for that also. Now what happens here again? This pyruvic acid is sent up again, and this pyruvic acid is converted to your PEP again, so the entire reaction can keep on happening. And from pyruvic acid to your PEP, 2 ATP is consumed to 2 ADP. 2 ATP is getting converted to 2 ADP. Is that clear to every single one of you? Is that complete cycle clear to every single one of you? Tell me in the chat. Is that point clear to every single one of you? If that point is clear to everyone, quickly like the video right now and we'll go ahead. Okay? We'll go ahead. Now, in your NCERT, there's a diagram. Let's understand, there's a table, sorry, there's a table. Let's understand every single line on this table, and your entire cycle of your C3 and C4 will become very clear to all of you. It will become very clear to all of you. Again, see what is happening here. Cell type in which Calvin cycle takes place. Now, where is this Calvin cycle happening? In your C3 plants, it is happening in your mesophyll cells, and Calvin cycle is happening in your C4 plants in your bundle sheath cells. We just learned. See here, Calvin cycle is happening in your bundle sheath cells. Yes. Cell type in which the initial carboxylation is happening. Carboxylation, the initial fixation of CO2. Initial fixation of CO2 is happening in the mesophyll cells, both in the case of your C3 and C4. See here, again proof. Mesophyll cells, initial fixation of CO2 is happening. Now, how many cell types does the cell leaf have? In the case of your C3, only one cell. In the case of C4 plants, there are two cells. One is called as your bundle sheet, other one is called as your mesophyll cell. Which is the primary CO2 acceptor? In your case of your C3 plants, it's RUBP. In the case of your C4 plants, it is phosphoenol pyruvate. Did we just learn? Here, phosphoenol pyruvate is taking up the first carbon here. The number of carbon here is 5 carbon RUBP, 3 carbon in your PEP. Which is the primary CO2 fixation product? In the case of your C3 plants, it's PGA. In the case of your C4 plants, it's OAA. And all of us know oxaloacetic acid is 4 carbon while your PGA is 3 carbon. Does the plant have rubisco? Both the plants have rubisco. Clear. Does the plant have pepcase? No. In the case of your C3 plants, there is no pepcase, but pepcase is present in your C4 plants. Present in your C4 plants. Here, can you see? The pepcase is present in your C4 plants. Now, which cell in the plant have rubisco? Here we have mesophyll cell will have rubisco. Here we have bundle sheet cell will have rubisco in your C4 plant. Again, can you see here? In your C4 plant, bundle sheet will have rubisco. Rubisco will be absent completely in your mesophyll in the case of your C4 cycle. In the case of your C4 cycle. Done. CO2 fixation rate under high light condition, medium, and in high light condition, C4 plants will work better. In high intensities, they will work better. Whether photorespiration is present at low light temperatures? No. If there is low light, photorespiration is not there. And moreover, in the case of your C4 plants, photorespiration is completely absent in your C4 plants. Remember that. Okay. Whether photorespiration is present at high temperatures? Yes. In your C3 plants, in high light condition, high temperature condition, C3 plants will undergo photorespiration. In C4 plants, again no. Whether photorespiration would be present at low CO2 concentration, very high, they're not present here. Whether photorespiration would be present at high CO2 concentration, no. In high CO2 concentration, why will the photorespiration even happen? If the photorespiration should happen, there should be high oxygen content. Remember, I told you in what places the photorespiration happens. Remember that and you'll be able to explain everything. Okay. Clear? Then we have temperature optimum. Here we have 20 to 25 degrees Celsius. Here we have 30 to 40 degrees Celsius. Example is going to be wheat and rice. Here we have maize, sugarcane and sorghum. Maize, sugarcane and sorghum. Clear? Clear? Students, are you understanding? Are you understanding, students? With the help of this table, we summarize your entire C3 cycle as well as your C4 cycle. See here, entire points in a C3 cycle as well as C4 cycle has been done and dusted. Clear? Now let's understand something called as your factors affecting your photosynthesis. There's something called as your internal factors, and we also have something called as your external factors. It's not given, I'll show you that is. Here, internal factors mainly includes your number, size, age and orientation of the leaf, number of mesophyll cells, number of chloroplast, amount of chloroplast. That is, depends on genetic predispositions. So all the internal factors mainly depends on the genes. If the gene, it says that a plant will have 2 leaf, it will have 2 leaf. If it says it will have inverted leaf, it means inverted leaf will be there. Then we have something called as your Blackman's law of limiting reagent. There's something called as your Blackman's law of limiting reagent. Let me tell you a basic definition. You should factors. Many factors control a biochemical process. Controller so in your photosynthesis, many factors control a biochemical process. Then photosynthesis speed. Photosynthesis speed is determined by that factor which is found in least quantity, which is present in your least quantity or amount. Understand this line. Understand this line. Many factors controlled by that factor which is found in the least quantity. What is the meaning of this? What is the meaning of this? Let me explain you with the help of a simple, very simple example. In the case of your winter season, when the sunlight is just approaching, come.
Hello, sir.
Hello, sir.
Hello. Oh my God.
Wow,
What
very
is
good,
Basavaraj
sir.
Sir doing?
I was just teaching them some black man.
Sir is all sweating and all.
Oh, this AC is.
Oh,
So much hard work, like we
I know.
are putting for
So
you.
what we're doing?
Wow.
Is recording some shots.
we recorded some amazing content for all the kids. Yeah.
It
And we
was
have
very-
nice plans for you also.
Huh.
Yeah.
Some creative, shots and all things for
Wow.
So, who is the real king or queen on the channel?
Sir, you only, sir.
No,
sir, you
it
only,
is
sir.
not me, it is
Sir,
not
you
you.
only,
You're
sir.
actually a puller and real king.
Yeah.
Sir,
maybe.
you are the best actor.
I'm actor, but
He's
somebody
He's Shammi Kapoor of our Neet.
Yeah, there is a history to that. Why she's calling Shami Kapoor? Because of the kind of expressions I was giving. Gopika ma'am is in the chat maybe. Hello,
Gopika ma'am,
Bano.
Gopika ma'am.
Hi, Yashaswini.
Sir, it's so cold here, no, sir?
Yeah, it is
I
very
can't
cold,
see
but I think you
anything.
need that.
I know, you need
Because
that.
you are so hot
Yeah.
already.
Yeah,
because
Yeah.
actually.
Yeah. the
Digital
cold and
boards,
all that.
it
It gets
so
hot
much
also, right? It gets so
Yeah,
hot.
it's hot.
Yeah.
Yeah.
Ooh.
But yeah, we need this kind of
Oh, I know the
taste
entire
and
thing.
all. So how's the session going, sir?
It's going great. It's going great. The students are there. They're enjoying the entire session.
Great.
You can see my voice is becoming hazy already.
So you told me that you are completing 3 chapters.
Yeah, full
today,
3
is it?
chapters today. The entire plant physiology will be done today.
Wow, that is
Yes.
the beauty of block strategy.
Yes.
Right? So in the next few blocks, I think the entire most of the syllabus is done.
Done.
And we are done.
So
Yes.
what you are going to do next?
I'm planning on something like electromagnetism or mechanics, one of them, or even waves. That is also a possibility. So I'll let students know very soon. So guys, we are going to be here for the entire week. So make the best use of us, and we are going to make the best use of this time for all of you. So a lot of sessions planned. So stay tuned and come with full energy. I can see a lot of new students on the channel. That shows your newfound love.
Exactly.
Yeah.
Yeah. So get new students, get your friends onto the channel. It means a lot because there are a lot of students who are still struggling to find the right content on YouTube.
Right.
So this is the channel for all of you.
Yes, students.
We have a proper proof for you for that.
Huh?
Yeah.
Like you can see the PYQ session and you can see our content, right? So all the questions that we take,
Correct.
the content is matching. It's not like we are teaching you something and the PYQs you see some different kind
Yeah.
of questions. Things are relatable. There's proper analysis behind the questions, practice, and content that we give you.
Very true.
Exactly. So Sir has actually made an entire plan where we know which chapters, the high weighted chapters and everything, right?
Yeah.
Yeah. So
So-
guys, we are having a lot of fun. Trust me, it doesn't feel like we are working. The question is whether you want to have fun with all of us. After May 5th, all of us together over here, party full on. We will celebrate this, critical moment of your life. So if you want to party, study now. Some students, they are partying now. Okay.
They'll study
we are
again
not.
later on.
They will be studying for
Dropper next year.
Dropper next year. You study now, we will party later. Okay?
Yeah.
So be very serious, but at the same time, we'll have these light moments because I feel you also need that refreshment, or else you cannot sit and study for 12, 15 hours.
is
It's very
actually a
difficult, sir.
task. Kudos to you for doing that, but yeah, that is, if you do that now, you will be at different level next year. Like you will
Yeah.
be in your dream college with
Exactly.
your friends.
Wow.
See, we would love to have you right now, but we do not want you again next year.
Yeah.
So we do not want you next year. So I want everyone to go to your college.
Yeah. How many of you are going to say Tata bye bye to this channel after a few months? After a few months.
1 month, sir, hardly.
No, they will come for counseling. No,
So 3 months.
2-3 months.
2-3 months maximum.
So how many of you are going to say bye bye? Sure, pakka, but don't unsubscribe, okay?
Yeah, yeah.
Yeah.
Please subscribe. There will be a lot of
Fun
activities
sessions
for
also,
you.
yeah.
So we would ask you to suggest the channel to your juniors, right? That would be the
Yes,
best way you
that's
can pass on the baton.
Correct,
Yeah.
correct. Awesome, awesome. Cool. Kudos, guys, and keep up the good work. And Baswa sir is sweating it out for all
I
of you.
know.
Make sure
You must AC
attend
that
the
too.
class, throughout, and get your friends and share the link of this video. Subscribe and do not forget to smash the like button. Okay?
Sharing is caring, always remember.
Yes.
Yeah, correct. So, all the best, Baswa sir.
Thank you, sir. Thank you.
Bye bye, sir.
Bye bye. I'll be coming out in next few minutes anyway.
Yeah, sure.
Students, can we start with the Blackman reaction? Now, what is this Blackman limiting reagent? Now, I told you in the starting that many factors control a biochemical process. Then the photosynthesis speed is determined by that factor which is found in the least quantity. Now, what is the meaning of this? That is, in your winter season, when the sunlight is just entering the surface, in the early morning time, sunlight is very low. In the starting, sunlight is very low. What is the meaning of that? The meaning of that is sunlight is the limiting factor that time. Now, once the morning happens, what happens later on? Sunlight is plentiful and it is no more a limiting reagent. Similarly, in the case of your summer, initially in the start, light is the limiting reagent because it is present in the least quantity. It is able to control the photosynthesis. But as soon as the day is starting, we have plenty of light. But in the case of summer, do we have less water? Now, what is the meaning of that less water? The meaning is very simple. If the water is very less that time, water will become the limiting reagent. Water will become the leading reagent. So whatever reagent or whatever the, factor which is present in the least quantity is called as your limiting reagent. Clear? That is your Blackman's law of limiting reagent. Now, what are the external factors which influence a particular rate of photosynthesis? The first one is going to be your light. Then we have carbon dioxide concentration. Then we have temperature. Then we also have the water. Now, first let's start with the light. Now, what is there in the light? In the light, we can say something called as duration of exposure. In light, when light plays a crucial role, we have something called as duration of exposure. That is short day plant, we have long day plant, then we have medium day plants. Then we also have your light quality. That is red light and blue light. In those two regions, photosynthesis will be higher. All of you know that. With the help of TW experiment, we learned that in your blue region as well as your red region, photosynthesis is very high. Yes. Similarly, there is something called as your light intensity, which is the most important one. Out of the three things, that is light duration, light quality, as well as light intensity is the most important one. Now, what does the light intensity tell you, students? That is, light intensity is basically telling you that this directly controls photosynthesis. It directly controls the photosynthesis. Now, if you notice here in the light intensity to the rate of photosynthesis, in your A, this is your O. This is your O. In O to A, can you see from O to A, rate of photosynthesis is directly proportional to the light intensity. Rate is directly proportional to your light intensity. Yes. As the rate is increasing, as the light is increasing, rate of photosynthesis is also increasing. So can I tell light intensity is directly proportional to the rate of photosynthesis from O to A? Yes, O to A. Now what from here? What about B to C? B to C, there is not much change. No much change. Now what is the meaning from B to C? If there is no much change, that is the meaning that light is no more a limiting reagent. It is no more a limiting reagent. It is from B to C. It is no more a limiting reagent. That's why if you look at your NCERT, there's a one line. It says that light is rarely a limiting reagent. It is very rarely a limiting reagent. Clear? And one more thing you should know here is what if there is very high intensity of light? What if there is a very high intensity of light? If intensity of light becomes very high, that can destroy the pigments. It can destroy the pigments. It can completely destroy the pigments if the light intensity becomes extremely high. Clear? The next one is your carbon dioxide concentration. Next one is going to be your carbon dioxide concentration. The first thing which you need to know about your CO2 concentration is going to be, it is a major limiting factor for photosynthesis. Very important line. Now you can ask me why. Why is it a major limiting reagent? Now according to the definition, whichever quantity or a factor is present in the least amount, that is the limiting reagent, right? And if you know in your atmosphere, just 0.038% is your CO2 concentration. CO2 concentration. So can I tell CO2 is a limiting reagent because it is present in very less quantity? Increase in concentration of 0.05% causes the increase in your CO2 fixation. Even if 6 more hours. Oh no, 4 more hours I guess. 4. Increase in concentration up to 0.05% causes an increase in the CO2 fixation. At low light intensities, neither C3 or C4 plants respond to high CO2 concentration. Obviously, if there is very low light intensity, what is the meaning? If light is a limiting reagent, if there is no light, can the plants even do photosynthesis? No, plants cannot do photosynthesis in low light. Similarly, at high intensities, both C3 and C4 plants show increased rate of photosynthesis in high light condition. But tell me in the chat, does the C3 plants or C4 plants, which plant will benefit more because of your increased CO2 concentration? Increased CO2 concentration? The answer is very simple. That is, C3 plants will benefit more because the saturation for your C3 plants is around 450 micro per liter. For your C4 plants, it is around 360. So C3 plants can benefit more from your increase in the CO2 concentration. Clear? That is the reason we make the greenhouses. Inside your greenhouse, we mainly grow your C3 plants. We mainly grow your C3 plants, that is your tomato and bell pepper. Examples from your NCERT, tomato as well as your bell pepper, because they are able to grow better in your high CO2 concentration. The next one is going to be your temperature. Very easy. All of you know, that is, temperature controls dark reaction as they are enzymatic. Because all of you know, if temperature becomes too much, what happens to the enzymes? The enzymes will degrade. Yes. If the temperature becomes very low, that enzymes becomes inactive. So can the dark reaction happen if in high temperatures? No. Can the dark reaction happen in low temperature? No. So there is a requirement of optimum temperature for a dark reaction to happen. That is, C4 plants respond to high temperature and show increased rate of photosynthesis. C3 plants have lower temperature optimum than C4 plants. All of you know this by now, right? Tropical plants have a higher temperature optimum because tropical plants are used to high light conditions, and because of the anatomy called as your Kranz anatomy. Because of your Kranz anatomy. Clear? Last one is going to be your water. Now water plays indirect role. Water plays indirect role. Now what is this indirect role, students? Listen to me very carefully. Listen to me very carefully. Now what happens here? This is your leaf. This is your plant leaf. This is your plant leaf. Now what if water is reduced completely? If water is reduced completely, can the leaf curl up? Yes, the leaf will curl. That is, decrease in surface area. Decrease in surface area. Yes. If the surface area is decreased, can I tell photosynthesis is also decreased? Yes. Photosynthesis is also decreased. So that is indirect role of water. Decrease in water, decrease in surface area, decrease in photosynthesis. Now you tell me in the chat, if there is decrease in water, can the stomata remain open? No. The stomata, if the stomata needs to be open, it needs turgidity because of water. If there is no water, the stomata will close. That is, closing of stomata. If stomata is closing, will there be any gas exchange? No. The gas exchange will completely reduce. Gas exchange will completely reduce. Grass I'm writing. Gas exchange will completely reduce. If gas exchange is completely reducing, can I tell photosynthesis is also reducing? Photosynthesis is also reducing. That is your indirect role by water. Indirect role by water. Water done, temperature done, your CO2 is also done. You should know saturation here in CO2, you should know the saturation, and it is the major limiting factor for your photosynthesis. Apart from that, you should also know your light intensity, which controls your photosynthesis. Rate is directly proportional to your light intensity. Clear? With that, chapter is done. With that, chapter is done. Clear, students? Now the time is your 5:35. Let's take a break till let's start at 5:55 respiration in plants. Okay, can we start respiration in plants at your 5:55 p.m. not a.m. post meridian? Yes, can we now? 5:55, let's start here. Yes, I'm doing every single chapter today. I'm doing every single chapter today, students, not just your first two chapters. I'm doing PGR also. I'm doing every single chapter. I want to eat something. So I'll just take a 20 minutes break. I'll sit for some time. I will re-energize myself, and then we'll start with your respiration in plants. In respiration in plants, we'll be doing with your what is respiration, what is glycolysis, what is Krebs cycle, what is electron transport chain. Every single concept we'll be doing today. Okay, clear? After that, we'll be doing your plant growth and regulators also today. So let's see you around. So all of you, I'll meet you at 5:55 p.m. Okay, 5:55. Okay? Cool. So until then, I'll see you that time at 5:55 p.m. I'll go drink some coffee. I'll get re-energized myself. I'll drink some water because I forgot to get some water here and my throat is drying. And I think we finished how long the stream started? I think the class started at around 3:15, right? 3:15 to 4:15 to 5:15 and 5:30. In 2 hours, 15 minutes, 2 hours, 15 minutes, we finished your entire chapter of your photosynthesis in higher plants, and I believe I have not left even a single word of NCRT. Every single word is covered. Okay. Similarly, we'll do your revision of your respiration. Similarly, we'll also do your PGR. Okay. Cool. Bye bye, students. I'll see you at 2:55. Okay. Hello, hello. Welcome back to your own channel, Vedantu Neet English. This is Basavaraj Sir, your biology master teacher. Students, how is everyone doing? Did you understand every single concept from your photosynthesis in higher plants? I believe that chapter is over now and the entire revision has been done. All you need to do right now is solve some questions from your photosynthesis in higher plants and you'll be done and sorted. Now students, let's start the next chapter in your plant physiology. That is going to be your respiration in plants. So are you ready? Can we start? Can we start with chapter respiration in plants now? Can we? Yes. Good evening. From good afternoon we became good evening now. Let's start with your respiration. Yes. Now quickly tell me in the chat, what is the difference between respiration as well as your breathing? Students, remember, respiration is more of a chemical process. Yes, respiration is more of a chemical process, while breathing is a more of a physical process, right? Breathing is a more of a physical process in which we breathe in, we breathe out. Physical process. Now students, what exactly is happening in your respiration? Listen to me very carefully. If this is your cell, if this is the cell, now in this cell, oxygen is entering inside the cell. Yes, oxygen is entering inside a cell. In the same cell, can I also tell food is also entering? Yes, food is also entering. Now this food can be any form of carbohydrates, glucose, sucrose, anything. Okay, glucose. Now this food is entering here. Now this oxygen is acting on this food. This oxygen is acting on the food and it is oxidizing the food. It is oxidizing the food. When oxygen is oxidizing the food, it results in the release of energy. It results in the release of energy. Now students, this energy is stored in the form of ATP. The energy is stored in the form of ATP. Now this is the entire respiration. Now if I ask you to give one more definition of respiration, if I ask you what is the another name of respiration, you will tell me, sir, respiration is a process in which complex substances are broken down to simple substances. From complex substance we get the simple substances, and when this breakdown is happening to the simple substances, this will release energy, and the energy is stored in the form of ATP. Since there is conversion from your complex substance to simple substance, can I call this a form of catabolic process? Yes. Can I call it a catabolic process? Yes, I can call it a catabolic process. Now students, in your respiration, energy is being released outside. So can I call it a exothermic reaction? Yes. Can I call it a exothermic reaction? Exothermic reaction. Yes, I can call it a exothermic reaction. Done. Exothermic reaction, and it is a catabolic process in which a complex substance is broken down to simple substances, and when this is happening, energy is being released and energy is stored in the form of ATP. Clear? The next concept which you need to understand is something called as your respiratory substrates. Now what is this respiratory substrates? Respiratory substrates, here I told you, complex substances which are broken down to simple substances. This complex substance is nothing but your respiratory substrates. The substrates which are producing the energy, that is called as your respiratory substances. Example is going to be your carbohydrates. We have carbohydrates which are respiratory substances. We also have proteins. We also have fats. We also have certain organic acids. We have carbohydrates, proteins, fats, as well as your organic acids basically belong to your respiratory substrates. Clear? Now what about minerals? What about minerals? Right? What about minerals as well as your vitamins as well as water? Minerals, vitamins and water, do they belong to your respiratory substances? The answer is no. Vitamins as well as your minerals as well as your water do not belong to the respiratory substrates. Remember that. The next thing we need to understand is ATP. Like, how can we talk about your respiration and not mention ATP, right? So ATP is called as your energy currency of the cell. ATP is called as what? Energy currency of the cell. That is, ATP is broken down to ADP. Now what happens when ATP is broken down to ADP? Energy is being released. If you look at the structure of your ATP, adenosine triphosphate, we have 3 bonds here. Every single time a bond is broken here, energy is being released, and this energy can be utilized by different reactions. A same energy can be utilized by your plants, energy can be utilized by your animals. Now let's understand the concept of ATP and NADPH with a basic example. In your houses, your mother or father or both can be working. When they work, do they receive money for it? Yes. So when your mother and father are working, they're receiving some amount of money, and this money is present in the bank. Yes. So every single time you go out, you eat burger, pizza or anything, what do your parents do? They take out their phone, scan and pay, or they take their credit card, debit card, and they pay. So what are they doing? They're using the money to buy some goods. Similarly, inside your cell, energy is stored in the form of ATP. Every single time there's a requirement, like how you add a requirement to eat burger and pizza or ice cream. Every single time there's a requirement inside the human body, ATP is broken down to release the energy. ATP is broken down to release the energy. Now, what is the meaning of NADH and FADH2? NADH2 and FADH2 is like your check. It is like your check. Like when you take the check and you deposit in the bank is paying you the money. So in this reaction in your photosystem, photosynthesis, when we take NADH and FADH2 and we deposit NADH and FADH2, what do we get in return? We are getting ATP in return. That is like a check. Like how you deposit check, you're getting the money. Money is your ATP here, right? That is your NADH and FADH2. Now students, let's understand the basic difference between your autotrophs and heterotrophs. What is your autotrophs? What is your heterotrophs? Can anyone in the class tell me what is the meaning of autotrophs? Anyone? Students, I told you initially, right? This is your cell. Yes, this is your cell. Inside the cell, oxygen is entering into the cell. Inside the same cell, I told you, food is also entering inside, right? And when the food is getting oxidized, energy is being released, and energy is stored in the form of ATP. Yes. Similarly, we also have some organisms in which oxygen is entering inside. What about the food? The food here is not entering from outside. So the organisms which are able to produce their own food, that is, they do not require food from the external source. See, here we had food was taken from external source into the cell. But in the case of autotrophs, such as your plants, food is prepared within the cell. The food is prepared within the cell, and this oxygen will react on the food, which will produce your energy, and energy is stored in the form of ATP. Energy is stored in the form of ATP. Now you know what is the difference between autotrophs and heterotrophs. In the autotrophs, the food is prepared inside the cell. In your heterotrophs, the food is entering from outside into the cell. Clear? That's how you can identify what is the autotrophs and what is heterotrophs. the next thing which you need to understand is respiration types. What are the different types of respiration? We have your aerobic respiration. We also have anaerobic respiration. Now students, tell me, what is the meaning of aerobic respiration? Tell me, what happens in aerobic respiration? Aerobic respiration happens in the presence of oxygen. Yes, and it is mainly happens, where does the aerobic respiration takes place? Aerobic respiration takes place in the cytoplasm as well as the mitochondria. Yes. Now what about your anaerobic respiration, students? Anaerobic respiration happens in the absence of oxygen. And remember students, anaerobic respiration mainly happens in the case of your cytoplasm. It mainly happens in the case of the cytoplasm. That you need to remember. Clear? Now one more thing. Clear? This is the main basic difference between aerobic respiration as well as anaerobic respiration. Now if you focus on anaerobic respiration in your NCERT, there are two types. That is alcoholic fermentation as well as your lactic acid fermentation. Alcoholic fermentation and lactic acid fermentation are the examples of your anaerobic respiration. Example is going to be yeast and rhizopus, and lactic acid fermentation can be seen in the case of your muscle cells as well as lactic acid bacteria. Now what about aerobic respiration? Aerobic respiration happens in your normal cells. See, this is your aerobic respiration. Glucose plus oxygen, energy is being released, carbon dioxide plus water is released as a byproduct here. Now remember students, aerobic respiration is said to be complete oxidation. Very important line. It is said to be complete oxidation. It is complete oxidation is happening in the case of your aerobic respiration. Now what about your fermentation? We just learned, right? We learned about fermentation here. The fermentation is said to be incomplete oxidation. It's said to be incomplete oxidation. That is your fermentation is incomplete oxidation. Clear? Now aerobic respiration happens in multiple steps. That is a multiple step process. Now you can ask me, why sir, why does it happen in multiple steps? The answer is very simple students, because in the case of your aerobic respiration, there is production of a lot of energy, so that to make sure some energy is not lost or do not get overwhelmed by a lot of energy, all the reactions happens in a step-by-step manner, and hence energy is stored in a step-by-step fashion. Example is going to be your glycolysis. Glycolysis happens in your 10-step process. At each step, some amount of energy is stored, some amount of energy is lost. Clear? Now, where do plants get the respiratory substrate from? The question is very simple. Where are they even getting it from? Are they buying it from supermarket? Like how you go buy your favorite vegetable like tomato, brinjal, everything, right? You go to the market and you bring it. How do plants even get the respiratory substrate? The first simple thing is, yes, how do they get it? Students can tell me, how do they get their substrates? Where do plants get their respiratory substrates from? Can anyone tell me? The respiratory substrates is prepared within the cell, right? Respiratory substrates is prepared within the cell. That's why they're called as autotrophs. Same question. Okay. See here. Carbon dioxide plus water, glucose is produced within the cell. Glucose is produced, and the same glucose can be utilized. The same glucose can be utilized by the plants for respiration within the cell. Next question is, do plants breathe? Can anyone tell me in the chat, do you think the plants actually breathe? The answer to the question is very simple. No, plants do not breathe. That is, the answer to the question is no. The plants do not breathe. But do they require oxygen? Yes. Plants do not breathe, but they require oxygen. Plants can't breathe. but they require oxygen. They don't breathe, but they require oxygen. But students, if the plants is not breathing, how are they obtaining the oxygen? Like, how are they getting it from? Do they again buy it from somewhere? I think I made a trap. I think I got a trap and like made you fall in it. Yes. So students, if they don't breathe, how do they get the oxygen? Students, you need to remember, in the case of your plants, there is on the leaf of a plant. On the leaf of a plant, on this leaf, do you see this tiny openings on the leaf? On the tiny openings on the leaf called as your stomatal opening. Yes, on this stomatal opening through which oxygen can enter inside, or your CO2 can enter inside, and exchange of gases can take place. Now that is with the help of leaves. Do you think respiration or exchange of gases is happening even in the stem? Can anyone tell me, does the exchange is happening with the help of stem? The answer is yes. Again, yes is the answer. That is, in the case of your higher plants, that is, the plants which are showing your secondary growth. Example is your dicots. Example is your dicots. In those particular stem or the trunk, there are special type of cells called as your lenticels. Can you see this? The special type of cells called as your lenticels. Now these lenticels also help in exchange of gases. Exchange of gases. Now students, why plants do not require special organ? The question is, students, do you understand how I am approaching this chapter? Do you realize how are we approaching this chapter? We are approaching this chapter in the form of question format. I am asking you a question and we are solving the question. Okay. Now, why do plants do not require special organ? There are three main reasons, students. The first reason is that each part of a plant can take care of its oxygen demand. They're like, I can take care of myself. I do not need a free food or anything. I can take care of myself. Similarly, when there is photosynthesis happening, if photosynthesis is happening, oxygen is the byproduct is there. So plants can utilize that oxygen from byproduct of your photosynthesis. Yes. So do they require a special organ? No. So oxygen is already present in the cell. So plants can just use that oxygen. Yes. And one more thing you need to remember is that in the case of your plants, all the chloroplast is present towards the outside. Yes. And most of the diffusion of air is being diffused by diffusion. Most of the air is being most of the air is moved about by diffusion. So they do not require any special type of organs. No organ required. They're like, I can take care of myself. Now the next question is, students, which is the first step in all types of respiration? The first question is, which is the first actual step in your respiration? That is gonna be your, can anyone tell me in the chat? I cannot hear. Can anyone tell me in the chat, which is the first step in your respiration? That is gonna be your glycolysis. That is gonna be your glycolysis. Can we start our students? Can we start with your glycolysis? Yes. Do you see how smooth the transition became? We started the chapter with a very basics, respiration. We spoke about plants. We spoke about everything. Now we are transitioning to a concept called as your glycolysis. Now students, what is happening in your glycolysis? Students, glyco means sugar. Glyco means sugar and lysis means to split or break. So in your glycolysis, what are we doing in glycolysis? There is technically splitting of your sugar. Splitting of your sugar. Now before we go to the entire glycolysis, I want every single student who are watching right now to understand two different words. I hope you know by now. One is called as your oxidation, one is called as your reduction. Now because when we understand glycolysis, these two words I'll be using constantly. I'll be constantly telling you what is oxidation, what is reduction. So you should know what is oxidation, what is reduction. Now oxidation is what? Loss of electrons and loss of protons. Sometimes loss of proton or loss of hydrogen. Remember, oxidation means loss of electrons or loss of protons. What is reduction? Reduction means gain of electrons or gain of hydrogen. So will you remember that? Quickly, everyone in the chat, please type this. I want everyone to type it or write it in your book right now, because we'll be using this word very frequently now. Okay. Now students, the other name of your glycolysis is also called as your EMP pathway. It is also called as what? EMP pathway. What is EMP pathway? It is called as your Emden, Merhoff, Parnas pathway. These are the three different scientists who discovered the cycle called as your glycolysis. Students, you need to remember one thing now. You need to remember one thing. Where does the glycolysis take place? Before we start glycolysis, I think I should have written it here only. I'll write it here only, students. What is the location of glycolysis? Can anyone tell me? What is the location of glycolysis? Glycolysis happens in the cytoplasm. Location of glycolysis is cytoplasm. What is the number of steps? The number of steps in glycolysis is 10 steps. It is a 10-step process. Clear? Now, in the case of glycolysis, is it a complete oxidation or is it a incomplete oxidation? Can tell me in the chat. Is it a complete oxidation or is it an incomplete oxidation? Or it is also called as partial oxidation. Or is it called as partial oxidation? Now, why is it called as partial oxidation? Because there is no production of CO2. Since there is no production of CO2, that is why it's called as your partial oxidation. Clear? Clear, students? The next thing is, where do you find it? Where? Do you find it in your animals? Do you find glycolysis in the case of your plants? Or do you find it in your bacteria? Or do you find it in your amoeba? The answer to the question is everywhere. Everywhere. That is, it is found in the case of your animals. It is found in the case of your plants. Glycolysis happens in your microorganisms. Yes, it happens in your aerobic bacteria. It also happens in your anaerobic bacteria. Both. Clear? Happens in aerobic bacteria. Anaerobic bacteria happens in your plants. Happens in the case of your animals. Understood? Are you understanding every word I'm saying? Yes. Now students, if you look at the glycolysis, the entire glycolysis is divided into, yes, as also, we are animals only, right? We are animals. Now students, we need to understand the glycolysis is actually happening in 2 steps. How many steps? 2 steps. See, technically there are 10 steps. But if you divide this 10 steps into 2 parts, the first part is called as your preparatory stage. The second part is called as your payoff stage. Now, what is this preparatory stage and what is this payoff stage? Let's understand, students. Let's start with the first step in your glycolysis. The first step in your glycolysis is called as your phosphorylation. What is the first step, students? The first step is called as your phosphorylation. Now, what do we have? We have one molecule of your glucose, that is a 6-carbon molecule. What are we doing? We are doing phosphorylation. Now, what is the meaning of phosphorylation? Meaning of phosphorylation is addition of phosphate. Are we doing that? So we have one ATP molecule here. So from this ATP molecule, this phosphate is broken down. This phosphate is broken down and this phosphate is attached to the 6th position in your glucose. So we have glucose, 6th position, one phosphate is added. We obtain glucose 6-phosphate. Yes. Do we obtain glucose 6-phosphate now? And students, the enzyme involved here is the hexokinase. Very important. Hexokinase is the enzyme which is present here. That is phosphorylation, glucose to glucose 6-phosphate. ATP is broken down and phosphate is being attached here. attached here. Clear? Can you see? We have now glucose 6-phosphate. Done. First step done here. Can you see? Let's tick here, all of you. Open your NCRT. Write down. Tick. Glucose to glucose 6-phosphate. 1 ATP is converted to ADP. Minus 1 ATP. Done. In the next step is called as your isomerization. Now, what is happening in isomerization? In isomerization, there is a change in the structure. Remember, students, in isomerization, what happens? Isomers are molecules with the same chemical formula. The basic chemical formula remains the same, but different in the structure. The structure will change now. That is, glucose 6-phosphate will get converted to fructose 6-phosphate. Chemically, they are same now. Structurally, they are different. What is the difference here? Can you see here aldehyde group here? And can you see a ketone group here? Aldehyde group, ketone group. Done. So we have glucose 6-phosphate is getting converted to fructose 6-phosphate, and it is called as your isomerization. It is called as isomerization. Done. Second step done. Glucose to glucose 6-phosphate. And students, remember, whenever there is isomerization, there is no release of energy, no consumption, no release of energy. Isomerization. Now, students, remember, next step is going to be your third step. In third step, what is happening? In your first step and third step, there is something called as phosphorylation is happening. So again, in your third step, what is happening? We have a ATP, bond is broken here again. Now this particular phosphate is attached to the first carbon. It is attached to the first carbon. So you tell me in the chat, it is called as fructose 6-phosphate. Now if I'm adding one phosphate to the first carbon, can I call it fructose 1,6 bisphosphate? Yes, I can call it fructose 1,6 bisphosphate. Yes, one phosphate here, one phosphate here. The enzyme involved here is phosphofructokinase. Phosphofructokinase enzyme is involved here. And if you look at your NCRT diagram here, in the third step, again, one ATP is converted to one ADP. And friends, remember, the third step, which we just learned, is called as your rate determining step, because it is technically the slowest step. Clear? Clear? I hope that point is clear to all of you. Now students, next, till here, I hope everyone is clear. Glucose to glucose 6-phosphate, one ATP is used up. Then we have isomerization. Then again, we have phosphorylation. ATP is converted to ADP. We have fructose 1,6 bisphosphate now. Now this fructose till here, everything is clear, crystal clear to all of you. What is happening in each step? Now students, listen to me very carefully, all of you. From here, fructose 1,6 bisphosphate, which is a 6-carbon molecule, is splitting into 3 carbon molecules. A 6-carbon molecule is split into 3 carbon molecules. The first one is called as your glyceraldehyde 3-phosphate, that is PGAL. The other one is your DHAP, dihydroxyacetone phosphate. See here, fructose 6-phosphate, which is a 6-carbon compound, is divided into 1 3 carbon, 1 more 3 carbon. One is called as your glyceraldehyde 3-phosphate. The other one is called as your dihydroxyacetone phosphate. Dihydroxyacetone phosphate, also called as your DAP, DHAP or PGAL and DHAP. Now students, remember one thing. Again, DHAP and PGAL are something called as isomers. Now all of you know what is an isomer. Can anyone tell me in the chat? Again students, can I tell this DHAP can isomerize into PGAL? Yes, it can isomerize to PGAL. They are isomers. DHAP and GAP are your isomers. So DHAP can interconvert to your PGAL. So what do we have now? fructose 1,6 bisphosphate is getting converted to your 2 molecules. Why 2? Because 1 DHAP got converted to PGAL. So we have 2 molecules of your glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate or PGA. Okay, or PGA. Clear? So that's why from here onwards, that is why from here onwards, can you see everywhere we have 2 molecules. Everywhere we have 2 molecules because there is isomerization is happening here. Done. Done. Now students, listen to me very carefully. Next is your, till here, did everyone understand? Till here we are done now. Till here we are done. Now that is the end of your preparatory stage. Done. Now all the investing is done. All the investment has been done. Now you take your return back. Like how you're writing the NEET exam, you're writing the NEET examination, you're studying for it, that is your investment. You becoming a doctor is going to be your payoff. So now we have the payoff now. Now we have the payoff stage. Now what is happening in your payoff stage, students? In your payoff, listen to me very carefully. Listen to me very carefully, students. In your payoff stage, we had 2 molecules of your glyceraldehyde 3-phosphate. All of you know we had 3 molecules of your glyceraldehyde 3-phosphate. Now students, this glyceraldehyde 3-phosphate will undergo something called as your oxidation. Now here, glyceraldehyde 3-phosphate is undergoing oxidation. Now quickly, my students, my dear future doctors, tell me in the chat, what is oxidation? Oxidation is loss of proton or loss of electron. Yes. So this particular glyceraldehyde phosphate, when it undergoes oxidation, it is releasing 2 electrons, it is releasing 2 protons, and it is also releasing energy. Yes, loss of protons, as well as loss of electrons and loss of energy. Now this protons and this electron and this proton are taken up by whom? There should be someone, right? There should be someone to take up these protons, to take up these electrons. Who is the savior? The savior is going to be your NAD+. That is NAD+, nicotinamide adenine dinucleotide. Nicotinamide dinucleotide will take up the proton, it will take up the electron, and it will do what? If this NAD+ is taking up electron, when we take electron, when we take proton, that is called as reduction. So your NAD+ will undergo reduction to produce what? NAD+ will undergo reduction to produce your NADH+. NADH plus H+. That is how you obtain understanding. This is how you understand glycolysis. There is no point in by hearting. Anyone can by heart, right? Anyone can by heart in the world. If you show this glycolysis to a class 6 students, he can also by heart. But you should by heart? No. Understand the concept. That is, NAD+ is taking in the 2 protons and 2 electrons and 2 protons given by the oxidation of your PGA, right? And we are finally getting your NADH plus H+, right? NADH plus H+. Now students, that is the fate of your protons and electrons. But what is the use of energy? What is the use of this energy? This energy is utilized. This energy is utilized to bring in a inorganic phosphate. So you take an inorganic phosphate and you take this phosphate and you attach it here. You take the phosphate and you attach it here. So what happens, students? What happens? So much have happened, right? See here, glyceraldehyde 3-phosphate is getting converted to your 1,3 bisphosphoglyceric acid. So can you see there is a formation of your NADH+ here in CRT? Yes. Now do you know how it is formed? All of you know how it is formed. The mechanism behind it. Yes. Students, don't by heart this. Don't just by heart this. Understand this. Okay. Now how do we have? We had here 3-phosphate, but we have 1,3 here now. How is it 1,3? Because at 3rd position we had a phosphate, but at the 1st position also we have a phosphate now because of the energy, and 1 phosphate is attached to the 1st carbon, and that's how you obtain, students, that's how you obtain 1,3 bisphosphoglyceric acid, BPGA. BPGA. Clear? Are you understanding? This is how you decode your glycolysis, and this is what I want every student of mine to understand. Okay, PGA. Now students, whatever bond is formed here, right? This bond is completely unstable. This bond is completely unstable. If a bond is completely unstable, will the bond break? Tell me in the chat. If the bond is unstable, will it break? Yes. Now you tell me, whenever the bond is breaking, is there a release of energy? Is there a release of energy? This particular bond is unstable. It is breaking and it is releasing the energy, right? That energy is taken up along with this phosphate. This phosphate plus energy plus 1 ADP plus ADP will result in your ATP formation. ATP formation. So whatever phosphate was here, it is released out and we get the ATP. NCR diagram, see, 1,3 bisphosphoglycerate, we have 3 phosphoglyceric acid. What do we have now? 3 phosphoglyceric. We have the 3 phosphoglyceric acid because first position phosphate is being jumped out. It is being removed from here. So what do we have now? 3 phosphoglyceric acid and 1 ATP formation. Yes, 1 ATP formation done. Now what happens again, students? Do you see again what happens? From 3 phosphoglyceric acid, we are making 2 phosphoglyceric acid. That is again isomerization. What is it? From third position, the phosphate is jumping to the second position. Release of energy. Done. Now what do we have now? We have what? 2 phosphoglycerate. We have the 2 phosphoglycerate now. 2 phosphoglycerate. Now this 2 phosphoglycerate will lose water. Look at the structure of your 2 phosphoglycerate. It is losing the water and it is forming a structure called as your phosphoenol pyruvate. The 2 phosphoglyceric acid is losing water and it is preparing PEP called as your phosphoenol pyruvate. All of you know PEP, right? The same PEP which is present in your C4 cycle. Yes, PEP, right? Phosphoenol pyruvate. Now students, again in your in your phosphoenol pyruvate, this bond is again unstable. This bond is again unstable. So can I tell this bond can be broken? When this bond is broken here, this bond is broken. This should be not pyruvate. This should be your phosphoenol pyruvate. This should be phosphoenol pyruvate. Okay, not pyruvate. This bond is broken. Energy being released. This phosphate is taken up by a ADP and ATP is being produced. ATP is produced and 2-phosphopyruvate PEP with the help of releasing energy, it is getting converted to finally the pyruvate. It is finally getting converted to pyruvate. Done. That done. Convert to pyruvate, and that is the end of your glycolysis. Beautiful glycolysis. So what do we have? Glucose to glucose 6-phosphate. ATP is produced. ATP is used up. Isomerization. ATP is used here. Again, then we have your carbon splitting up. Then we have your oxidation of your glyceraldehyde 3-phosphate. When oxidation is happening, what is happening? 1 phosphate is added here and NADH is formed. Now same, we have addition of bond is broken up, ATP formation, isomerization, water is removed, pyruvate, bond is broken again, and pyruvic acid, as well as your pyruvic acid is formed. Clear? Now students, what is the net profit here? What is the net profit here? Can anyone tell me in the chat? What is the net profit? What is the net profit? Can anyone tell me what is going to be your net profit? Let me write here this side. Net profit is going to be, students, in your preparatory stage. In a preparatory stage, minus 2 ATP. In a preparatory stage, 2 ATP was consumed. Yes, 2 ATP was consumed. Now students, in your payoff stage, initially we made NADH, right? We made NADH plus H plus. That was 2 molecules of this. We made 2 molecules of your NADH. Yes, because remember 2 to 2 everywhere. We also made 4 ATP. Now, how is this 4 ATP, students? How is this 4 ATP? Here, if you go back here, 1 ATP here, 1 ATP here. But it's happening twice, right? It is happening 2 times. Everything is happening 2 times. So, it is not just 1 ATP, it is 2 ATP. 2 ATP and 2 ATP. So, in total of 4 ATP. And we know 1 NADH provides 3 ATP. 1 NADH provides 3 ATP. 3 into 2 is going to be your 6 ATP. 6 ATP. Yes, 6 ATP. 6 plus 4 is going to be what? 6 plus 4 is going to be your 10 ATP. This is your gross. If you need to find net ATP formation, that is 10 ATP minus this 2 ATP, we get 8 ATP is your net production. 8 ATP is your net production. Clear? Clear? 8 ATP is your net production. Understood? That is total ATP formation. Now, students, they can ask you certain questions here. They can ask you a certain question. Now, in the examination, will they ask you the entire glycolysis? No. They'll not ask you the entire glycolysis. They'll ask you few questions here and there. Now, what are these few questions? The first question they can ask you is, in which step water is removed? Tell me, which step water is removed? That is Pg to your PEP. Pg to your PEP, right? Phosphoglycerate to phosphoenol pyruvate. Water is being removed. Done. Question number 1. They can also ask you, in which step ATP is being produced. ATP is produced. It is going to be your 1,3 B P G is getting converted to your 3 P BGA, right? Here 3 phosphoglyceric acid, 3 PGA. Yes. Phosphoglyceric acid. Yes, and that is going to be your 7th step. See here, first step, second step, third step, fourth, fifth, sixth, seventh. 7th step it is forming. 7th step it is getting formed. Yes, 7th step. There is also your PEP, last step, right? PEP is getting converted to your pyruvate. That is your 10th step. They can also ask you, in which step NADH is produced. All of you know it is produced in your PGAL. Formation of PGAL to 1,3 BPG. PGAL here. PGAL phosphoglycerate glyceraldehyde 3-phosphate to your glyceraldehyde 3-phosphate getting converted to 1,3 bisphosphoglyceric acid. 1 NADH is produced. That is technically your 6th step. Clear? And they can also ask you, which is the first enzyme in your glycolysis. Tell me in the chat, the first enzyme is going to be your hexokinase. They can also ask you, which is the last enzyme. That is pyruvate kinase. Students, they will not ask you the entire glycolysis. They can ask you points here and there, and you should know what are the points they can ask you. 5th question. Then they can ask you. Now they can ask you, which is the irreversible steps. They can also ask you, irreversible steps. That is 1, 3 and 10. 1, 3 and 10 are your irreversible step. They can also ask you which is the rate determining step. All of you know it is the third step. Third step is going to be your rate determining step. Clear? Clear, students? Apart from that, he can also ask you which step ATP is consumed. They can also ask you at which step ATP is being consumed also. Yes, that is your first step and third step. First step and third step. ATP consumed here. ATP is being consumed here. ATP is being consumed here. First step and third step. Eighth question. Or they can also ask you how much of substrate level ATP formation is happening. How much net substrate level photophosphorylation is happening. When we speak about substrate level photophosphorylation, we cannot include your NADH. So technically 4 ATP in your payoff, 2 ATP in your preparatory, 4 minus 2 is going to be your 2 ATP. It's going to be your 2 ATP. Students, do you realize, did we just solve so many questions from your glycolysis? The reason I did this point very slow because I know all of you will mug up this. See, all of you might mug up this or with the help of some mnemonic. You might mug up. But students, it is important to understand how each step is happening, because when you go to your MBBS, that's when you will know the importance of this lecture. That's when you will know the importance of this lecture right now. Okay. So when you talk about glycolysis, because you will study again all this in your biochemistry. You will study all this in your biochemistry. Students, again, if you notice this particular paragraph, it has almost all the questions which can come from glycolysis. Yes, they cannot ask you beyond this. It's almost covering all the questions. Here and there, one question can extra come. That you should take care if it comes. Okay. So that is your glycolysis done. Okay. Now, students, what is the fate of pyruvate now? See, we had pyruvate now. Pyruvate work. What is the next work of pyruvate? Now we have formation of pyruvate is done. Now what will we do with this pyruvate? When we say we, plants. So let's understand, we had your glucose. We had your glucose. Glucose was converted to pyruvate. Glucose was converted to pyruvate. Glucose is your 6 carbon. Pyruvate is 3 carbon. Yes. Glucose to pyruvate. This is happening in your cytoplasm, and I will call this process called as glycolysis. I will call this glycolysis. Now, what is this fate of pyruvate? Now, this pyruvate can undergo 3 different reactions. This pyruvate can undergo 3 different reactions. The first one here is called as in your absence of oxygen. In your absence of oxygen, and this is happening in your yeast cells. This is happening in your yeast cells, which results in the formation of which is called as your lactic acid fermentation. It is called as what? Lactic acid fermentation, in which there is production of lactic acid, production of your lactic acid plus energy. Similarly, there is all other process which is also happening in the absence of oxygen, but this is happening in the muscle cells. Yes. So you might have noticed, whenever you go run a marathon, when you have a sports day, suddenly the next day what happens? You cannot walk or anything. In your muscles, it will be like, so hurt, so hurtful. Why is it happening? Why is it happening? Oh, this is one second. Now I remembered. Sorry, students, sorry. This is your alcoholic fermentation, sorry. Yes, no. I remember lactic acid and alcoholic fermentation. In your alcoholic fermentation, what is happening? There's production of your ethanol. Production of your ethanol plus energy. Ethanol plus energy. In your absence of, in muscle cells, what is happening? In the case of your muscle cells, lactic acid fermentation. Lactic acid fermentation. That is production of your lactic acid plus energy. Lactic acid plus energy. The last one is happening in the presence of O2. Presence of O2. That is happening in case of your mitochondria. Now, what is being produced here? The main byproduct of your mitochondria is going to be your CO2 plus H2O. Yes, CO2 plus H2O plus energy. So, students, we'll be studying all these three now. Are you ready? Ready? I corrected it, right? So, we'll be studying the entire thing now. We'll be studying one by one alcoholic, lactic acid, as well as your mitochondrial. So, are you ready? We'll go in a much more slow passion, so I don't want to rush it. So, students, plant physiology is a unit where you need to understand the concept if you want to solve questions. In your photosynthesis and higher plants, we made sure every cycle, everything's drawn, right? So, you understood now plant photosynthesis. Similarly, I want you to be master of your respiration also. Now, the first one, your respiration is called as here. In anaerobic respiration, we have lactic acid fermentation and we have alcoholic fermentation. Now, students, what is happening in your lactic acid fermentation? Listen to me very carefully. What is happening in your lactic acid fermentation? Listen to me very carefully. We have the pyruvate. Now, this pyruvate is undergoing reduction. This pyruvate is undergoing reduction. Now, tell me in the chat quickly, what is the meaning of reduction? Tell me in chat. The meaning of reduction is that gaining of electrons or gaining of protons. Yes, reduction. Now, students, which is the reducing agent here? The reducing agent here is going to be your here. This is your reducing agent. And remember, students, reducing agent is the agent which is going under oxidation. This is undergoing oxidation. NADH is getting oxidized, meaning is what? NADH is losing protons and losing electrons, and it is becoming NAD plus. Yes, and the electrons, it is releasing, NAD plus is releasing 2H plus plus the 2 electrons. These 2H plus and 2 electrons are taken up by the pyruvate. If the pyruvate is taking these 2 protons and 2 electrons, pyruvate is undergoing reduction to form the lactate. Now, students, you will get a question here. That is, which is the first electron acceptor in the case of your lactic acid fermentation? Tell me quickly. Which is the first and the final electron acceptor in the case of your lactic acid fermentation? Electrons are being accepted by your pyruvate. So, pyruvate is going to be your first and the final electron acceptor in your lactic acid fermentation. And how will you know this? The only way you can know this if you know the mechanism. If you just by heart that pyruvate to lactate, enzyme is lactate dehydrogenase, a concept you will never understand. You will never understand the concept. Okay? So remember, NADH is undergoing oxidation to NAD plus. What is the meaning of oxidation? The meaning of oxidation means loss of protons and loss of electrons. Lost. Now these electrons or protons are taken up by your pyruvate. Pyruvate is becoming reduced to lactate, and the enzyme involved here is lactate dehydrogenase enzyme. Lactate dehydrogenase enzyme is involved here. Clear? Now you need to remember one more thing here is, that's all. That's all in your lactic acid fermentation. Done. All of you are able to solve? The first electron acceptor is going to be your pyruvate. Now let's understand alcoholic fermentation. Very simple again. Very, very simple. In the first step, pyruvate is getting converted to acetaldehyde. Pyruvate is getting converted to acetaldehyde, students. Pyruvate is 3 carbon, acetaldehyde is 2 carbon. It means that 1 carbon is lost. 1 carbon is lost here, and that is why it's called as pyruvate decarbolase enzyme is involved here. Pyruvate decarboxylase enzyme here. So with the help of pyruvate decarboxylase enzyme, from pyruvate we are removing 1 carbon. 1 carbon dioxide is released. 1 carbon dioxide is released outside, and which results in the formation of acetaldehyde. Students. Now listen to me very carefully. Now this acetaldehyde is undergoing same reduction. It is undergoing reduction. How? Remember, this NADH is getting oxidized to NAD plus. It means that it is losing H plus, it is losing electrons. Now these electrons and H plus is taken up by acetaldehyde. It is taken up by acetaldehyde. So acetaldehyde is undergoing reduction to form ethanol. Ethanol is a product of your alcoholic fermentation. And remember, the enzyme involved here is alcohol dehydrogenase. In your examination, they will directly ask you in examination, which are the 2 enzymes in your alcoholic fermentation. You should know. The first one is called as pyruvate decarboxylase. Why? Helps in decarboxylation. The next one is called as your alcoholic dehydrogenase. Hydrogenase enzyme is involved here. H plus. Yes. Will you remember this? Concept is important. You need to remember the enzyme here. Similarly, you need to remember the enzymes even in the case of your lactic acid. Lactate dehydrogenase. Pyruvate decarboxylase, alcoholic dehydrogenase. Now the question arises, which is the primary electron acceptor in the case of your alcoholic fermentation? Tell me the check quickly. The primary electron acceptor in the case of your alcoholic fermentation, it's going to be acetaldehyde. It is going to be acetaldehyde because acetaldehyde is gaining the protons from here. Done. Now students, listen to me very carefully. Again the diagram. Glucose 6, for this is your glycolysis. Then we have pyruvate. Pyruvate can undergo lactic acid fermentation or can also go alcoholic fermentation. In both the process, can you notice NADH plus H plus, NADH plus is getting utilized. And we have the NAD plus in both cases. Now this NAD plus is sent back. This NAD plus is sent back to the glycolysis. Okay, to make sure NAD plus is being replenished. Imagine if all NAD plus is being used up here. Can you do the glycolysis? No. So NAD plus is sent back to glycolysis. Now you tell me in the chat, what is the net electron gain here? Net ATP gain. Net ATP gain. Net ATP gain. Students, remember, in the case of your alcoholic fermentation and lactic acid fermentation, in the preparative stage we have -2 ATP. In payoff stage we have 4 ATP. But what about your NADH and FADH, NADH? Both are used up. So technically we are only left with 2 ATP. 2 ATP is the net gain of, in the case of your alcoholic as well as lactic acid fermentation. Clear students? Students, every single concept is becoming clear to you. You do not have to mug up anything. Just watch my lecture just to revise any concept. and I will give you these notes. I will provide you these notes on the Telegram channel. Download the notes and revise once. That's it. Now, there's one more concept which I want everyone to understand is alcoholic poisoning. Where's it given here? Yeast poisoning. Yeast poison themselves to death when the concentration of alcohol reaches about 13%. That is called as your alcoholic poisoning. In your lactic acid, in your alcoholic fermentation, the maximum available of alcohol concentration is going to be 13% naturally. But if you want to obtain more percentage of alcohol, you need to go to a separate process called as distillation. Now students, anaerobic respiration is over. Let's focus on your aerobic respiration now. Okay, aerobic respiration. Okay, in your aerobic respiration we have what? Glucose plus oxygen, energy is being released. We have 6 carbon and 6 molecules of water is released. Is this complete oxidation? Yes. 6 carbon, 6 carbon. Complete oxidation is happening here in the case of aerobic respiration. You know, that is, in case of your aerobic respiration, pyruvate molecule that is formed in your cytoplasm of the cell due to the glycolysis enters the mitochondria. Because you know, where does the aerobic respiration happens? Aerobic respiration happens inside the mitochondria. Yes. So the pyruvate which is present outside now, the pyruvate which is present outside due to the glycolysis is now entering inside the mitochondria. It is entering inside the mitochondria. Finally, it is inside the mitochondria. Now what is happening inside the mitochondria? We have your oxide decarboxylation, which is also called as your link reaction, also called as your gateway reaction. We also have Krebs cycle. We have oxidative phosphorylation. That is nothing but your electron transport chain. So we have in the mitochondria oxide decarboxylation, which is happening in the mitochondrial matrix. Krebs cycle, mitochondrial matrix. We also have your electron transport chain, which is happening in your crystal. Clear? That is your inner mitochondrial membrane. Now students, let's start with the oxidative decarboxylation. Also called as your link reaction. It is also called as your link reaction or gateway reaction. Now what is this link reaction and what is this gateway reaction? Yes, link reaction and gateway reaction. Now all of you know we have the pyruvate here. Now this pyruvate, the first thing which will undergo here is decarboxylation. It is called as oxidative decarboxylation, right? So this pyruvate is undergoing decarboxylation. Now what is happening in decarboxylation? Can you see the CO2? This CO2 is completely removed. This CO2 is completely lost here. CO2 is lost. Now what is happening in the second step? In the second step, NAD plus is getting converted to NADH plus. Now for this NAD plus to become NADH plus, for it to undergo reduction, this is reduction, right? Gaining of electrons. Something has to undergo oxidation. Yes, something has to undergo oxidation. That is going to be your pyruvate. That is why it's called as what? Oxidative decarboxylation. Pyruvate is undergoing oxidation. It is undergoing oxidation. What is oxidation? Loss of protons as well as loss of electrons. So pyruvate is losing a CO2. It is losing protons and electrons. It is losing protons as well as electrons. Yes, the electrons and protons are taken up by your NAD plus, and NAD plus is getting reduced to NADH. Now what is the reduction? Reduction is gaining electrons. They are gaining electrons and NADH is produced. Now to this pyruvate, if this part is removed, this CO2 is removed, what are we left with? We are left with your COCH3. Now what is this COCO3? I think someone came. Now, what is this COCH3? Can anyone tell me in the chat? This COCH3 is nothing but your acetaldehyde, right? Chemistry. Acetaldehyde. It is your acetal group, not aldehyde group. There's no aldehyde here. It is your acetal group, right? COCH3 is your acetal group. Now, this acetal group is undergoing oxidation to produce your acetyl CoA. But where does this CoA comes from? This CoA is from your third step, which is addition of an enzyme called as your coenzyme A. Students, let me repeat one more time and let me know in the comment section if you're understanding also. Pyruvate, first step, losing of CO2, that is your decarboxylation. In the second step, what are we left with? After CO2 is gone, we are left with the acetyl group. This acetyl group is undergoing oxidation. Now, what is the meaning of oxidation? Loss of protons, loss of electrons. These electrons and protons are taken up by your NAD+. NAD+ gets reduced to NADPH, so NADH. Now, once your acetyl group is undergoing oxidation, this will result in the formation of a group called as acetyl CoA, and this CoA is because of coenzyme A. One step, second step, third step. First step, second step, third step. Clear? Tell me in the chat. If you're able to understand the actual process, how a glycolysis, how your link reaction, oxidative decarboxylation is happening for the very first time, quickly like the video and share the video and green has in the chat. See, you can by heart this. All of you know pyruvate is getting converted to acetyl CoA. That is what given you in NCERT, right? But do you know how NADH is produced? You should know what is NADH production, right? And you should also know why is it called as oxidative decarboxylation, because there is decarboxylation and there is oxidation is also happening. Oxidation is also happening here. Okay, clear all of you? So I have made sure I've written the points here. You can read these points and also understand. You can read these points also and understand later on. Now, students, oxidative decarboxylation is a link between your glycolysis and as well as your Krebs cycle. It is a link between your glycolysis and Krebs cycle. Done. Now, what do we obtain? The final product. The final product of your link reaction is acetyl CoA group. All of you know acetyl CoA group. That is, it is a 2-carbon molecule. It is a 2-carbon molecule. It's an acetyl group. All of you know chemistry, right? Basic chemistry. Acetyl group. Synthesized by the oxidative decarboxylation. Link between glycolysis and Krebs cycle serves as a fuel for your citric acid cycle. That is, this acetyl coenzyme A will enter the citric acid cycle. Now, there are 2 points which is missing here. The 2 points are your, if you look at your NCERT right now, pyruvic acid, CoA, NAD+. Acetyl coenzyme A, CO2, I told you, decarboxylation, formation of your NADH+. The enzymes involved. Enzyme is called as your pyruvate dehydrogenase. The enzyme involved here is pyruvate dehydrogenase and a cofactor, that is Mg2+. Mg2+ is the cofactor. Remember these points, students. Question will come from here. And you know how CO2 is coming out. You also know how NADH is producing out. And you also know the cofactor. You know the enzyme now. You also know the enzyme. Now, this acetyl coenzyme A now. This acetyl coenzyme A is entering your Krebs cycle. It is entering your Krebs cycle now, students. Friends, now let's start with Krebs cycle. Students, till now I'm teaching you detailed, even though this may be a complete plant physiology. I am teaching you from the scratch. I'm teaching you how I would teach every other student when there's a lot of time also. I'm taking your time and utilizing it. Now let's start with your Krebs cycle. The Krebs cycle was named after a scientist called as your Hans Krebs, who was the person who discovered it, Hans Krebs. Now, Krebs cycle is also called as your energy produced. Now, what is the, it's also energy produced. Why is it called energy produced? Because lot of energy is being produced in your Krebs cycle. Now, I will draw the Krebs cycle for you with the help of a trick. Do you want the trick or do you want the trick or do not want the trick? Let me know that if you want the trick, just say yes sir or some fire in the chat. I will know you need a trick for your Krebs cycle. And I can draw the Krebs cycle with the help of the trick. Okay, tell me in the chat. And also quickly students, everyone should like the video right now, because students, this detailed, this explanation you will not find on YouTube anywhere. No teacher will actually sit there. I have seen so many videos. They usually tell you the entire process, but the entire detailed explanation you will rarely find it. Very few teachers have done it. Okay. The trick here is very simple. The trick here is very simple. That is, let me know the trick somewhere else. I'll write here. The trick is, officer, can I keep selling sandwiches for money? Officer, can I keep selling sandwiches for money? Now, what is O? O is going to be your oxaloacetate. O is going to be your oxaloacetate. Now, oxaloacetate is a 4-carbon compound. Yes. Now, this oxaloacetate is getting converted to your citrate. Now, how is it entering? I told you, we just learned that acetyl coenzyme A is entering the Krebs cycle. It's entering the Krebs cycle. Acetyl coenzyme A is entering the Krebs cycle. I also told you acetyl coenzyme A is 2-carbon. Yes. 2-carbon plus 4-carbon is going to be how many? It's going to be your 6-carbon. Now, students, this citrate will get converted to, officer, can I is going to be your isocitrate. Isocitrate is also going to be your 6-carbon. From isocitrate, there is something called as, I'll tell you later on. There is oxidative decarboxylation happening. There is a release of CO2 and there is also formation of NADPH. There's also formation of your NADH. There are no DP. P will only come in your previous chapter. NADH. We just learned in your oxide decarboxylation, there will be formation of your NADH. That is your oxide decarboxylation is happening here. So students, you remember in the case of your Krebs cycle, there are 2 oxide decarboxylation. One is happening between your isocitrate to K, right? K means alpha keto glutarate. The first oxidative decarboxylation. The first oxide decarboxylation is happening between isocitrate and alpha keto glutarate. Now you tell me in the chat, 6-carbon, 1 carbon is lost. So can I call it 5-carbon molecule? Yes, I can call it 5-carbon molecule. Now what do we have? S. S is your succinyl coenzyme A. S is your succinyl coenzyme A. Now here again the second oxide decarboxylation is happening. The second decarboxylation is happening again here. That is formation of NADH and also releasing of your CO2. We learned right in your oxide decarboxylation link reaction. In your link reaction, there was a formation of your NADH and also release of CO2. Yes. So finally we have succinyl coenzyme A. So 5-carbon, 1 carbon is lost here. How many carbon? 4-carbon. Simple. Now what do we have? S, succinyl coenzyme A. S in the sandwich stands for your succinate. Then we have your succinate. S stands for succinate. Succinate is going to be your 4-carbon. Now students, succinyl coenzyme A to succinate, there's a major change. Can you see? There's the release of this coenzyme. There is also a formation of your intermediate step, which I'll tell you later on. When this happens, there is a formation of a GTP. Now this GTP later gets converted to ATP also. Later gets converted to ATP. Now students, this succinate will get converted to F. F is for fumarate. F is for your fumarate. When succinate is getting converted to fumarate, there is formation of your FADH2. Formation of your FADH2. Now fumarate to M4 malate. M4 malate. Now students, from here 4-carbon, 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 right? Now how is it forming fumarate? To form fumarate, there is entry of water here. There is entry of water. Now from malate, it is getting converted to your oxaloacetate again. Now when you form malate to oxaloacetate, there is also formation of a NaDH. There is again a formation of your NaDH. Again a formation of your NaDH. So if you notice here, one NaDH is produced here, one NaDH is produced here, and one NaDH is produced in this step. One FAD is produced here, and one ATP is produced here. That is the total energy which is produced in your entire Krebs cycle. The trick here is going to be your officer, can I keep selling sandwiches for money? O in the officer stands for oxaloacetate, C stands for citrate, I stands for isocitrate, K stands for alpha ketoglutarate, S stands for succinyl coenzyme A, second S stands for your succinate, F for fumarate, M for malate. And students, this carbon number is very important. They can ask you a direct question based on this carbon number. Right? Clear? Done. That is your Krebs cycle. Now, but can we decode the Krebs cycle also? Yes, we can decode the Krebs cycle. See, if you look at this Krebs cycle, which I just draw here, let's see the first step. Let's see the first step. What is happening in the first step? Oxaloacetate is forming citrate, right? Oxaloacetate is forming the citrate. So, oxaloacetate is with a 4 carbon. Acetyl coenzyme is entering from here. Water is also entering from here. And with the help of citrate synthase, coenzyme A is removed out. This coenzyme will again go join where? It will again go and join back to your link reaction, and which will result in the formation of your 4 carbon, 2 carbon, a 6 carbon called as your citrate. Done. Now that is citrate. Now students, citrate, first molecule formed in your cycle. They can ask you a question, which is the first molecule which is found in your cycle? That is going to be your citrate. It contains 3 carbon groups. It contains 3 carbon groups. Clear? It should be 4, right? 3 carboxyl groups, sorry. 3 carboxyl groups. That's what I'm wondering. 1 carboxyl here, 1 carboxyl here, 1 carboxyl here. Yes. 3 carboxyl groups. So citrate, that is done. This is done now. Okay. Now we look at citrate to isocitrate. Now this is again isomerization. Citrate to isocitrate is just isomerization, and remember, in isomerization there is no release or entry of energy. Isomerization. Now isocitrate to alpha ketoglutarate, I told you this is the first oxidative decarboxylation. First oxidative decarboxylation, right? Isocitrate to alpha ketoglutarate. What is happening here, students? See here. What is happening? Let me tell you. We have isocitrate, right? In isocitrate, what is happening? Isocitrate is undergoing oxidation. Isocitrate is undergoing oxidation. When isocitrate is undergoing oxidation, is it losing energy? Sorry, it is losing protons and electrons. Yes. Isocitrate is losing electrons as well as protons. Yes. Just like your link reaction. Now this electrons and protons can be taken up by your NAD plus, and it is getting reduced to your NADH. And since it is a oxidative decarboxylation, will there be a removal of CO2? Yes. So the first oxidative decarboxylation which is happening in your Krebs cycle is between your isocitrate and alpha ketoglutarate, in which there is production of two, where your isocitrate is undergoing oxidation. When this is undergoing oxidation, electrons and protons are lost, and also CO2 is lost here, and this electrons and protons are taken up by your NAD plus, and NAD plus getting reduced to your NADH, and that's how NAD is produced, CO2 is produced. Similarly, alpha ketoglutarate to succinyl coenzyme A, similar thing is happening. See here, same thing is happening here. Okay, similar thing is happening from alpha ketoglutarate to succinyl coenzyme A, but there is addition of your coenzyme now. There is addition of a coenzyme part. Clear? Again, oxidative decarboxylation is happening. Alpha ketoglutarate is undergoing oxidation. undergoing oxidation, and when if alpha glutamate is undergoing oxidation, will it produce H plus in electrons? Yes, those are electrons are taken up by your NAD plus. NAD plus is getting reduced to your NADH. Similarly, there is a release of CO2 and there is addition of your CoA, and that's how we obtain alpha ketoglutarate to succinyl coenzyme A. Clear? Clear. That's how you obtain oxide decarboxylation. So there are two different oxide decarboxylation between your isocitrate to alpha ketoglutarate. First one, alpha ketoglutarate to your succinyl coenzyme A. The second, oxidative decarboxylation. And all of you know how is it happening. That is oxidation, electrons are coming, electrons are taken up by your NAD plus. Now students, from your succinyl coenzyme to succinate, very important. See here, from succinyl coenzyme A, what is happening? We are forming a intermediate substance. NCERT is not telling you this. NCERT is not mentioning this, but my students should know the mechanism. Don't by heart. This succinyl coenzyme A is forming a structure called as your succinyl phosphate. It's a much more intermediate, unstable product. When this is forming, what happens? Your coenzyme A is released outside, which is going to your next step. There is a addition of a phosphate here. Addition of a phosphate in your unstable product. So we have an unstable product here called as succinyl phosphate. Now this succinyl phosphate, what happens is, if something is unstable, can it break? Yes. So similarly, the phosphate bond is broken. Phosphate bond is broken, and that phosphate is taken up by your GDP, and GDP gets converted to GTP. Why? Because of the phosphate from here, and that's how later on ADP gets also converted to ATP, and we get your succinate, and we obtain the succinate. Clear? And we obtain the succinate. Done. So we have the succinate, which is GTP is produced here. Now what happens to succinate fumarate? In your succinate fumarate, that is one production of your FAD to FADH2 is produced. But how are they obtaining? How are they obtaining this H plus as well as your NADH, H plus and electrons? How? Because of the enzyme called as your succinate dehydrogenase. So if you remember very well, this succinate dehydrogenase, do you know it is also part of your electron transport chain, and it is also called as your inner membrane protein. Inner membrane protein. With the help of this inner membrane protein, it is able to obtain H plus, and that's how we obtain FADH2, and finally we have the fumarate. Finally we have the fumarate. From fumarate to myrate, water is added. Look at the structure. Can you see here? Water has been added here. Yes, water has been added here. H2O. Then we have malate to oxaloacetate again. Malate to oxaloacetate again. NADH is added. Now this H plus is actually obtained from the matrix. It is obtained from the matrix directly. From your matrix directly. Clear? Students, that is your entire Krebs cycle. Every single step, do you realize? Every single step we decoded. I also gave you a mnemonic to remember this. I also told you how this is formed, how oxidative decarboxylation is happening. Oxidative decarboxylation is happening. Now students, if you open your NCERT right now, they have given you equation. They have given you an equation, amazing equation, that is pyruvic acid plus 4 NAD plus FAD plus, makes sense, water, ADP plus inorganic phosphate. In the mitochondrial matrix, we obtain 3 CO2, 4 NAD plus, and H plus, and FADH2 and ATP. Yes, this makes sense. We have 1 ATP from your Krebs cycle. But how do we have 3 CO2 here? Because I told you there is 1 CO2 is here, 1 CO2 is obtained here. That is also a question, students. That also is a question. In which step CO2 is being produced? You should know oxide decarboxylation. We have 2 CO2 here. We have 1 NADH, 2 NADH, 3 NADH. But your NCERT is mentioning 4 NADH here. What is the game? What is this game? Why are they playing with us? This is where you need a teacher. This is where you need a teacher to explain with us. Very simple explanation. NCERT is actually combining your oxidative carboxylation, that is your link reaction with Krebs cycle, I'm mentioning here. It is not just the equation for your Krebs cycle, this is the equation for your Krebs cycle plus link reaction. Because we know in the link reaction, one CO2 is coming and also one NADH is also produced here. One NADH is also produced there. Okay, don't get confused. Here. Okay, students, clear all of you? Now, students, next thing is, what is the role of NADH and FADH2 that is synthesized here? What is the function? What is the main role of your NADH2 as well as your FADH2? Can anyone tell me in the chat? What is the fate? Do they go back home? Do they go to their native? Or do they go to a place called as electron transport chain? Tell me in the chat quickly. Do they enter somewhere else? Yes. Students, your this NAD plus as well as your FADH2, they enter a place called as your electron transport chain, also called as your oxidative phosphorylation. Is happening here. NADH2 and FADH2 are transferred to the electron transport chain. ATP is formed as a result of transfer of electrons. That is, there will be jumping of electrons. Which I'll teach you in the next 5 minutes. Electron transport chain, students, I still remember when I was teaching offline, when I used to ask students, which is the most difficult part for you? When I used to ask in this chapter, which is the most difficult part for you? Every single student used to say electron transport chain. Sir, I do not understand electron transport chain. What is this jumping of electrons? What is this photosystem? Are you also like that? Tell me in the chat, are you also like that? Do you still have confusions in your electron transport chain? If yes, in this video, it will get cleared. This will get cleared. Okay. Now, where does this happen? Next question is, where is it happening? The location for your electron transport chain is your inner mitochondrial membrane. Can you see this part here? On the inner mitochondrial membrane, electron transport chain is happening. And in this region, Krebs cycle is happening. So can you start? Students, can we start with your electron transport chain? Quickly like the video, and students, trust me, try to share this video as much as possible, because this amount of clarity in concepts you rarely find anywhere. You rarely will find concept clarity so much. Now I'll make sure you will get a clarity on your electron transport chain like never before. Trust me. You will tell electron transport chain on your fingertips. Okay. Now, before we start your electron transport chain, let me tell you about the different protein complexes involved. In your electron transport chain, there is something called as protein complex. Now, what is this protein complex? Protein complex is nothing but your protein plus certain metal ions. Proteins plus metal ions. Now, students, in your protein complexes, in the case of your electron transport chain, there are 5 different complexes. We have first complex is called as your NADH dehydrogenase. Complex number 2, succinate dehydrogenase, also called as your FADH2 dehydrogenase. Complex number 3, cytochrome BC1 complex. Complex number 4, cytochrome oxidase. Complex number 5, ATP synthase. Done. Similarly, we also have something called as ubiquinone, which is a mobile carrier. We also have cytochrome C, which also is a mobile carrier. Now, students, quickly write it. I'll move aside for a second. I want everyone to write this down. Done. Tell me, is it done? If it is done, tell me in the chat, because I want you to understand this with me and understand this draw this with me. Now I want every single student to actually draw this, write down this. What is this part here? This is going to be your electron transport chain. We have the matrix region here. One second, let me write it in the. This is your matrix region. This is the matrix region. Clear? Now in this matrix region, what is happening? Krebs cycle is happening. All of you know, in the matrix region, Krebs cycle is happening here. Krebs cycle is happening. C by CLE. Krebs cycle is happening. Matrix region. Then we have inner mitochondrial membrane. This is your inner mitochondrial membrane. Then we also have your intermembrane space. Intermembrane space. We have matrix, inner membrane, and intermembrane space. Clear? Now students, quickly draw this. All of you students, every one of you, quickly draw this. Can you draw this? Done? Draw this. Take a sheet of paper. Take a sheet of paper, divide into 3 parts and draw. First part is your matrix, inner membrane, intermembrane. Okay? And we have 1, 2, 3, 4, 5 different complexes. This is your complex number 1. This is your complex number 3. Then we have complex number 2. Then we have complex number 4. And then we have complex number 5. Write down. Done. Now let's try to fill up this. Before we go to the electron transport chain, I want everyone to fill up the complexes. What are the different metal ions present in your complexes? For example, in your complex number 1, NADH dehydrogenase, the main metal ion is going to be your FeS. FeS and FMN. We have F. Let's use this color. Let's use white. Let's use different color. Which will go on that? I think yellow will go better. Here we have FMN, FMN. Along with FMN, we also have FeS. Sulfur iron, sulfur centers. Here we have cytochrome B. See, this is your complex number 3. Cytochrome BC1 complex. It is called as cytochrome BC1 complex. So we should have cytochrome B, cytochrome C, BC1, right? BC1 complex, C1 complex. Along with that, it also has iron sulfur complexes. Then we have complex number 2, that is your FAD dehydrogenase. So can we write FAD? We also have FeS centers. Students, iron sulfur centers are going to be very common here. Iron sulfur centers are going to be very common here. The last complex is going to be your oxidative cytochrome oxidase. Cytochrome oxidase. We have 2 different copper centers there. We have 2 different copper centers here. Copper center A and copper center B. Copper center A as well as your copper center B. Written down. Done. Tell me in the chat quickly. Done. Let's use black. So once we fill your entire complexes, let's understand what is happening in your matrix region. Students, all of us know in the matrix region there is NADH is present there. NADH is present in your matrix region from your Krebs cycle. Yes. So what is happening here? NADH2 is being oxidized to NAD+. Now what is oxidation? Oxidation is basically loss of electrons or loss of protons. So can I tell proton is being lost? Electrons are being lost? Electrons are being lost here. That is 2 electrons. 2 electrons are being lost. Yes. 2 electrons are being lost here. What about the protons? Do we have protons here? Yes. Students, in this, from your first complex, these 2 protons are directly sent to the intermembrane space. 2 protons are directly sent to your intermembrane space. And apart from these 2 protons, your NADH dehydrogenase, the complex itself, does extra work and it transfers 2 protons from your matrix directly to your, apart from this, also 2 proton is also entering here. See, this 2 proton is because of your NADH. Now this 2 proton is because of what? Because of the complex itself. The complex is doing extra work and it is transferring 2 protons from your matrix region to your intermembrane space. So in total of 4 H plus are present here. In total of 4 H plus. Are you able to see this? Are you able to see, students? If you're not, I'll write in different color. Mm-hmm. 2H plus from your NADH, 2H plus from your matrix region because of the complex itself. So in total of how many we have? 4H plus we have. In total we have the 4H plus here, right? 4H plus. Done. First complex done. Now what happens to this particular electrons? Now these particular electrons are sent to your mobile carrier called as ubiquinol. UQ. It's sent to a mobile carrier called as UQ. Yes. Now this UQ will take the electrons and it gets reduced to UQH2. It's getting reduced to UQH2. Listen to me very carefully. The 2 electrons are sent here. It is using 2 electrons and it is getting converted to UQH2. That is, ubiquinol is becoming ubiquinone. Ubiquinone is becoming ubiquinol. Ubiquinol. Now, but how was it obtaining this 2H plus? Using H plus here. That is, it is taking 2H plus from your matrix region. 2H plus is taken from your matrix region by ubiquinol. It is becoming ubiquinone and it is doing a cycle here, which is a mobile carrier. Now, what happens here? Listen to me very carefully. Once the cycle is over, one electron is sent to your complex 3, and along with that, the 2H2 plus. Can you see this H2? The 2H2 is also H plus is also sent. So 1 electron and 2H plus is sent to your, I think 2H plus is sent to your complex number 3. Complex number 3 is getting your 2H plus and 1 electron from here. 2H plus and 1 electron from there. Done. Now students, listen to me very carefully. Your complex number 2 is called as what? Complex number 2 is called as FADH2 dehydrogenase. Yes. So, can we tell here FADH2 is being reduced to FAD plus? Yes. When this is happening, can I tell the electrons and protons sent here? Yes, sent here. They are sent here. Now from here, the electrons are again sent to UQ, ubiquinol. Yes. Now again, this is running a cycle. It's becoming now UQH2. Again, it's turning a cycle. That is, electrons are being transferred here. 2 electrons are transferred here. Now from here, just like here, 1 electron is being sent. Out of these 2 electrons, 1 electron is being sent, and also, can you see this 2H plus H2? Now from this 2H2 is also taken up by a UQ, and 2H plus is also sent to your cytochrome 3. So 2H plus from here, 2H plus from here, 1 electron from here, 1 electron from here. Now you can ask me a question, right? Sir, why only 1 electron is being sent here? Why not 2 electrons? You can ask me, right? Because students, 1 electron is required to make sure this cycle continues. To make sure this cycle continues, only 1 electron is sent, not both electrons. Clear? Now you can also ask me, sir, why is this H is sent here not directly into your intermembrane space? Why? Because remember, succinate dehydrogenase, yes, this particular enzyme here, your complex number 2, succinate dehydrogenase, I told you in the starting, it is a intermembrane protein. It is a, inner membrane protein. It is a inner membrane protein. That is, can you see it is stopping here? This particular protein complex is not transferred to your intermembrane space. It is only stopping till here. So technically, can this H2 be sent to your intermembrane space from here? No. Because your complex number 1, complex number 3, complex number 4, complex number 5 are intermembrane proteins. They are intermembrane proteins. Intermembrane. But your complex number 2 is inner membrane. It is inner membrane. So your H2 cannot be sent to your complex 2. It cannot be sent to your complex 2. That is why the H2 is sent to your ubiquinol. Now you tell me in the chat. Which color should I use? Okay. Now here, 2H plus from here, 2H plus from here. So how many H plus are going out? How many H plus are going out, students? 2 plus 2 is how much? 2 plus 2 is 4H plus. 4H plus is sent to your intermembrane space. So did you understand why your H2 is being sent here? Because it is the inner membrane. It is only stopping here. It cannot go further. Okay. Now students, 4H plus is sent here. Now what about the electrons? What about the electrons? Now we have the 2 electrons here. 1 electron from here, 1 electron from here. Now can this 1 electron be transferred to your complex 4? Yes, it can be sent to your complex 4. But is it an easy job? No. For this particular 2 electrons to send to your complex number 4, it needs a something called as your mobile carrier. And mobile carrier, we have a mobile carrier that is called as your cytochrome C. We have a mobile carrier called as your cytochrome C. So this electron is sitting on this mobile carrier and it is sent to your complex number 4. So electron is where now, students? Electron is in your complex number 4. Electrons, 2 electrons, 2 electrons, and 2 electrons, and 2 electrons. Now who is the final mega boss, big boss here? Who is accepting your electrons? The final electron acceptor in your electron transport chain is gonna be your oxygen. Half O2, right? Half O2 plus 2H plus proton will produce water. When this is happening, when the electrons being sent here, in total of 2H plus out of this, from the matrix region, 2H plus is sent to your intermembrane space. 2H plus is sent to your intermembrane space. 2H plus is sent to your intermembrane space. So how many H plus do we have? 4 plus 4 plus 2. In total of 10 H plus is present in your intermembrane space. In total of 10 H plus is present in 1 ATP. 4 H plus is required for 1 ATP, and that's the end of your electron transport chain. Done. Students, if you have any doubt, this is such a colorful diagram, right? Electrons are being sent here, mobile carrier, protons joining here, from space is coming, one is coming from here, electrons. Done. Students, if you have any doubt regarding your electron transport chain, let me know in the comment section, and if you have any doubt, I will clarify it myself. And you can actually get so many questions from here, so please practice this one more time in your notebook. Watch the video again in a 2x, in 1.5x speed or 1.75x speed. You can draw this again. Okay, students, I trust me, you should not have any confusion after this. After this, you should not have any confusion regarding electron transport chain. Okay, it's very easy. Just draw it once and understand it once, and you will get to go. You're set for life. So if you see this NCI diagram, can you see this complex number 1? What is happening here? Same thing which I told you, 4H plus sent to intermembrane space. Can you see here? To ubiquinol, electrons are being sent here. From here also, from UQ to UQ, H2 electrons are being sent. From here only one electron is being sent here. One electron is being sent. In total there are 4H plus here again. See students, NCI diagram. This is NCI diagram, I'm telling you. NCI diagram. Then mobile carrier, send to your last one, H plus, 2H plus is sent to your intermembrane space. Clear? And I didn't tell the last part only, no? Enter the last part only. What is the last part here? From out of this, whenever 4H plus, I told you here only, right? When 4H plus is sent to your, from your F0 ring rotates, F1 has catalytic activity. When 4H plus is sent to your intermembrane space from here to here, 1 ATP is produced. 1 ATP is produced here. That's it. 1 ATP is produced. Clear? Copper where? Copper centers here, I told you, no? Copper centers. Copper center A, copper center B. Copper center A and copper center B. Clear? Complex phi is also called as your ATP synthase. Also called as your ATP synthase. Clear? I can't play cricket. I'm sorry, Subhendu Sahu. Can't play cricket. I can only play football. Football, badminton, chess. Then we have table tennis. Then we have other games, sports, running. Yes, that's the games I give. Cricket, I can't play. Very bad cricket. Okay, done. Now let's do a ultimate balance sheet. Can we do a ultimate balance sheet? Can we do an ultimate balance sheet now, students? First, we have the glycolysis. First, we have the glycolysis. That is going to be your minus 2 ATP in your preparatory stage. Then we have 2 NADH is being produced here, which is roughly your 6 ATP. And we have 4 ATP in your payoff stage. 6 plus 4 is going to be your 10 ATP minus 2 ATP because of your preparatory phase. So in total of 8 ATP is produced. 8 ATP is produced from your glycolysis. Now what about link reaction? What about link reaction? From a link reaction, we are obtaining 2 NADH. Yes, we are obtaining 2 NADH because from one glucose we are getting 2 pyruvate. 2 pyruvate undergoing link reaction, so 2 NADH, not just one. So we are getting how many ATP? 1 NADH is 3 ATP. 3 2 is 6. 6 ATP. Now what about your Krebs cycle? What about your Krebs cycle? In Krebs cycle, in one cycle there are 3 ATP. With a cycle for one pyruvate, cycle is going 1 times. But we know for one glucose, we're doing for one glucose, there are 2 pyruvate. The Krebs cycle will go twice. In one cycle we have 3 NADH. In 2 cycles, how many NADH will we have? In 2 cycles we'll have 6 NADH. 6 NADH, right? If one cycle we have 1 GTP, in 2 cycles we have 2 GTP. In one cycle we have 1 FAD. Because 1 NADH is giving you how many ATP? 1 NADH is giving you 3 ATP. 3 2s are 6. GTP is going to remain the same, 2 ATP. 1 FADH is 2 ATP, 2 2s is 4. 2 into 2s is 4. So 2 ATP. So how many ATP are giving you totally? That is 2 plus 18 plus 2 is 20, 22 plus 2 is 24. 24 ATP. So we have from your Krebs cycle we have 24 ATP for one molecule of glucose. 24 ATP for one molecule of glucose. Let's do a final total. 24 plus 6 is 30, 30 plus 8 is 38. 38 ATP is the final. 38 ATP is the final ATP produced from one molecule of glucose. From one molecule of your glucose. Clear? Clear, students? That is the molecule of glucose. Total of, in my case, I open a cup noodles. Cup noodles, all of you, I love cup noodles. I love ramen noodles. I add some hot water. That is reality explanation is not required for you. That is not given in NCD. I want to tell you that, but I'm not telling you that reality expectations because that is not in your NCRT, and don't focus on that. I want to focus on what is given in your NCRT right now. So whenever their mother is not home, what do? I take up her cup noodles and I eat cup noodles, or I order food from outside. And all of you might be doing the same. If your mother is not preparing the food and you're very lazy to make your food, what do? You make Maggi, or you take Zomato, or call from Swiggy, get some food. That was your backup, right? That was your backup. Yes. Similarly, inside your body, students. Similarly, inside your body, if all the carbohydrates are utilized, what is the backup? If in your body, if all the carbohydrates are used up inside your body, the backup is going to be your lipids, that is your fats, as well as your proteins. Clear? That is breakdown of fats is going to be your backup, is going to be your fats as well as your proteins. Fats as well as your proteins are going to be your backup. Right? Backup. Clear? Now students, let's understand how fats are your backup. How fats are acting as a backup. See, all of you know there are two types of fats. If you classify fats, if you know biomolecules, fats can be of two types. One is your fatty acid. One is your fatty acids. The other one is going to be your glycerol. Right? One is going to be your glycerol. Yes, fatty acid and glycerol. Now this fatty acid can undergo something called as beta oxidation. It can undergo something called as your beta oxidation. Similarly, this glycerol can also undergo oxidation. When this fatty acid can undergo beta oxidation, it releases something called as your acetyl coenzyme A plus energy. Now this acetyl coenzyme A, can it enter your Krebs cycle? Yes, it can enter Krebs cycle. Krebs cycle can produce your energy again, that is after ETC. Yes, backup is done. Even though there is no carbohydrate, there is no glucose, fats can be converted to acetyl CoA. It can enter Krebs cycle and the cycle can be run. Yes. Similarly, your glycerol can, when you take glycerol and undergoes oxidation, it will produce your DHAP. Now, what is this DHAP? DHAP, remember, glycolysis. Now, this DHAP can undergo isomerization to form your PGAL. This PGAL can undergo glycolysis completely to produce your pyruvate. Pyruvate can undergo Krebs cycle. Krebs cycle products can undergo electron transport chain, and finally energy can be produced. So students, are you noticing that from your fats we can obtain energy? That is the backup. That is going to be your backup. That is, fatty acids can undergo beta oxidation to produce your acetyl CoA, FADH2 and NADH. Clear? Similarly, glycerol can undergo oxidation to produce DHAP, that is dihydroxyacetone phosphate, to produce your PGL, glyceride 3 phosphate, enters your glycolysis pathway, pyruvic acid, acetyl CoA, Krebs cycle. Done. So all of you would have learned by now there's something called as your burning of fats. Now what is this burning of fats? Like, I want to burn some fat, tell, right? Students, remember, the burning of fat means usage of fats in your body. And when does the burning of fat happens? Only and only when your body does not have enough of carbohydrates. So if you go to any gym, they'll tell you, if you want to burn the fats, you need to reduce your intake of carbohydrates a little bit less and increase the concentration of your protein. Increase the concentration of your proteins, Remember that. That's how you burn the fats. That's how you burn the fats. The next one is going to be your respiration of proteins. How can we use proteins? How can we use proteins for your respiration? See, tell me in the chat, do plants have fat? No, plants will not have fat. So students, in case of plants, most of the respiration backup is going to be, in the case of plants, the backup is going to be proteins. The backup is going to be proteins. That is, a protein can undergo proteases to produce your amino acids. Obviously, you know, protein, if you put proteases, they become amino acids. Now, if this amino acid can undergo deamination, if it can undergo deamination, we should be able to produce our desired product. That is, one example is your glutamic acid. If glutamic acid undergoes deamination, it will produce alpha ketogluteric acid. Can this enter your Krebs cycle? Yes, it can enter Krebs cycle. And the Krebs cycle is done, ETC is done, energy is done. Glutamic acid. Similarly, aspartic acid, which is the amino acid, if aspartic acid can enter your deamination, which will result in the formation of oxaloacetic acid. Can this oxaloacetic acid enter the Krebs cycle again? Krebs cycle, electron transport chain, energy, done. Students, are you noticing? This is how a backup is done in case of your plants. Now, what did we just learn? We just learned that proteins and fats can form, which is the product of your amino acids and fatty acids. From proteins, we can make amino acids. From amino acids, we can make respiratory substances. Respiratory substance can enter your respiratory pathway. Did we just learn this? Proteins, amino acids we can make. From amino acids, we can make respiratory substances such as your alpha ketoglutarate as well as your, we just learned, right? Alpha ketoglutarate as well as oxaloacetic acid. That is, from simple substances. From simple substances, we are making complex substance. From simple substance, we are making complex substance. Is this called as catabolism or is it called as anabolism? Tell me in the chat. We just learned that from proteins and fats, which are simple substances, we are making respiratory substance. That is your anabolism. Yes. Similarly, we also learned that your substrates such as your carboxylic, such as your glucose, fructose, sucrose, this complex substances such as your carbohydrates can also be broken down into proteins and fats. Yes, this is your catabolism. From complex to simple. Yes, from simple to complex, complex substances broken down, fragments, catabolism. So in your respiration, is the respiration catabolism or is the respiration anabolism? The answer is both. That is, respiration is called as your amphiboic process. It is an amphiboic process in which catabolism is also happening in your body, anabolism is also happening in your body. So remember students, respiration is a amphiboic pathway, not just respiration, not just catabolism or not just anabolism. Clear? It's an amphiboic pathway. If you look at your NCERT diagram right now, see, carbohydrates are entering your Krebs cycle directly. Carbohydrates, glycolysis is happening, pyruvic acid, acetyl-CoA link reaction and Krebs cycle. Similarly, fats can be broken into fatty acid and glycerol. They are entering the food chain, they are entering the cycle DHAP. They can also enter through acetyl-CoA. We learned, right? Fats and glycerols. See here, acetyl-CoA and DHAP. We just learned. See, we just learned this. Glycerol is ending DHAP. Fatty acid, that is fatty acid, entering through acetyl-CoA. Similarly, proteins are making amino acids. They're entering your pyruvic acid directly. Yes, that's what we learned just now. Yes, that's how you understand botany. Okay. Similarly, this last concept for this chapter is going to be your respiratory quotient. Now, what is this respiratory quotient? That is volume of CO2 evolved divided by volume of O2 consumed. Now here we need to understand 3 points. What is the respiratory quotient for your carbohydrates? What is the respiratory quotient for your lipids, or that is fats? What is the respiratory quotient for your proteins? For your carbohydrates, the respiratory quotient is going to be 1, complete consumption. For your fats, example is your tripalmitin, Neet PYQ. So tripalmitin respiratory quotient, which is a fat, is going to be 0.7. Similarly for a lipid, similarly for your protein, it's going to be 0.9 for proteins. 0.9 for proteins. See here NCRT. See here carbohydrates 0.7 for your fats. For proteins is going to be 0.9. Proteins is going to be 0.9. Done students. Respiration in plants, done. I think we started respiration in plants around 6, right? 6 o'clock. I think 6 o'clock, right? 6 to 7, 7 to 8. No, 5:55. That was the break time I gave you, right? 5:55 to 8:55 in just 2 hours. In just 2 hours with detailed explanation. Glycolysis, Krebs cycle, as well as your electron transport chain and the starting part of your respiration. I told you what is autotrophs, what is heterotrophs, everything covered in 2 hours. Are you seeing what we just did? So 5:55 to 6:55, 7:55, 3 hours, right? 3 hours, sorry. Right, 3 hours, right? 5:55 we finished, right? 5:55, 6:55, 7:55, 3 hours. Done. Now students, the next chapter is going to be your plant growth and development. Yes, the next chapter is going to be your plant growth and development. We'll be doing this chapter in 1.5 hours. Now we'll be doing this chapter in 1.5 hours. Just 1.5 hours. Just 1.5 hours. I'll finish the chapter. But before that, can we take a 10 minutes break because I'm freezing here because of the AC? Can we take a 15 minutes break? We will start at your. Can we start the class at 8:15? 8:15 to 1.5 hours, the class will be done. 2 hours itself, right? Yes, 2 hours itself. Maths weak, little biologist. So, yes, break. I need a break. More than you, I need a break. So you are more than you, I need the break, right? Because I'm standing here from like 2 hours and standing there. Right? Last 2 hours, it's not 3 hours, 2 hours. Let's start at 8:15. Can we? 8:15, all of you? Yes. So class will resume. Dinner break. Yes, all of you eat the dinner or drink water, eat chips, eat anything. But I want everyone to be present back. I want all of your souls. I want every single mind back at 8:15. Class start. 8:15. Okay. 4 hours is completed. In 4 hours, we did 2 chapters. In the next 1.5 hours, we'll finish, or I think 2 hours ho jaye, 2 hours it might happen. Let's take 1.5 hours. 1.5 hours, last chapter also will be done. So this is a long marathon, and I don't, since it's a long marathon, I don't want it to become even more longer. See, I can make this chapter into 11 hours. Plant physiology, I can make it 11 hours, but I don't want to. Time is very crucial, right? So 8:15 we'll start the class. We'll do the last part of we'll do the last chapter. Okay, we'll do the last chapter. Okay. 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 Okay, students, 8:15, I'll see you. Yes, I want all of your souls also here back. 8:15, okay? so cold here. Sound check, sound check. Yes, are we live? Yes, we are live. Welcome, welcome, welcome everyone. Welcome back to Vedantu Neet English. If you know, my name is Basavaraj Sir, your biology master teacher. Students, we have completed the 2 chapters now. That is, first chapter, photosynthesis in higher plants, done and dusted. The second chapter of your plant physiology, respiration plants, done and dusted. And students, if you have any doubts regarding the first 2 chapters, let me know in the comment section and I will clear it for you. Personally, I will reply to every single message out there and you will be able to solve it. Okay. So let me quickly add a message starting right now. Plant growth and regulators. Starting. Yes. So can we start, students? Can we start with the one second, where is my live? Yes, this is the live. Can we start with the it's hot. Students, yes, welcome back. So students, quickly like the video. If you have not liked the video, this is gonna be the ultimate solution for your entire plant physiology. And also let me know in the chat what should we do in your block 2. See, this is block 1 from your botany where we have done entire plant physiology. And I was thinking, which is the most troublesome unit in your entire biology? So Gopika ma'am and I were discussing, what do you think for your block 2? We will do genetics. What's it? For block 2, we'll do genetics. High weightage, right? High weightage. Ma'am, I'll be taking principles of inheritance. Ma'am will be taking your molecular base of inheritance. Done. Block 2 will do genetics. For block 3, we will still think. What to do in block 3 and block 4? We'll still see which is going to be important. What is your input on everyone, and what is the demand of our students. Okay, students. So can we start with the plant growth and development? Let's start with your plant growth and development. Now students, in this entire chapter, biotech is there, yes. Block 2 is going to be biotech. Block 2 is going to be your biotech. Block 3 is going to be your genetics. Yes, block 2 is going to be biotech. I remember now. Block 2 is going to be your biotech, don't worry. Yes, and block 4, okay, let's take block 4 as cell. What's it? Block 4, I'll do the entire cell chapter. That is your cell division as well as the cell unit of life combined together. Okay. Now let's start with your plant growth and regulators. That is starting off with what is a growth. Can anyone tell me what are the 2 different types of growth? Yes. There is something called as your intrinsic growth. One is something called as your extrinsic growth. Yes. There is something called as intrinsic as well as extrinsic growth. Now students, tell me, where do you find intrinsic growth? Intrinsic growth is mainly seen in the case of your humans or living organisms. What about your extrinsic growth? Extrinsic growth can be seen in the case of your non-living things. In the case of your non-living organisms or non-living things, let's just say non-living things. Example is going to be a mountain. That is, if this is a mountain, on top of this mountain, can you see accumulation of snow? Yes, accumulation of snow on a mountain. That is your extrinsic growth. Extrinsic growth. But students, what exactly is a growth then? Can I tell growth is said to be irreversible change? Yes, I can call growth as irreversible change. Growth can be called as irreversible change. Let me give you a quick example. Let's take anyone from the chat. Deepak. Yes, Deepak is here. Now Deepak was in his class 8th, he was 4 feet. Now he's in class 12th, he became 6 feet. From 4 feet, he became 6 feet tall. Deepak, is that growth? Tell me in the chat, is that growth? You'll be like, yes, sir. That is a growth in which a particular organism is growing and it cannot be reversed back. It cannot be reversed back. It is a growth. Now, let's take one more example. Me. I am the example. That is, I was around 60 kgs last year. I was 60 kgs last year. Right now, I am 65 kgs. Imagine I was 60 kgs last year and right now I am 65 kgs. Is that growth? Is that also called as your growth? Tell me in the chat. Is that also a type of growth? The answer would be no. The answer would be no. Why? Why is it not growth? That me increasing my weight from 60 to 65 kgs is not a growth. Why? The reason is that because if I go to gym, if I work out a lot, will I reduce my weight? Obviously. So what did I just tell you? Growth is an irreversible change. If I'm able to reduce my weight back to 60 kgs, does that constitute to growth? No. That is not growth at all. Right? That is not growth at all. I can see Shreyas in the chat. I would be really happy if Shreyas would come here. I thought did Shreyas leave? I don't think Shreyas left. Right? So that is not a growth. Weight is not a growth. Now tell me in the chat, what about a wood? All of you know wood, right? If you take a wood piece, if you take a piece of wood and you add it into water, obviously the wood will swell. And when we take the wood, keep it back on the sunlight, will the wood shrink again? Yes. So that also is not a growth. That is, swelling up of the wood is also not a growth because it is reversible in nature. Similarly, what about your guard cells? What about guard cells? You know, guard cells, when they take up water, they swell completely. Yes, they swell completely. Is that also a type of growth? No. Because if the water is removed, will that guard cells become flaccid again? Yes. So that is also not growth. So what is a growth, students? Growth is a irreversible change, a change which you cannot bring back. That is actually a growth. So all of you know what is the growth now? And remember, students, in the growth, is it anabolic or catabolic? Growth is mostly anabolic process, more of a anabolic process, because you're taking a lot of energy inside, you're building a lot of blocks inside and you're growing bigger, right? So it's an anabolic process. It is a anabolic process. Now, does growth requires energy? Yes. It's an energy required process. Energy required process requires process. Clear? That is your growth. So students, all of you know a basic definition of growth now. Here and there some ideas there, right? Some idea you have about growth now. It is catabolism also. It is catabolism also, but anabolism is more than the catabolism. Anabolism is more than catabolism. Clear? Now students, let's understand a concept called as germination. All of you know what is germination? That is, a particular seed is growing. So what is germination? That is growth of a seed. Growth of a seed can be called as a germination. Technically embryo is germinating and we get a seed. If you look at this, see, seedling, root, shoot, and the seed is growing. If you look at the diagram of your NCERT, we have the seed germination, cotyledon, hypocotyle. We have cotyledons, epicotyle, seed coat, epicotyle and hypocotyle, and finally we have the entire plant here. Now students, let me teach you some basic plant growth terminologies about plant growth now. Now if I tell you plant growth is said to be unlimited or indeterminate. Now what is the meaning of that? Unlimited growth or indeterminate growth? It means that a plant can keep on growing. See, in the case of humans, we stop growing at certain age. At exactly 21 years for boys, we stop growing. You cannot keep on growing, right? You cannot keep on growing 6 feet, 7 feet, 8 feet. No. But in the case of plants, it can show ultimate growth or unlimited growth or indeterminate growth The main reason is that because of meristematic tissues. Because of meristematic tissues or meristematic cells. We have your primary meristem as well as your secondary meristem. Primary meristem is responsible for the initial growth. Secondary meristem is responsible for the lateral growth in the case of plants, because plants growth is unlimited or indeterminate. See here, it is present here. Can you see this? Root apical meristem and shoot apical meristem, responsible for the lengthening of the root, responsible for the increase in shoot. Then we also have vascular cambium, which produces your lateral meristem. Lateral meristem is responsible for the increase in girth. Similarly, if you look at the students, plant growth is said to be open. Yes, the second point is going to be plant growth is said to be open. Plant growth is open. Now, when I say open, what is the meaning? The meaning of open is that in the case of plants, new cells are added continuously. Meaning of open is that new cells are always added. New cells are always added. Done. Similarly, a plant growth is localized. What is the meaning? Plant growth is localized. That is, if you look at the growing parts of a plant, it is mainly in your shoot apical meristem and root apical meristem. That is, it is localized to meristematic regions. It is localized to meristematic regions. It is very localized, meristematic regions. But in the case of humans, it is uniform. Now, what do I mean by uniform? That is, in the case of humans, growth is happening throughout. You will not find a human where head is around full long. You will not find a baby whose arms are very long. No. In the case of humans, it is uniform growth. But in the case of plants, growth is very localized. That is, we can have development of root, we can have development of shoot continuously. Clear? Now, similarly, in the case of plants, the number of organs keep on increasing. Yes. Now, what is the meaning of this, sir? In case of plants, number of organs keep on increasing. Simple. We have the leaf. Does the leaf number increase? Yes. We have the flower. Does the number of flowers increase in the seasonal variation? Yes. Flower is increasing, leaf is increasing, organs are increasing. Done. That is, number of organs are increasing in the case of your plant growth. The first one is going to be your unlimited growth. Second point here is the plant growth is open, that is, new cells are always added. Plant growth is localized in the meristematic regions. Plant, in the case of plants, number of organs are keep on increasing. Now, students, the next concept is going to be your growth is measured. Now, in case of humans, we measure height 4 feet, 6 feet, 8 feet. That is your growth. But how do we actually measure growth, students? The actual way to measure the growth at the cellular level. Actual measure to measure is at the cellular level. For example, one second, students. For example, all of us know if this is a cell, the living part of cell is called as your protoplasm. Now, if this cell is undergoing growth, will the cell increase in size? Yes, the cell will increase in size. But can I technically tell the protoplasm is increasing? Protoplasm is increasing. So can I call it protoplasmic growth is the actual growth? Protoplasm is your living part of cell. So if you want to actually tell of the particular organism is growing, you need to look at the protoplasm. But can we actually measure the protoplasmic growth? Yes, you can, but it is a very difficult process. Protoplasmic growth is very difficult to measure. Very difficult to measure. Very difficult to measure. Very difficult to measure. So do we have alternatives? Yes, we have alternatives. If you do not, if you cannot measure with the help of protoplasm, there are other methods. That is, increase in the cell size and cell number. You can count the number of cells increasing. You can see the number of sizes increasing in the cell size. The example is very important. For example, for a cell number is going to be your maize root apical meristem. Maize root apical meristem, where 17,500 cells are added per hour. What about cell size? Can we see a cell size? Yes. If this is the cell size of the watermelon, all of you know watermelon, right? The same cell can increase in such a bigger size, that is approximately 3.7 lakh times. It can increase its size to 3.7 lakh times. It is increasing in size. That's how big a cell can become. So we can actually measure it. Can we also measure increase in fresh weight and dry weight? Yes. Increase in length. Example is going to be a pollen tube. All of you know, right? Pollen tube length. Pollen tube length can be measured. Increase in surface area. Imagine one leaf is this tiny. The same leaf can become bigger. Surface area can be easily be managed. There is increase in volume. We can also measure the increase in volume. Clear? These are the different methods in which growth can be measured. Now, what about phase of growth? Are there any different phases of growth? See, if this is happening first, then this is happening, and then this is happening. Yes, friends. There are 3 different phases of growth. Can anyone in the chat tell me what are the different phases? For example, if this is a cell and it is dividing into 2 other cells, there are 3 different phases. First one is called as your meristematic phase. Meristematic phase. The second one is called as your elongation phase. Third one is called as your maturation phase. Meristematic, elongation, maturation phase. In these 3 phases, you can see a development of cell. Now let's see the first one, that is called as your meristematic phase. What are the properties? Generally, I would make you write this, but if I write this, it will take a lot of time, and I have made sure some points for you, and these points are going to be important for you. That is, the first point is first phase. Here it is present in the meristematic region. The cells are very small here. It is a dense cytoplasm or protoplast. Vacuoles are absent here. Why? Because it is the initial phase. Do we even require vacuoles? If there is no product, do you need a storehouse? No, we don't need any storehouse. Conspicuous nucleus, cell wall is primary because it is initial phase. Multiple plasmodesmatal connections can be seen in the case of your meristematic phase. What about your elongation phase? In case of your elongation phase, it is the second phase. Here, cell length increases and cell size also increase. You can see increase in the cell size and cell number here. Similarly, vacuoles are present now. Vacuoles are starting to appear now. Then we have secondary cell wall formation. That is, depositions in the cell walls will start. That is your pectin depositions. Yes, depositions in your pectin. Correct? Now students, what about your maturation phase? Maturation phase is the third phase, and here differentiation of cell happens. What happens? Differentiation of cell happens. Now apart from this, are there any regions, are there any things which is happening here? Yes. Apart from these two, differentiation, there are 2 major events. That is, one event is happening in the cell wall. That is, in the cell wall, you can see lignin deposits now. we can identify lignin deposits can be seen here. And similarly, vacuoles will reach maximum size. Vacuoles will reach maximum size, and cell wall will have certain lignin depositions. Apart from this, a cell will have fixed structure and function. Now, fixed structure and function. By the end of your differentiation, a particular cell will have fixed structure and fixed function. Done? Clear? This is what is happening. Protoplasmic modifications, that is lignin depositions in cell wall. Vacuoles will reach the maximum size, and every single cell will have fixed structure and function by now. Clear? Now students, let's understand one small concept called as your distal and proxial. If this is the soil region, we have the roots are developing. We have the taproot. We have this is your root hair development. This is the growing tip called as your meristematic region. Then we have region of elongation. Then we have region of your maturation. Now, if you take soil as your reference, this part becomes distal region, very far from it. Similarly, if we take soil as a reference point, this becomes proximal region. Yes, this becomes proximal region. Why X coming? This becomes proximal region. Now, if I ask you a question, which is the distal region to elongation, you should tell me the distal region to elongation is going to be meristematic region. If I ask you a question, which region is the distal region to maturation, it's elongation. If I ask you a question, which is the proximal region to meristematic region, elongation, proximal region. If I ask you which is the proximal region to elongation, then it is going to be maturation. So you should take soil as a reference point, and the closest part is called as your proximal region, and the furthest part is called as your distal region. Proximal as well as distal region. Clear? See, if you notice this diagram is given you NCERT diagram, this is your three different regions. This is your region of meristematic, region of elongation, region of maturation. That is division of a particular detection of zones of elongation by the parallel line method. Now students, let's get to the growth rate. Let's understand growth rate now. Now, what is this growth rate? Growth rate is basically at which rate growth is happening. Yes, growth rate is basically increased growth rate per unit time. Increase of growth per unit time. Yes, that is your growth. So if you look at it, this class will go up for one more hour. Okay. What is the increase of growth per unit time? That is the growth rate. So if you in your NCERT, there are two types of growth. One is called as your arithmetic growth. The other one is called as your geometric growth. Now, what is this arithmetic growth and what is this geometric growth, students? Arithmetic growth is very simple. In your arithmetic growth, we have one meristematic cell. Yes, we have one meristematic cell. Now this one meristematic cells is divided into two parts. This meristematic cells, when it divides, one cell will remain meristematic, the other cell will become permanent. Similarly, this cell is dividing into two parts. One cell will be meristematic, the other cell will become permanent. Similarly, this cell is dividing, what will happen again? One cell will become meristematic, the other cell will become permanent. So here, out of the two daughter cells, one will remain meristematic, the other will become permanent. The other one will become permanent. Clear? Clear? Oh, you cannot see this. So students, I have written here increase of growth per unit time. I don't know, I cannot reduce the screen now. I don't think I can reduce the screen right now. I can go and reduce the screen right now. So this is growth rate is what? Increase of growth per unit time. I'll give you the notes. Use the notes. Okay. Now students, that is your arithmetic growth. Now what about geometric growth? In the case of geometric growth, if this is your meristematic cell, it is dividing into two daughter cells. Both the cells will remain meristematic. Both the cells will remain meristematic here. Meristematic cell, meristematic cell, meristematic cells, meristematic cells. That is your geometric growth. Now, do we have a graph for it? Yes. If you look at the graph for your arithmetic growth, you can see a linear constant increase in the height of a plant with respect to time, and it is not starting at zero. That is very important. That is a linear constant increase there. The constant increase is found in your arithmetic growth, while your geometric growth it is very weird. Geometric growth will give you a sigmoid curve where we have a lag phase, then we have exponential phase, also called as your log phase, then we have a stationary phase. That is, initially a plant is preparing for growth, then the plant is actually showing the growth, and after some time growth becomes reduced. Why? Because lack of nutrients. Clear? This is an ideal sigmoid growth curve typically of cells in your culture and many higher plants and plant organs. This is linear growth, which is example is for your root elongation. Example here is root elongation. Clear? Arithmetic is going to be your root elongation. Geometric is going to be a most of plants. But is there a formula for this? Yes. For your arithmetic growth, the formula is LT equals L0 plus RT, where LT is equal to length at time t. L0 is going to be length at time 0. R is going to be your growth rate or elongation per unit time. Similarly, your geometric growth also we have a equation, that is W1 is equal to W0 e to the power RT. What is W1? That is the final size. W0 is initial size at the beginning of the time period. R is going to be a growth rate. T is going to be a time of the growth, and E is going to be a base of natural logarithms. Clear, students? Here R can also be called as relative growth rate. Clear? R can be also called as your relative growth rate. Clear? Relative growth rate. Students, is this point clear to all of you? What is relative growth? For what is the meaning of your arithmetic growth? What is the meaning of your geometric growth? Easy. Now students, if you look at the NCERT diagram, can you see one cell is meristematic, the other cell has become permanent. Arithmetic permanent. What about your geometric? All the cells are meristematic region nature. Now here we have a separate example called as your embryo development. That is zygote to embryo. From zygote to embryo formation. The type of growth is geometric growth. When zygote to embryo, geometric growth. From embryo to the rest of the growth is arithmetic. is arithmetic. Clear? You can see inside diagram. Geometric growth. Now after that there is arithmetic phase. Initially there is geometric, then we have arithmetic phase. Clear? Now what are the different ways we can measure growth now? What are the different methods which will help us to identify and measure growth? Can anyone tell me in the chat? How can we measure growth? There is something called as absolute growth. There is one more something called as relative growth. Now what is this absolute growth and what is this relative growth? Let us learn with the help of a simple example. Okay, shall we? A simple example will tell us what is the meaning of your arithmetic, what is the meaning of your absolute growth, and what is the meaning of your relative growth. Now I have always explained this. with a simple example. Always. That is, in absolute growth, it is just final minus initial. That is, if I was 60 kgs last year, now I am 65 kgs. How much of weight I have gained? It is not a growth actually, but example I'm telling you, just for example. I have technically gained 5 kgs. What did you do? You did final minus initial. What did you do? We did final minus initial. Final weight minus initial weight. That is your absolute growth. Direct. It will directly tell you. Absolute growth will directly tell you how much of increase is there. That is, it will show you actual growth. It will show you actual growth. Now, what is relative growth? Relative growth is increase per unit time. And it is always in the terms of percentage. Now here, the example which I'll use is the same example which I always use. Let's take Meghana from your example. Last time I took Ravindra, I still remember. Let's take Meghana from the chat. Okay. Meghana's pocket money, I've told you the same example I used last time also, because this is the best example to explain this. Meghana's pocket money is 5 rupees. Imagine Meghana's pocket money is, alright, Megha, pocket money is gonna be 5 rupees. 5 rupees. Let's just use symbol, easier. Our pocket money is 5 rupees. Now she is fighting in the house. Meghana is fighting in the house and she'll be like, no, I do not want 5 rupees pocket money. I want 10 rupees pocket money. I want my pocket money to be increased. So 5 rupees becomes 10 rupees. 5 rupees is becoming 10 rupees. Her pocket money is increasing. Now let's take one more example from your chat. It is Kushi. Okay. Kushi's pocket money is going to be 50 rupees. Her pocket money is also increasing by 5 rupees. That is 55 rupees. Now you tell me in the chat, who is going to be more happy with the increase? Is it going to be Meghana or is it going to be Kushi? Tell me in the chat, who is going to be more happy? Obviously the Meghana, because her pocket money actually doubled. Right? Her pocket money actually doubled. And how do you identify this? With a simple formula. That is final minus initial divided by initial. That is final minus initial divided by initial. That is 10 divided by 5 10 minus 5 divided by 5. That is going to be your 5 by 5 into 100 into 100. That is going to be your 100%. That is going to be your 100% increase. 100% is increasing here for your Meghna, right? 100% increase. There's a 100% increase there. 100% increase in your pocket money. But what about Kushi? What is final here? 55 minus 50 divided by 50 again. Final minus initial divided by initial. 50 minus 5 is going to be your 5 by 50, which is roughly around 10% increase. Roughly around 10% increase, right? 10% increase. 10% increase. So you see in both the cases, the increase in amount is just 5 rupees. Increase in amount is just 5 rupees. But for Meghna, it is a 100% increase. But for Kushi, it is just a 10% increase. Just 10% increase. That will show you the relative growth. That will show you that is the relative growth. That is relative growth. Yes, sir, come in.
Yo. guys. I'll be coming live soon. Till what time?
I think 1 hour, sir.
1 hour?
Uh-huh.
Okay, you're planning to give a break. You're not hungry.
Sir,
Okay.
sir, you come live. I'll take 10 minutes break if you want.
If you okay. After some 10 minutes, sir.
Huh.
If you want, you take 5 minutes water break.
Huh.
I have a very important announcement to make.
Yes, sir. Okay then.
Yeah. Okay, you just take 5 minutes break.
Okay.
Okay, thank you guys. And look at the Josh, guys. Crazy level of dedication.
Huh.
Keep it up and keep the fire alive. Okay, bye. See you.
I'll say bye. So do you notice, students, it's 9 o'clock and Shreyas sir is here, Diksha ma'am is here, Gopika ma'am is here. Everyone is working around the corner. All of us are working around the corner to make sure you get the best content here. Yes, students. So before we start your PGR, I'll give you a 10 minutes break. Okay, clear? Now, students, where were we? So here for your Kushi it's 10%, for Meghna it is 100%. Now, what is the meaning of this? The meaning of this is that Meghna is 100% increase. She's more happy. That is your relative growth. Let's understand with the help of example. Let's understand with the help of an example that is going to be here. What is the example here, students? Example is your leaf. We have leaf A and we have leaf B. In your leaf A, what is the absolute growth? What is absolute growth? Tell me quickly. Absolute growth is going to be final minus initial. 10 minus 5. That is going to be 5. 10 minus 5 is going to be 5. So the actual increase is how much? 5 centimeter square. Similarly, in the case of your leaf B, it's going to be 55 minus 50. That is going to be again 5 centimeter growth. Yes, final minus initial 5 centimeter square growth. Final minus initial. Similarly, students, let's look at the relative growth now. Relative growth. Now, what is the relative growth here, students? Final minus initial divided by the initial. Final minus initial divided by initial into 100. That is around 5 minus 5 is 5 divided by 5 into 100. That is 100% increase. That is 100% increase in the case of leaf A. What about leaf B? In the case of leaf B, we have final minus initial. Final minus initial divided by initial. That is your minus 5 is 5. 5 divided by 50. That is around 10%. That is around 10%. Now, what is it telling you? It is basically telling you, students, it is basically telling you that leaf A is showing higher relative growth. Leaf B is showing less relative growth. Even though they may look equal from outside, in absolute growth, both are same. But relative growth is going to change. Relative growth is going to change. Now, there are different conditions for growth, students. There are different conditions for growth. That is temperature, nutrients, light, water, and oxygen. If you look specifically, how does the water play a role, students? How does water actually play a role? How does water actually play a role here, students? In case of water, turgidity of cell helps in extension growth. Similarly, water also plays a major role in your growth of a plant because it is a medium for your enzymatic action. Done. How does oxygen play a role in the growth of a plant? That is releasing metabolic energy. Nutrients, all of us know, that is micronutrients and macronutrients. Micro as well as macronutrients, which play a very crucial role in the growth of a plant. Then we also have temperature, light and gravity. These are the different factors in which influences a particular growth of a plant. Clear, students? Now, the next thing which you need to understand is differentiation, dedifferentiation and redifferentiation. Clear? Let's understand differentiation, redifferentiation, redifferentiation. After that, we need to understand development. So after this, we will take a 10 minutes break because Shreyas sir has to come and tell you a small surprise. Okay. Yes. So can we start now? Differentiation, dedifferentiation, redifferentiation. After this, we'll take a 10 minutes break and Shreyas sir will be here. Okay. Let's understand, students. Let's understand differentiation. Let's understand differentiation. Now, what is happening in differentiation? Imagine we have a meristematic cell. The meristematic cell will completely lose its ability to divide. Completely loses ability to divide, and it will become a differentiated cell. It will become a differentiated cell. And this differentiated cell is called as your primary permanent cell. Example is going to be your primary xylem, primary phloem, as well as your primary phloem, and we can also say primary medullary race. Now, how will you remember this, students? How will you remember this? Imagine there is one student after class 12th. There is one student after class 12th. He cannot study further because there is some financial condition in his house. He is not lucky as you. You can study, you are preparing for Neet. He is not lucky as you. After class 12th, he's like, I cannot study further. He will get a permanent job and he will become permanent. So he will lose its ability to study here. That student will lose its ability to study. One student, because of the financial conditions, he cannot study further. He will lose his study and he will get a job called as your he will get a permanent job called as permanent job. That is what is happening here. Example is going to be meristematic cells lose its ability to divide, become differentiated, and it will become a primary permanent cell. Similarly, let's understand dedifferentiation. What is happening in your dedifferentiation? What is happening in differentiation? The permanent cell here, the permanent cell will regain ability to divide. It will regain its ability to divide and it will become secondary meristem. It will become secondary meristem. Remember the student here who had to stop studying, who had to get a permanent job. After some years, he is like, okay, the family is all right now. The family is able to survive. What does he decide? He decides that he starts to study again. That is, regain ability to study. And once he joins a college, like a BSc course or something, then he becomes secondary meristem. And remember, students, secondary meristem has ability to divide. Secondary meristem has ability to divide. Yes. Example is going to be your medullary race here. Medullary race here, same. The medullary race here can become vascular cambium. Can become vascular cambium. That is your dedifferentiation. Now, what is this redifferentiation, students? What is this redifferentiation? Third one. What is this redifferentiation? Now, imagine this is your secondary meristem. Imagine if this secondary meristem will lose its ability to divide, completely, and it perform specific function. It will perform a very specific function. This secondary meristem loses ability to divide and perform a very specific function, and it will produce a secondary tissue. Secondary tissue that is called as your redifferentiation. Imagine a student who went to college here. He decides, no, I don't want to study any further. I want to do my old first dream. That is, he decides to become a musician again. That is what is happening here. Redifferentiation is lose ability to divide and perform a specific function. Secondary meristem will losing ability to divide and it is becoming a secondary tissue. It is becoming a secondary tissue. That is, vascular cambium will produce your secondary xylem. Produce your secondary xylem. There it is, students. There it is. There it is. Clear? Clear? Secondary xylem, secondary phloem. That is your dedifferentiation, dedifferentiation and redifferentiation. And remember, this happens in both monocot and dicot. Monocot and dicot. What about dedifferentiation? Dedifferentiation only happens in case of your dicots. It only happens in the case of your dicots. Clear? Clear, students? Clear? Yes. This is how you should learn, students. I want everyone to write down this. I want everyone to write down this and learn. Write down and learn. Then only you will remember it better. Then only you will remember it better. Okay. Now, students, I think Shreyas wants to go live. Shreyas sir has to go live right now, I think. Let us give a 5 or 10 minutes break. Students, after this we have development. Well, I'll teach you development. Then I'll teach you plasticity. What is the meaning of plasticity? And then after that, I will teach you every single, students, see this. Every single hormone. Do you see this? Every single hormone I've written. What is happening? Cytokine, what is happening? Every single hormone, students, every single hormone function I've mentioned. Every single hormone in the end of the session. In the end of the session, I'll give you a complete summary. Type, function, gibberellin, function, cytokinins, function, ethylene, function. You do not have to go through the entire NCERT later on. All you can do is flip the slides, which I will give you, and you can see what is happening in cytokinins, ethylene. Every single thing will be given in your notes. Everything will be given in the notes. So don't worry, the entire process of your remembering the hormone function is going to be very easy. And I'll make sure it becomes very easy because I have made the perfect notes for you. Okay? Clear, students? Clear? So students, after that, let's take 10 minutes break now. 10 minutes break. I want Shreyas to go live. Shreyas can go live right now, and we'll come back and we'll do development, plasticity, and hormones. Okay? That will approximately take around 40 minutes, maximum 40 minutes, done and dusted. Okay? 40 minutes, done and dusted. Clear? So I'll just call Shreyas sir. I'll raise the outside. Probably he's waiting outside. I'll take Shreyas sir. Okay? Shreyas sir left. Students, Capto is coming. So, Sir is coming. If while we wait for Sir. Check, sound check. Can you hear me, students? Can you hear me, students? Can you hear me? Check. Capto is coming. Sir is coming. I think students. Do you don't believe me? Sir is actually, if you go check right now, Vedantu JE, he's teaching there also. Do you realize Sir is actually teaching on Vedantu JE? And he'll come back here and teach on Vedantu NEET English. He was in English in the morning. So Sir is doing so much of multitask today, and I'm baffled by how can Sir manage so much, right? So let's wait for Sir to come. While we wait for Sir to come and join, like, he's live everywhere. I can see Sir live everywhere. Like, he's here, he's there. I don't know where to find Sir now. Okay, students, before we, by the time Sir joins, do you want me to teach you what is development? What is development? Friends, what is development? Development is very simple. I can tell you 1 or 2 lines of development as you will understand very well. You will understand very well. Students, development is nothing but all changes that occur in the plant's life cycle. All the changes that happens in a plant life cycle is called as your development. What is called as your development? If you look closely, if this is your seed, this seed is germinating. Can you see a radical development here? This seed is germinating and it will show radical. Later it will form a juvenile phase. It will form a juvenile phase. Now in this juvenile phase, can it do reproduction? No, it cannot do reproduction in the juvenile phase. Not possible. Juvenile phase reproduction is not possible. Only it's also called as your vegetative phase. Also called as your vegetative phase. Now students, this juvenile phase later becomes mature phase. Now in this mature phase, what happens? Reproductive organs develop. Reproductive organs developing. So particular plant can do flowering. Plant can do flowering. The same plant after flowering, it will reach old age. It can undergo senescence. The same plant can later on, same plant will later die also. That is seed, cotyledon development, juvenile phase, mature phase, flowering, senescence and death. This is the life cycle of plant. If you look at the diagram of your NCERT, sequence of development of process in your plant life cycle, we have meristematic cells. We can see plasmatic growth, increase in the cytoplasm. Then we have differentiation. Then we have elongation. Then we have maturation. Yes, see, remember, phase of growth, meristematic phase, elongation phase, maturation phase. Then we have mature cell, senescence, finally death of a particular cell. This is a proper life cycle of a cell. Now students, if a particular cell is going away from it, is see, right now it has a life cycle. It has a particular life cycle. Now what if a particular plant is deviating from its life cycle? It is forming an alternate life cycle. That is called as your plasticity. Clear? That is, the ability of plants to alter their development is in response to environment conditions as well as the phase of life. That is called as your plasticity. We have one proper life cycle. If a particular plant is deviating from that life cycle, like due to some environmental problems, environmental issues, or due to different phase, that is called as plasticity. With response to environment and life and phases, they change their life cycle only. Example is going to be very important. The example is your heterophyly. That is, production of different leaves forms on the same plant during the different phase of life cycle or different environmental condition. Listen to me very carefully. Example, there are two different types here. There are two different types here. One is based on the phase, the other one is based on the environment. Based on the environment. There are some cases, there are some juvenile plants, some juvenile plants, if it has a leaf like this, imagine if there is a juvenile plant, it has a leaf like this. In adult stage, the leaf will look like this. The leaf will completely change. The leaf will completely change in adult stage. The leaf is completely changing. See, the leaf was like this in the juvenile phase, but in adult stage, the leaf is completely changing. That is, the life cycle of particular plant completely modified based on the different phase of life. Juvenile phase, leaf like this. In adult phase, leaf is looking completely different. Example is going to be your cotton, coriander and larkspur. Three examples, very important. Cotton, coriander and larkspur. Similarly, due to environment, imagine there's one leaf which looks like this. Imagine a leaf will look like this. Leaves are very narrow leaves. Why? In aquatic conditions, students, in aquatic conditions, if water is flowing, if it is very thin leaves, will the water stop the leaf? No. The water will easily pass through. Water will easily pass through and it will not damage the plant. It will not damage the plant. Imagine the same plant in terrestrial ecosystem. In terrestrial ecosystem, it has very broad leaves. It has very broad leaves. So it can do photosynthesis very well. Example for this type of modification based on environment is going to be buttercup. It's going to be your buttercup. It's going to be buttercup. Clear, students? So now we'll take a 10 minutes break so Shreyas sir can go live. If you have any doubt in this particular plant, let me know in the comment section. Buttercup for your environment, for your juvenile, and this is going to be your cotton, coriander and larkspur. After this is the example given, you see, terrestrial, aquatic, juvenile, adult. Now once we come back, we'll be doing your development, that is growth plus differentiation. Okay, clear? Also called as ranunculase. Yes. I'll be coming back in 2 minutes. 5 minutes. I'll see you on Shreyas sir's live chat. Students, how was the update? Students, how was the update? Students, are you excited? Because the NEET game is gonna change now. Now you will see more and more students registering. Because I still remember on a Telegram, I got a lot of messages saying that, sir, I could not, register on time. My 2 years of preparation is going waste. Now this is for all the students. It's good news. If you were not able to register before, please register to your NEET examination right now for this next open window. Yes. Hello. Students, are you excited for the last part of the chapter? Because I am. Even after the 5 hours of class, I am still here and I'm going to teach you the last part of the chapter. That's going to be your development. It is growth plus differentiation. Growth plus differentiation. Now students, your entire development can be divided into 2 parts. Development of a particular plant, it can be divided into 2 parts. One is called as your extrinsic factor. One is based on your eccentric factors. Other one is based on your intrinsic factors. Now, what are these eccentric factors, students? These eccentric factors are mainly your water. All of you know. Water, oxygen, temperature, gravity. Yes, temperature, light, as well as nutrients. Now, what are these intrinsic factors? Intrinsic factors can again divide into 2 types. Intrinsic factors can be again divided into two parts. That is gonna be your intracellular, as well as intercellular. Now, what is this intracellular, students? Intracellular are technically the genetic factors which determine the development. That is, it is present in the DNA of the plant that this plant has to grow this much. It is present in the DNA. Now, what is this intercellular factors? Intercellular factors includes your PGR. Now, what is this PGR, students? PGR is your plant growth regulators. Are your plant growth regulators or plant growth hormones. Now, what are these plant hormones, students? What are these plant hormones? Plant hormones are technically chemicals. Yes, plant hormones are technically chemicals that control physiological processes. Now, what are these physiological processes, students? These physiological processes can include like cell division, cell elongation, cell division, as well as your cell maturation. Clear, students? That is, cell elongation, cell maturation, and cell division can be controlled with the help of hormones. And just like your human hormones, plant hormones are also produced in your low concentration. Very low concentration they're produced. Okay, very low concentration they're produced. Now, students, if you want to classify your plant growth regulators into 3 different categories. We can divide the entire plant growth regulators into 3 different categories. The first one is going to be your plant growth promoters. The second one is going to be your growth inhibitors. The last one is going to be both. That is, they have a dual function. They can inhibit a growth or they can also promote a growth. The example for your growth promoters are going to be auxins, gibberellins, as well as cytokinins. For your growth inhibitors, we have ABA, abscisic acid. For both category, we have the ethylene. Which is the gaseous hormone, which has both promotion as well as inhibition activity. Now, students, one by one, can we go auxin, gibberellin, cytokinins, ethylene and abscisic acid. One by one, I'll be telling you every single point which you need to remember for your photosynthesis. So can we start? Can we start, students? Can we start with the last part of this plant physiology, which will actually fetch you a lot of marks? I'm telling you, students, the plant physiology, the last part, the plant growth regulators, definitely one question will come in the next 30 minutes. If you give me your next 30 minutes of your life, I'll make sure you will get some amount of marks in your NEET examination from the following topics. Ready? Ready, students? Is everyone ready? I want some josh in the chat. I want everyone to quick fire in the chat. I will know that you are excited. And if you are new to the channel, quickly like the video and subscribe to the channel, and we'll go ahead. Yes? The first one is going to be your auxin, students. Auxin is a word which is derived from a Greek word called as your auxin, which means to grow. Which means to grow, right? And the chemical nature of your auxin is going to be your indole compounds. The chemical nature of your auxin is going to be indole compounds. What is the precursor? The precursor is going to be your tryptophan. Tryptophan is a type of amino acid. Remember that. Tryptophan is a type of your amino acid. Now, students, auxin are broadly classified into two categories. The first category is going to be your natural auxin, that is indole 3 acetic acid, indole 3 butyric acid, 4-chloroindole 3 acetic acid. These type of acids are your natural occurring auxins. Similarly, we also have your synthetic auxin. What is the synthetic auxin, students? Synthetic auxin mainly includes your naphthalene acetic acid, 2,4 dichlorophenol acetic acid. We also have 2,4 trichlorophenoxyacetic acid. You might have heard it as 2,4D, yes, or 2,4,5T. Now, what are this? These are basically VD sites. They're VD sites. Okay, students? Now, who discovered this? Now, who discovered the auxin? The discovery of auxin was done with the help of two scientists, father and son, that is Charles Darwin as well as your Francis Darwin. Charles Darwin and Francis Darwin, I discovered your auxin. That is in the plant canary grass. The topic which they are trying to study, the topic which they were trying to study is your phototropism. That is, they took a canary grass. And they noticed the canary grass is, if this is the sun, the canary grass is growing towards the sun. And they identified there is something in the canary grass. There is something in the canary grass through which a grass is moving towards the sunlight. So what do? They cut this part now. In the next plant, they cut this part, completely decapitated this part. Now, did they notice a particular curvature? No. When the head region was completely cut, there was no curvature. There was no curvature. No curvature at all. Similarly, what happens, students? Similarly, what happens? They took the same avina, they took the same canary grass, and they completely covered the top with a black color paper. Even then, there was no curvature. So this basically tells you that there is something in the tip region. With the help of that, a canary grass is bending towards the light. Now you tell me in the chat, who was the person who did isolation? See, discovery was done by Charles Darwin and Francis Darwin, but the isolation was done by your F.W. Wendt. Isolation was done with the help by F.W. Wendt. The plant he used was Avena sativa, that is called as oats. The bioassay is called as Avena curvature test. So he took a oats plant. And he also identified there is something in this region. Which is able to produce a hormone which is bending the plant. Now, students, what did he do? He took this top part and he added this top part in a agar-agar media. This is your agar-agar media. He added here. Now, whatever was present in this particular tip, everything diffused into the agar-agar media. Everything diffused into agar-agar media. Now we have agar-agar media. Now we have a agar-agar media which has your hormone. Imagine we have two plants now. Imagine we have two plants here. Two plants. Plant number A, plant number B. Plant number A and plant number B. Now we are taking this agar block. Now we are taking this agar block and we are adding the agar block here. Similarly, we are taking this block, agar block, and we are adding it here. What do we notice? If auxin is present here, auxin will promote growth. So this particular part will grow more. So you will see a growth this side. Similarly, if auxin is present on this side, this region will grow more, and you will see the canary grass moving to the opposite direction. That is the bioassay. That is the bioassay, students. Done? Did you understand, students? Did you understand how it happens in your bioassay, in your Avena curvature test, students? Done? Now let's focus on the functions. Let's focus on functions, all of you. Now, what is the main function and what are the other commercial applications? I'll tell you in the next 2 minutes. The main function of your auxin is going to be apical dominance, feminizing effect, that is, it produces carpels, cell division, cell enlargement, root initiation, flower initiation, parthenocarpy, that is seedless fruits, prevention of abscission. Prevention of abscission. Clear? That is the main functions of your auxins. Main functions of your auxins. Clear? Main function of your auxin. Apart from this, is there any other function of auxin? Is there any other extra function of your auxin? Can anyone tell me in the chat? Students, remember, this inducing flower is there. Yes, flower initiation is gonna be in your pineapple. Pineapple. It is given in NCERT. Pineapple. Now, what about your apical dominance? Apical dominance, which is found in your apical dominance, can be used for growing tall plants. Now, this prevent of abscission is very peculiar here. That is, prevention of abscission is mainly done in your younger plants. Mainly done in your younger plants. What if a plant is old? What if a plant is old? In those cases, auxin will not prevent abscission. If a plant is already old, if a leaf is already old, it will completely kill the leaf. It will completely kill the leaf and the leaf will fall down. And the leaf will fall down in the old leaf. But if it is a younger plant or younger parts, auxin will do what? Prevent abscission. Auxin will prevent abscission. In old leaves, auxin will accelerate acceleration. In case of old leaves, auxin will accelerate abscission. Clear? That is very important. Now, students, that's all. There's nothing. I think we covered every single point. The next one is going to be your gibberellins. Next one is going to be your gibberellins. Listen to me very carefully, everyone. What is the chemical nature of gibberellins? Terpenes and terpenoids. Precursor is going to be your acetyl-CoA and mimonic acid. Discovery was done by Kurosawa in 1962. Which plant? Plant was rice plant, and the disease was called as Bakane disease, also called as foolish disease. Now, why is it called as Bakane disease? Because imagine you see a tall plant and there is no fruit on it or there is no seed on it. You will laugh at the plant. No, the plant is so tall, but there is no seed on it. That was noticed in your rice plant. That is, the rice plant is growing so tall, there was no seeds at all. There were no seeds at all there. That is Bakane disease. Isolation was done by Yabuta and Smoky. Bioassay is called as alpha amylase barley endosperm test. Not in a disease, not in your NCERT, but remember that. Now, what is the main cause of this elongation which they observed in your rice plant? The main cause of this elongation was a fungus. And the fungus name is called as Gibberella fugicori. Gibberella fugicori. Can you see the ice in this internodal region? In this internodal regions, can you see there is accumulation of this fungus? And because of this fungus itself, there is increased concentration of gibberellin and there is internodal elongation. There is internodal elongation. See here, more than 100 gibberellins, are reported throughout the world, denoted as GA1, GA2, GA3, and the most popular one was your GA3, that is gibberellic acid. Gibberellic acid was first gibberellin to be discovered. Very important. Very important. The first gibberellin to be isolated was your GA3, called as your gibberellic acid. Now, what are the general characters here? Listen to me very carefully, students. Listen to me very carefully. All gibberellic acids are acidic in nature. Point number 1. Transported either through xylem or phloem. They are formed in the plastids. They are formed in the plastids. Now, if you look at the functions, it stimulates stem growth, promotes seed germination. The seed will start to germinate here, right? Say germinates. We have bolting in rosette plants. Now, what is the meaning of bolting in rosette plants? Listen to me very carefully, students. If this is your rosette plant, such as your cauliflower and cabbage, there will be elongation of stem here. There will be elongation of stem in your rosette plants. Okay. Then we have delay senescence, that is your function of your gibberellins. But apart from this, your NCERT also tells you other different functions. That is increase in length in your grape stalk. That is, if this is the grape stalk, imagine if you spray some gibberellin, this grape stalk will increase so much. If the stalk is increasing, grapes are also increasing. Yes, with the help of gibberellins. Clear? Increase in length of stem, thus increase in yield. Example is case of sugarcane, increase yield as much as 20 tons per acres. That is, if you add more and more gibberellins, the entire sugarcane will become very tall. So more food, more sugarcane. Elongate and improvise shape of apples, speeding up the malting process in your brewing industry, that is in alcoholic industry. Spraying juvenile conifers with gibberellic acid hastens the maturity period, that is, they become mature very faster. Students, every single question here, every single point here can be asked as a question. I am not telling you just for the sake of it. Students, every single point here can be asked as a separate questions. So please remember that. Okay, please remember that. Now, next one is your cytokinins. What is the chemical nature of cytokinin, students? It's a purine derivative, that is adenine. Precursor is your tRNA. Discovery was by Skoog and his co-workers. Isolation by Miller et al. Isolation was done by Miller. Now, how did they discover this? When Skoog was doing his experiments, when Skoog was doing his plant biotic experiments, he noticed that in your extracts of, whenever he did the experiment, when he added only auxin, growth was not happening very well. But along with auxin, when he added extracts of your vascular plant, yeast extract, as well as coconut milk, that is, all of this plus auxin, he noticed that the callus formation is happening very well. Then he noticed something is present in coconut milk, something is present in your yeast extract, something is present in your extracts of your vascular tissues, which is actually doing cell division. And later it was identified by Miller in your herring fish sperm. In the herring fish sperm, that there is something called as your kinetin. There's something called as kinetin, which is a type of your cytokinins, which is actually doing cell division. Which is doing what? Cell division. See here, Miller et al. Later identified the crystallized the cytokinins promoting the active substance that is called as kinetin. Now this kinetin was obtained from your herringfish sperm. What about plants? How did we obtain in plants? In the case of plants, it was obtained from your corn kernel as well as coconut milk. That is, cytokinins along with auxin promote your cell division. Natural substance with zeatin. Zeatin was obtained from your maize plant. Zeatin is obtained from your maize plant. Clear, students? That is your cytokinins. Now if you ask me the function, what is the function of your cytokinins? All of you know the main function of your cytokinins is cell division. Yes, cell division. Modify apical dominance and promote lateral growths. So can I tell cytokinins, this function is actually opposite to the auxin function? Because auxin will do what? Apical dominance. In the case of your cytokinins, there is what? Lateral growth promotion. There is lateral growth promotion. Then we have promotes nutrient mobilization, promote chloroplast development, and also helps in producing new leaves and adventitious shoots. Adventitious shoots. Then we have ethylene. Students, remember, ethylene has a dual function. It can act as a promoter as well as a inhibitor. And remember, this is the only hormone which is gaseous in nature. It is gaseous in nature. That is, ethylene is a gaseous hormone. Precursor is going to be methionine. Again, students, methionine is the amino acid. If they can ask you a question, which are the two hormones where the precursor is the amino acid? What is the answer? The answer is going to be ethylene as well as auxin. Auxin, remember, auxin precursor was tryptophan. We have discovery by H.S. Cousins. Bioassay is called as triple response test. Students, trust me, when you have information like this, it is easy to study. It's very easy to study. In your NCERT, it is so messed up. NCERT is giving you discovery first. Later on, they're giving you the information. But when you have information like this, it becomes very, very easy to understand and study. Now, let's look at the effects of ethylene. Let's look at the effects of your ethylene. The main function, that is effects on seed and seedlings, that is breaks seed dormancy. It is breaking the seed dormancy, initiating germination, horizontal growth in case of seedlings, swelling of embryonal axis, apical hook formation in your dicot seeds. What about in the case of shoot? How is it making shoot development? That is, it is break bud dormancy. It is breaking the bud dormancy in the case of your shoot. Sprouting the potato tubers, internode and petiole elongation in your deep water rice plants. This again is the PYQ, students. Every single line here is a PYQ. They can ask you a question, and I'm very sure from the points given here, one question will definitely come in your NEET examination. One question will definitely come. Okay. Then we have root. In case of root, it promotes root elongation and its formation of root hair. Now this is very important. Root hair formation in the case of your ethylene. Root formation, auxin. Remember that. And also cytokinins. Then we have ethylene effect on flower. That is, it induces flowering in your pineapple and apple. Again, PYQ. Promotes female flowering in your cucumber and hence increases the yield in case of your cucumber. Students, I want every student to actually see. There's only I can tell you what is important here. I can't make you remember this, right? I want every one student to download this PDF of this particular notes and go through the notes once and understand and remember all this information. Okay? And I have made sure point wise which will benefit every student. If you can't read the NCERT and make your notes from that, I have made notes for you. You realize? And everything. Similarly, what is the effect of ethylene on the fruit? What is the effect of ethylene on fruit? That is commercially used to ripen fruits, after picking, that is ethophon is a source of ethylene. The plant converts ethophon to ethylene. Ethophon to ethylene. Now, effect on various organs, it can promote senescence and also abscission. It will promote senescence as well as promote abscission. Yes, we can make the horse. I cannot make the horse drink the water. I can only take the water to the horse. I can only give you the notes. I can't make you read the notes, right? I can't make you read the notes. I can give you the information. It is up to you if you want to utilize the information, if you want to take the advantages of such beautiful notes. That is that is up to you, right? Students, before we go to abscisic acid, I think I forgot to mention one point here. That is going to be your discovery. How did they discover here? HS cousins. How did they discover? Simple. They took a orange. Tell me in the chat how many of you like oranges, because I don't like oranges. I can't eat oranges. They took a orange which is very ripen orange. Then they took a raw bananas. They took raw bananas. Now what they notice, after a few days, something from orange, some gas from orange converted the raw bananas into ripe bananas, ripened bananas. Raw bananas to ripened bananas. That was noticed by your HS cousins. Clear? The last one is going to be your abscisic acid. Now, what is the chemical nature? Catenoids. Precursor is going to be your viroxanthan, which is a type of your xanthophyll. Discovery, abscissin, dormin, and inhibitor B. That is, discovery was done independently by 3 different scientists. Discovery was done with the help of the 3 different scientists. The 3 different scientists call it different names. One scientist call it abscissin 2, one scientist call it dormin, and one scientist called it inhibitor B. Later they identified all these 3 different substance is actually abscisic acid. It is actually abscisic acid itself. Clear? Now, what is the effects of abscisic acid, students? This is very important. What are the different effects of your abscisic acid? For example, the first one is increase tolerance to stress. That is why it's called a stress hormone. In general, plants grow inhibitor. It is a growth inhibitor of plant metabolism, seed development and maturation. That is, it promotes seed dormancy and inhibitor of germination. It is promoting seed dormancy. Stimulates closure of stomata. Every single time there is stress, stomata is being closed. Now tell me in the chat if it is promoting seed dormancy, right? Promotes seed dormancy and inhibition of germination. It is inhibiting germination. Can I call it antagonist to gibberellins? Because gibberellins are promoting seed dormancy. Yes, antagonist to gibberellins. There it is, students. Every single point of your NCERT has been covered here. Every single NCERT diagram is covered here, students. The only thing which you need to do right now is download these notes. And see here, auxin functions. Every single function is given here. Gibberellin functions. Breaks bud dormancy. Promotes seed germination. Seed promotes seed germination. And we learned here it inhibits germination. Obviously, so your ABA is antagonist to gibberellins. Similarly, we have cytokinins functions, ethylene functions, as well as your function of your abscisic acid in a form of a table. Students. So what is left now? All you need to do is download these notes and tell me in the chat confidently, plant physiology done and dusted. I want every single student in the chat to tell me today, every single student who is watching right now, regardless of how much you watched, how much you were talking in the chat, I want every single student to actually go to the comment section and tell plant physiology done and dusted. And with the help of this session, you can revise your entire plant physiology in one go. Okay. Students, this took a lot of time and effort. So I want you to let me know in the comment section if you have any doubts. Let me know in the comment section. Okay. And I will personally reply to every single message and answer your doubt. Okay. Students, and where will you find these notes? Students? These notes will be found in the Telegram channel. Neet English Telegram channel. And you can also join my Telegram channel if you want botany tips. If you want any type of botany tips, you can join my Telegram channel. Let us Basavaraj Sir Biology. If you type Basavaraj Sir Biology, you will get my Telegram channel. And with that being said, students, your entire plant physiology is done and dusted. Mic drop. Done. Entire plant physiology. Do you realize? We started the session at 3 p.m. 3 p.m. to right now we finished it. Completely done and dusted. I know we took a breaks in the middle and I will trim out the breaks later on. But entire plant physiology as promised is over. And I will be taking, if you are, you might be wondering, sir, where are the questions, right? Where are the questions? Don't worry, I'll be doing the questions on YouTube. But if you are a crash course student, tell me in the chat. If you are a crash course student, I will be doing PYQs and the most important questions from plant physiology on crash course tomorrow live at 10 a.m. Tomorrow on crash course I'll be doing questions. And if you're not enrolled to crash course right now, you can enroll the crash course right now. There's a link in the description. Go on, click on the link, join the crash course. Do you know the price of crash course? The price of the crash course is going to be 999. Join the crash course and you can join my lecture tomorrow 10 a.m. on crash course. Okay. Done. Students, let me know in the comment section how the session was, and do not forget to like the video and share the video, and good night. Bye bye students, all of you. Take care.